of this process, and my project is on spontaneous activity role on cortical development.
Good. Elena?
I'm a postdoc at the Sainsbury Wellcome Centre at UCL. I did my PhD in systems neuroscience in looking at cortical states in mice, and now I've kind of switched fields a bit, and I'm working with zebrafish because I think it's really cool that you can look at the entire brain at the same time. And I kind of would like to look at kind of development of neural circuits and how kind of environmental perturbations can, affect that.
Dean?
Hi, I'm Sean. I'm working in Trinity College. I just started a PhD, and I'll be looking mostly at computational techniques in neuroscience. And I'm going to start letting people in as well. We don't need to start talking yet, but I'm going to start letting people in.
Okay. Should we start?
Yeah, there's a few people joining now. I'm going to turn my video off and stuff, but thanks everyone. I'll be in the chat.
Thanks, Dean.
Thanks.
Thanks. Welcome everyone to Neuromatch 3.0 Day 4. Today we have a very amazing line of speakers in this particular session, and we will be starting it up with Sarah. She is working at Manitas University. Sarah, please introduce yourself and start up with the talk.
Okay, hello everyone, I'm Sara. I'm postdoc in Simona Lodato's lab in Milan. And we in the lab study cortical development. Sorry, I can, okay. The cortical development, as you know, cerebral cortex is a very complex structure in the dorsal forebrain and is composed by a huge variety of neurons. Among them, the 80% of neurons are excitatory projection neuron, while the remaining 20% are inhibitory GABAergic neuron. As you can see in the picture, this neuron can be further classified based on their morphology, their laminar position, their electrophysiological properties, and most importantly, by their molecular signature. It is now well known that the diversity of neurons in the cortex arises during development. In particular, the projection neuron and interneuron derives from two different and distant structures of the brain. In particular, projection neuron originate from the dorsal pallium of the embryonic brain, while interneuron originate from the ventral part. During development, the neuron migrate, invade the cortical plate to form the functional unit of the cerebral cortex, that is the cortical microcircuits. A lot of papers have described the genetics behind the subtype specification of neuron in the cortex. And now we know a lot of transcription factor and pathways that are responsible for different fate acquisition. Interestingly, recently has been shown that also environment play a role in the modulation of the fate of a cerebral cortex neuron. And most interestingly for me, it has been also shown that activity that has been always studied for in the context of neuronal maturation seems to have a role also during development. In agreement with this suggestion, it has been also shown that ion channels, that are the molecules that are responsible for the electrical properties of neuron, are expressed also early during development in the embryonic and the fetal stages. And also it has been shown that mutation in the ion channel that are expressed very early, are responsible and associated with a very rare form of epilepsy, the neonatal epilepsies, that are almost always coupled with brain malformation, corroborating the hypothesis that activity plays a role also in the early phases of development. Nevertheless, the molecular correlation with the spontaneous and early activity and molecular pathways is still poorly understood. That's why the aim of my project is to link the spontaneous activity of the embryonic brain with the acquisition of neuronal diversity in the cortex and the microcircuit assembly by studying the activity genetic crosstalk that acts early during development. On the other hand, I'm also interested in understanding if a spontaneous activity is itself a subtype-specific feature by studying ion channel expression in different subtypes of neurons. So to do that, I took advantage of two tools, molecular tools. One is a channel called KIR, that is known that in the mature neurons is able to, reduce excitability, and the other one is NAC channel that instead enhance the excitability of mature neurons. I overexpress this channel early during development through endothelial electroporation because it is known that this channel have also an effect on spontaneous activity. And I collect the samples at birth. And as you can see, especially in the case of NAC, so in the case of enhanced excitability, there is a problem in cortical development. Indeed, the neurons migrate poorly. So because I'm interested in the molecular crosstalk, I took these samples and I sequenced the samples at single cell level. And now we are performing differential gene expression analysis to unravel activity-dependent pathways that acts early during development. On the other hand, I'm also interested in understanding if spontaneous activity is a subtype-specific feature. So for this reason, I took advantage of a data set we have in the lab in which we have purified 3 different subtypes of neuron of the cortex and sequence we performed RNA-6. I took the signature genes that are the genes that are expressed selectively in one subtype versus the other 2. And interestingly, I found that inside this data set, I found a lot of ion channels that are specific for each subtypes. So now I'm selecting this channel and I'm validating if they have an effect on spontaneous activity. And I'm doing this screening in vitro in primary cortical neurons by using a pharmacological approach, inhibiting this channel, or using a genetic approach by knocking down overexpressed them in primary cortical neuron. And on the other hand, because I'm interested in the crosstalk, I'm also perform in vivo validation by performing couple molecular and electrical analysis of the selected channels. I thank my boss for the help and the support of the laboratory members, the collaborators, and also you for your attention. Thanks.
Thank you, Sarah. It was really a wonderful talk. Do we have any questions from the audience? In the case you want to come up and ask directly, you can definitely tell. You can raise your hands or just put up a chat that you want to speak. Any questions from the panelists?
Yeah, I kind of, if I
please,
can ask a
Alina.
question. So on your last slide, you showed that you do, I think, GChem imaging. I was just wondering at what time point you're doing that and how you're doing that. Because, I mean, the mice are really, really small. So
yeah. I'm performing calcium imaging in 2 ways. First of all, in vitro, in primary cortical neuron, by overexpressing GCAM proteins and performing a live recording on confocal microscope. And I'm particularly interested in these very little calcium transients that are defined like local miniature calcium transients. In vivo, I'm just started to do this, and I also do two ways of recording. The first one is on section by using a calcium dye that is called Cal 520. And the second approach is to perform in utero electroporation of GCAM protein and performing 2-photon microscope analysis. And for the moment, I can able to do it, this analysis only at P0, but it is also possible to do it at, in embryonic stages.
Great. I have a question, Sarah. Is the data that you have gathered, is that available for public use? If yes, all of us.
Paper regarding my project or the field in general?
The data that you have used in today's presentation, basically.
No, these are all unpublished data.
Okay.
Only this work is on preparation, but is not published yet. The work regarding the RNA-seq, this one.
Okay, so we'll be looking forward to your published paper, and hopefully we will be getting the public data as well. Thank you so much, Sarah. If we have any further questions from the audience, anyone who would like to ask something? Okay, take your time. We can definitely come back to Sarah. She's here with us for the whole session. And we have our next talk out here. The next talk is by Tamara. She's going to speak about astrocytic mitochondria, regulates astrocytes, morphogenesis, and supports synaptogenesis. She's from the Department of Fundamental Neuroscience. Tamara, please introduce yourself and start with the talk.
I have to unmute myself. So thank you very much to give the opportunity to present my work. So as you said, I work on the astrocyte during the postnatal development at the university. I was working at the University of Lausanne. So in the lab, we are interested to understand what is happening during the postnatal maturation in the astrocyte. So as you know, there is different wave of generation of cells in the brain, especially in the cortex, and astrocytes are generated in the late phase of the embryonic phase. And when they reach the cortex, they develop their population by local proliferation. And in this moment, they expand their population 6 to 8-fold, always in the cortex. This period is really important because there is this bidirectional communication of neurons with neurons. So not only astrocytes can sense the neuronal activity, but they can also respond to them. And indeed, you can see that astrocytes start to expand their population right before the moment that there is the synapses initiation and maturation. And after we have the coincident moment when astrocytes have the mature morphology and also neuron. So in this project, we were looking at some parameters to characterize the maturation of the astrocyte. And one was the morphogenesis. So as you know, as you can clearly see here, astrocytes, when they are born, they are really small. They have not a lot of branches, but when they become, during the postnatal development, they become bigger and bigger, and they expand their arborization. Another important feature is the non-overlapping territory, meaning that astrocytes, when they are burned, they are overlapping between them. They are really near if you look at the cell bodies. But after they separate and they have these so-called non-overlapping territories, and they overlap of about 5% in order to communicate between them. Another feature can be the gene rearrangement. So astrocytes have some genes that are really high during the period of this first or second postnatal week, and after they go down, and other that are lower at the start of the postnatal development, and they go up after. For us, it was interesting to understand if in this period there is distinct demands of availability of energy, because astrocytes don't have only to support themselves to go from the proliferation to the differentiation phase, but also the neuronal activity and the synapses formation. And we know that astrocytes during the adult age, they are mostly glycolytic, but we don't know what is happening during the postnatal development. So we started with screening, and we did some fact sorting of the astrocyte, and we look at the gene expression for some gene of mitochondrial metabolism and glycolysis. And as you can clearly see here, during the third week of postnatal development, we have an increase in the gene for the mitochondrial metabolism, also for the glycolysis. But when we were looking at the adult age, astrocytes have this decrease in mitochondrial gene expression that go down similar to the second postnatal week, while they stay higher with the glycolytic genes, like it was observed before, that astrocytes are glycolytic cells. Similar when we were looking at the oxygen consumption or ATP production or lactate, they stay higher. One gene that was really high expressed was this PGC1 alpha, that is considered the master regulator for mitochondrial biogenesis and mitochondrial metabolism. So to start, we decided to look at what was happening to the mitochondria in our astrocyte in the developing phase and in the adult phase. And we did this with the in utero electroporation, with a plasmid containing a mitochondrial filler and a cytoplasmic filler, in collaboration also with the group of Frank Polleux and with Tommy Lewis. So as you can see here, when we were looking at the distribution of the mitochondria in our astrocyte, here the mitochondria are in blue, you can see that they cover a different volume of the astrocyte here in gray when we were looking to the development to the adult age. So we had about a 30% reduction, and this was more clear when we were looking more distant from the soma. We developed a 3D color-coded map in order to characterize different morphology of the mitochondria in different categories. And what we were observing is that in the developing astrocyte, we have this huge interconnected mitochondrial network that it was really decreased and significantly decreased in the mature astrocyte, where we had an increase in this small and round mitochondria that we were observing here in red. So in the two developmental stages, we have a different distribution and morphology of the mitochondria. Keeping in mind that we had the PGC1 alpha that was increased in the third postnatal week, we create a mouse model, thanks to the human GFAP promoter. We could decrease the level of PGC1 alpha specifically in astrocyte during the postnatal development. We started with a screening in cell culture in order to understand if we were able to modulate PGC1 alpha level and the mitochondrial metabolism by decreasing the level of PGC1 alpha or overexpressing it with a lentivirus. And indeed, it was the case. So we were able to modulate the gene expression, the mitochondrial metabolism, and also the mitochondrial morphology in cell culture. So at this point, we wanted to know what was happening in vivo, because as I said before, it's really important this period of astrocyte to maturate and go from the proliferation to the morphological maturation and differentiation. So we treated our animal with the tamoxifen during the postnatal period, and we looked what was happening to the proliferation. And as you can see here, we had an increase in the BRDO expression, so the proliferation activity in our astrocyte that we see with the TD tomato expression, in the layer 2-3 and in the layer 5 of the cortex during the development. And this was reflecting in an increase in the number of TD tomato positive cells in the adult brain. Not only, but because we had more proliferation, we found that these astrocytes were smaller. We measured also with the diameter that was smaller in the layer 2-3 and in layer 5. They were overlapping between them, so the cell body was more nearest, and also they were less complex compared to the control. Because we know that there is this bidirectional communication with neurons, we wanted to know if we were able to modulate the level of PGC1 alpha. And thanks to the fact that we know that we have this metabotropic glutamate receptor 5, that it's really high expressed during the second postnatal week in astrocyte, we wanted to know if by modulating mGluR5 and maybe via the mTOR pathway, we were able to modulate the PGC1 alpha level. And indeed, it was the case. By decreasing the level of mGluR5, doing the same mouse model as we were doing before for the PGC1 alpha, so with the Krylov system, deleting the mGluR5 in astrocyte specifically, we found a decrease in the PGC1 alpha level. And this was reflecting in the mitochondrial morphology and distribution. So we had a drastically reduction in the volume covered by mitochondria, and also a reduction in this huge interconnected mitochondria network that we found in the control developing astrocyte, and an increase in the small and round mitochondria. And again, we wanted to know what was happening in vivo. So again, we found an increase in the proliferation activity, an increase in the number of TD tomato positive cells in the adult brain if we were perturbing the mitochondrial, the mGluR5 levels. And also we found that the astrocytes were smaller, less complex, and overlapping between them. To understand if this was impacting on the formation of the synapses, because it's really important, this coincident period, to support also the synapse formation, we looked at the colocalized sphincter in the territory of the astrocyte of a presynaptic and postsynaptic markers in both animal models, so the mGluR5 knockout and the PGC1 alpha knockout, and also in this case, we found in both animal models a decrease in this colocalized sphincter, so in the number of the synapses. Not only, but thanks to the collaboration with Mirko Santello and Jennifer Romanos, we did some electrophysiological analysis, and we found that not only we had less synapses, but these synapses were also immature. To conclude, we wanted to understand if what we were seeing in the mGluR5 knockout animals was really linked to the level of PGC1 alpha. So we re-expressed PGC1 alpha thanks to the lentiviral specific for the astrocyte containing PGC1 alpha. So at this point, we look at the mitochondrial morphology again, and as you can see, we had a partial rescue of this mitochondrial morphology. So we had an increase in the volume covered, and also an increase in the interconnected mitochondria. Not only, but this was also rescuing the level of the proliferation. So we had less proliferation, we had less astrocyte in the layer 3 and the layer 5, similar to the control. The astrocytes were also bigger, more complex, and in this case, they have again the non-overlapping territory features. Again, we wanted to know if this was relevant for the formation of the synapses, and indeed, we could rescue also the level of the synaptic the puncta that we were counting in our mouse model, mGluR5, treated with the lentivirus. So to conclude, mGluR5 deficient astrocytes show a shift in their metabolism toward glycolysis, that a decrease in mitochondrial morphology and distribution, impairment in proliferation and morphological maturation, and also impairment in synaptogenesis and synaptic function. By re-expressing PGC1 alpha in our mGluR5 knockout animals, we were able to restore the phenotype that we were observing, meaning that the mitochondrial metabolism seems really crucial and critical for the postnatal morphogenesis of astrocytes, and also for the formation and function of the synapses. And this can be really important if we were looking after to some pathologies like where we have synaptopathies. And to really conclude, I would like to thank the people involved in the project, my PI Paola Bezzi, Eva, the PhD student that is finishing the project, Francesco Petrelli, that is the co-author with me, and all the collaborators, and you for listening. Thank you.
Thank you, Tamara. Very fascinating work.
Thanks.
Do we have some of the questions? Any questions from the panelists or the audience? Okay, I have one simple question, and it might be very stupid to ask, but it is really very simple. But, since I'm not from the background, after listening to your talk, can I say this thing that basically I would like to know in the layman terms, how is the mitochondria and the synaptic strengths, related as per your work? Can you, explain that in a simpler terms?
I couldn't understand because it was jumping the question. So in the connection, sorry. Can you?
Okay, no, it's okay. It's no problems. Basically, my question is, like in the layman terms, can you just, summarize the role that mitochondria is playing in the synaptic connections?
So it seems that so we know that in neurons, mitochondria are really important where we have spine and the synaptic connection, and this we know. But in astrocyte, we don't know exactly what is the how I can say. We see in this project that by perturbing the mitochondrial metabolism, so probably the fact that we have less ATP production also, and more lactate, that it can influence different mechanisms. We are perturbing this formation of the synapses. So especially in the territory of the astrocyte, so where astrocytes are controlling the neuronal, the formation of the synapses of the neurons. And by perturbing, yes, this activity of the mitochondria, we are perturbing this, and yes. But I don't know if I answer exactly your question.
No, thank you so much. Yes, you did answer my question. Thank you, Tamara. Do we have any other questions from the audience or maybe from the panelists?
Yes, I have maybe a stupid one. Thank
No
you,
question
Tamara.
is stupid.
Yeah.
Thank you, Tamara, for the talk. The work is very, very nice. I was thinking if this metabolic shift you show is only genetically determined, or the environment could influence the strength of this shift or the timing of this shift?
Yeah, this I cannot clearly answer, so I have no clear answer. We see this genetically increase in the gene expression, but what I can say maybe it's that like at the point of view, or really the basic, I don't know, but it was shown that PGC1 alpha can also be regulated in other tissue and other cells, for example, by sport. So we know that we can increase the PGC1 alpha level. So I don't know if in astrocyte it's the same. So if we can change the PGC1 alpha level, and so this mitochondrial metabolism, because it will be super interesting if we can say in the pathologies where we have, for example, low mitochondrial metabolism, or we can say, okay, if you do more sport, you can increase your PGC1 alpha level, and you can find some way to rescue some phenotypes. But for the moment, what I can tell you is the genetically, that this genetic increase. And also the morphology, because you see, we have quite a difference in this morphological distribution of the mitochondria. So it seems that we have more coverage of the mitochondria volume in the volume of the astrocyte, and the morphology is different. And what we know is normally that when you have this long mitochondria that are more interconnected, that they are more oxidative cells. So yes.
Thank you. Sorry, another one. I'm curious. It is not a link between metabolism and morphology in literature, or it's something new?
Which morphology?
The morphology of the astrocyte.
No, this is it's something that we found it, so that by seeing the difference in changing the metabolism, we found this change in the morphology. Yeah.
Okay, thank you.
Thank you, Tamara. Thank you, Sarah. We have a question from Carlos. He's asked, the question is, did you check the calcium activity associated to mitochondria?
No, not for the moment. We didn't check the calcium. I don't know the calcium activity. Yeah, but for sure it's something that we could do, and it could be like linked, because we know that astrocytes have really important in the calcium activity, and mitochondria are linked to this calcium activity. But I cannot tell you in this model how it is the calcium activity. Sorry.
Thank you, Tamara. Thank you, Carlos, for the question.
Thank you.
Do we have any further question? Okay, we move on to our final speaker for this particular session, Elena Jacobs. Elena Jacobs is from University College London, and she will be talking about the effects of the stress hormone cortisol on spontaneous activity in the larval zebrafish tectum. To you, Elena.
Hi. Thanks for the intro. Hi, my name is Elena. I am a postdoc at the Sainsbury Wellcome Centre at UCL. But the particular work that I'm going to be presenting to you today was in collaboration with Martin Meyer's group at King's College London. Hang on. And yeah, here's just a picture of my puppy to remind me to tell you that, yeah, I have a puppy. So he's usually a pretty good boy, but if you hear any funny noises in the background, my apologies in advance. Okay, so the really big picture question that I'm interested in is, how does stress affect the maturation of neural circuits? So first of all, what do I mean with stress? It's kind of a ubiquitous term. So the definition that I like to use is the one proposed by McEwen, which is that it's a challenging event, whether it's emotional or physical, that triggers the stress response, which in turn triggers this reorganization of physiology and behavior. Now, why is this important? So we know that stress is ubiquitous, and it has these, we know that excessive stress can have negative consequences on health. But it looks like particularly during early life when we have development going on, that this might be a critical period, because if you experience a lot of stress and traumatic experience during that time, we know that this can have impacts on the health of an individual for the rest of their life. Now, these impacts can be fascinatingly varied. So you can have effects on the immune system and the cardiovascular health. People are more prone to developing chronic pain and a host of mental health issues, from depression and PTSD, for example. Now, this has been well reported in epidemiological studies in humans, but this has also been replicated in animal models. So from these studies, we know that this early life stress has lasting impacts on the brain. And even though there's been a lot of work going into this topic, we still kind of lack an understanding of the fundamental mechanisms that kind of cause these consequences. So I'd like to draw your attention again to the definition that I gave you, and particularly emphasize that stress also has this effect on behavior. Now, what I'd like to mention here is that some part of circuits that are really important for modulating behavior are the sensory motor circuits. All the information of the world comes to us through our senses, and it's these sensory motor circuits that are really important in modulating how we respond to the sensory information, including stressful information, and how we respond behaviorally. Now, in terms of the sensory motor circuits, we also know there's an increasing amount of evidence that they are affected by internal states, including stress, which is an example. And here is a cartoon from a recent clinical paper that kind of looked at these effects, and it suggested that in health, you have these brain stress systems that are kind of in balance with these sensory processing regions. But when you have a lot of early life stress, these brain stress systems kind of like take over, and they also influence the sensory processing. So there is clinical evidence that stress affects the sensory processing. So if we want to study the effects of the stress on the developing brain, including the sensory systems, what do we need? So first of all, we want to have a model where we can obtain these robust behavioral measurements. Secondly, ideally, what we really want to do is be able to access the whole circuitry. So we can look at all the elements of the brain at the same time, including the sensory systems that I mentioned before. And ideally, we want a model organism where we can have easy monitoring of development so that we can look at these effects over time. And what I'm proposing is that the zebrafish is an ideal model organism for this because it ticks all of these boxes. First of all, it's a vertebrate model, and a lot of the brain structures are evolutionarily conserved, including the stress axis. Here I'm showing a cartoon comparing the HBA axis in mammals to the one in fish. And there are a lot of similarities, including the brain structures and also the hormonal effectors. And secondly, zebrafish develop really quickly, and in one pairing we get a lot of offspring, so we can do a lot of high throughput screening, which is another advantage of this model. And most critically, potentially for this aspect, is that they're optically transparent during development. So here's an image of a zebrafish brain, and this is pretty much the entire brain that you can look at the same time. And the really amazing thing is that we can do this at cellular resolution. So we can obtain data from every cell in the brain simultaneously. And because of this feature, it's becoming a really established model in systems neuroscience. Now, so for today's project, the hypotheses that I'll be addressing is that stress alters the internal brain state. Now, what do I mean with that? Particularly, I mean with this is that stress will result in a predictable change in spontaneous activity dynamics. So what I mean with spontaneous activity is kind of this resting state, so when there's no other stimulation going on. And particularly that this will also have an effect on the sensory motor circuits that I mentioned. And this hypothesis leads to a second one, which is that this physiological change will in turn have a functional effect, and this will have an effect on behaviors, including visually guided behaviors, for example. Now, I should say here that this is very much a work in progress. A lot of the data that I'll be presenting, so all of the data that I'll be presenting today is still very preliminary. And yeah, for that reason, I really love some feedback and your ideas and suggestions. And because it's a work in progress, so what I'll be talking to you about today is this first step. So how did I address this hypothesis? First of all, one of the main effectors of the stress response is this hormone cortisol. So I isolated the stress response kind of to look at the physiological effect of cortisol, and also hypothesized that cortisol alone will also have an effect on this internal brain state. And then I performed 2-photon imaging of the optic tectum. So in my experimental setup, my zebrafish were fixed in a gel and in water, and under the microscope, they were in the darkness, no other sensory stimuli going on, and I was imaging the spontaneous activity for an hour for a baseline, and then I added the solution with the cortisol. And here's an example of what the 2-photon imaging looks like in zebrafish. And here in this bluish gray color is highlighted the zebrafish tectum, which is the region of interest that I was focusing on, as this is an element of the sensory-motor circuit. And the zebrafish tectum, the tectum is the evolutionary conserved part that we in mammals know as the superior colliculus. So one of the first things I asked is to look at this, whether there's a predictable effect, is there an increase or a decrease in activity in the tectum? And the answer is, it's not quite that simple. We get this kind of like salt and pepper effect, where some cells increase or decrease their activity. So here what I've done is, here's all the cells from this zebrafish tectum, and I've colored them according to whether they increase their activity, which is given a red color, or they decrease their activity, which is a blue color. And here are 3 different example fish, and in each fish you can see that you've kind of have this scatter pattern of cells that increase and decrease. And here's a quick summary graph from all the experiments that kind of quantify like how many of the cells increase or decrease their activity or remain equal. And you can kind of see that there's a small percentage that didn't change their activity, and roughly equal measures like increase or decrease their activity after adding the cortisol solution. So this kind of suggests that cortisol doesn't uniformly affect activity in the tectum. But with this result alone, I couldn't really understand what is the effect of cortisol. For example, this is spontaneous activity. If I were to look at just beginning of the recording of spontaneous activity versus the end of it, I could potentially just by chance see these increases and decreases in activity. So what I wanted to know is this difference in population activity actually meaningful and predictable? So in order to look at this, I trained a classifier to predict whether the activity pattern at any given time point came from the baseline or from the cortisol treated condition. So in order to reduce the dimensionality of my matrix, which is a highly dimensional matrix of the neuronal activity over time, I first performed PCA on the activity matrix, and I kept the first 10 principal components. Then I trained a logistic regression model to predict whether at any given time the neural activity pattern came from the baseline or the treated condition. And I got an accuracy between 93 to 100% using these top principal components. So this is quantified here on the right in this graph, where on the top hand side in the dark blue dots show the decoder accuracy for the example fish that I showed you earlier. And I also did a shuffle control where I mixed up the label, whether it came from a baseline or the cortisol condition. And in that case, you get this around 50%, so chance level prediction, which kind of confirmed that, yes, the cortisol is doing something predictable to my population dynamics. Now, if we visualize what is happening, visualize the PC space. Here are again the same 3 example fish that I showed you before, and each dot is about a time point, and I'm representing them in the top 3 kind of neural state space in terms of the principal components. And each dot I've colored according to whether it came to baseline or from the cortisol condition. And you can see that you get actually already with only 3 principal components, you get a pretty clean separation in terms of the neural population dynamics, which kind of tells us why it was possible to get this really high decoder performance, because you get this very clear clustering in this highly dimensional activity space, and whether the activity came from the baseline or the cortisol treated condition. So what I'd like to do in the future in this project is, first of all, I'd like to try a canonical correlation analysis. So the idea being is, so in this case, the results that I showed you, I looked at every fish, I looked at whether in each fish I could see a difference between the baseline and the cortisol. Now, what would be interesting to see is whether this kind of cortisol leads to a kind of stereotype response in the population dynamics. And for that, I need to try and correlate this kind of like the latent dynamics in this highly dimensional space between the different fish. And canonical correlation analysis is a technique that might be able to allow me to do that. And then I could find out whether there's a stereotyped response to cortisol in the population dynamics. And also, I really want to understand the effects of cortisol more precisely. I've done some control experiments, and from that, it's still, I want to do more work on really understanding like what exactly the effect of cortisol is. And particularly importantly, coming to the second hypothesis that I proposed is, I want to know what is the functional relevance of this? So what is happening to behavior, for example? And lastly, I told you what I'm interested in is kind of these long-term consequences of a developmental perturbation. So the data that I showed you today are all from an acute experiment. And what I'd like to do in the future is investigate whether there are long-term consequences. From rodent models, for example, we know that a single stressful event can have lasting effects on the neurons. And it will be interesting to see what the effects are in terms of population dynamics. So with that, I would like to thank you very much for your attention. It's a great pleasure to be able to present my work to you. I'd like to thank Yo Izakai and Tom Otis from the Sainsbury Wellcome Center, who very kindly let me work on this collaboration with Martin Meyer's lab. And I'd also like to thank Matt Harvey, who's a PhD student in Martin's lab, who's really great and helped me with the experiments, and everyone else in the Meyer lab who provided help and feedback. And I'll be happy to take any questions.
Thank you so much, Elena. Really a very wonderful work. Do we have any questions from the audience or from the panelists? Any questions? Well, I would be definitely looking forward to the long-term consequences that you will find in your work. It's really very interesting work, to be honest. And I guess we can have its use, is we can have a better understanding of how the stress plays the role with this particular work. Thank you so much, Elena. So I guess that is it. If we have any questions. All right. So thank you, everyone. Thanks the attendees and all the three panelists and Scene for the backend support. Thank you so much. It was nice having you all. Have fun and continue with the NeuroMatch session. Thank you so much. Bye.
ഞാൻ കൊണ്ടുവരാൻ കുറച്ച് എനിക്ക് കൊണ്ടുവരാം അതെ ഇല്ല ഇന്ന് മുതൽ പാലുണ്ടാവില്ല.
All right, I think we're good to go. Welcome everyone to this session. Good morning, midday, afternoon, night, wherever you are right now. This is a session in the theme D, which is cognition, motivation, and emotion. And we're having a series of traditional talks. That means speakers will have 12 minutes, and then we have 3 minutes to ask questions and discuss what kind of they're doing next and where they're going with their findings. Please do ask questions. There are no silly questions at all. Anything you want to know about kind of the methods and the work is allowed, and there's definitely no problem if you know some things are new to you to ask about them. You can use the Q&A panel in the Zoom function to ask your questions. You can also use the webinar chat, or as an attendee, you can just kind of lift a virtual hand, and then I can unmute you and you can ask your question yourself. Our first speaker is Alexandra Sobczak from the University of Lübeck, and she's going to be talking about alpha-beta oscillations in the human substantia nigra, signal the anticipation of appetitive and aversive social feedback, evidence from intracranial recordings. Alexandra, you should be able to share your screen now and start with your talk. Thank you very much.
I will do so. Thank you very much, Ellie, for the nice introduction. And welcome everyone to my talk today. You have just heard the title, so we just dive right in. First of all, I would like to give you some background information on the topic. So on the behavioral level, when we expect to get a reward, this definitely modulates our motivation and our behavior. And it leads to increased speed or vigor of movements, for example, motor movements, which is a kind of approach behavior. And on the neural level, the key region to do that, which mediates these effects, are the dopamine neurons in the substantia nigra and the ventral tegmental area. You can see on the right-hand side in the figure, the substantia nigra. I have marked it in red for you, so you can easily spot it. These dopamine neurons, they signal appetitive and aversive stimuli. These can be, for example, food stimuli when you're handling mice or other animals. For humans, it would be monetary gains or losses, or social approval like smiling faces, and social disapproval, frowning faces, for example, or electrical shocks. And the dopamine neurons are excited by the reward cues themselves, so not only the rewards, and they're excited when you receive unexpected rewards. On the other hand, they are also inhibited by the omission of an expected reward. And this is what happens in the single dopamine neuron, but on a broader level, we can also have a look at group neural mechanisms, which is, for example, possible by looking at neural oscillations. And there we have observed, or other people's work shows that reward anticipation and reward feedback, so the receiving part, has been associated with oscillatory power increase in the beta and theta range. But the dopaminergic system declines with age. This is a non-pathological normal aging process, but we experience a gray matter loss, a myelin loss, a loss of dopaminergic neurons, and iron accumulation in the basal ganglia, which are part of the reward circuit. And this impacts the processing of reward-related information in the kind that, for example, elderly show no prediction signal in the nucleus accumbens, which is part of the basal ganglia and receives input from the SNPTA. But the response to reward received is still there. The study has also found that the administration of L-DOPA, which is the precursor of dopamine, restores these prediction signals. And another study has shown that reward-related invigoration of response times is linked with the structural integrity of the SNPTA. Another aspect of neuronal degeneration is, or a good example is Parkinson's disease, where we have a pathological degeneration of dopamine neurons, and the patients accordingly demonstrate issues with learning from feedback. So far, so good. So what we wanted to investigate in this study is how the human substantia nigra signals the anticipation of positive and negative events. And we use social approval and disapproval as a model for these positive and negative events. And we use indirect recordings from a human SN in PD patients. Otherwise, these direct recordings are not possible. And these patients were undergoing surgery for deep brain stimulator implantation. And what we expected is that the substantia nigra responds to positive and negative predictors, and that there might be a difference between negative and positive predictor processing as well. On the behavioral level, we were interested in age differences, so younger healthy participants versus older healthy participants. And furthermore, we were interested in whether dopamine has an effect in this paradigm that I'm going to show you in a minute or in a second. Yeah, so our hypothesis there for the behavioral response is that younger participants increase their response speed when anticipating to gain positive and avoid negative feedback. This is what I will refer to as the RT effect, response time effect. And we further hypothesized that the older adults show an attenuated RT effect due to the age-related decline of the dopaminergic system. And furthermore, we hypothesized that the PD patients show no RT effect of medication. So when they withdrew from the dopaminergic medication due to dopamine dysfunction. And on the other side, of course, we predicted that the PD patients would show the RT effect when receiving medication as planned by their doctors. So this is the paradigm that we use to investigate our research questions. It's the social incentive delay task, which is a modification of the well-known paradigm, the monetary incentive delay task. And it's specifically designed to investigate the neural mechanisms underlying the processing of reward and punishment. And another important aspect of this paradigm is that it allows for the examination of the different stages of reward processing. That means the anticipation phase and the consumption phase. On the right-hand side, you can see our trial design. We have three conditions. One is the neutral condition, then we have the social reward condition, and the social disapproval condition. And in every trial, we would first present a cue signaling the respective condition. Then a target appears, and the task of the participants is to press a button as fast as possible when the target appears. Depending on the condition and the participant's performance, the participants receive feedback in form of a picture or photograph. And in the social reward condition, they would receive positive feedback, so smiley face and thumbs up for fast responses, and neutral feedback for slow responses. In the social disapproval condition, they would get neutral feedback for fast responses and disapproving feedback, so frowning faces and thumbs down, for slow responses. And in the neutral condition, the feedback was always neutral. So we tested 4 groups of participants. The first group is healthy young adults, 51 in total with a mean age of 23. And the second group was our healthy older adults, in sum 52, with a mean age of 64. Those both groups were tested purely behaviorally. And the third group is our first PD patient group with 12 participants in total. And those were measured intraoperatively off medication with intracranial EEG. And the last group is a group of 21 PD patients. We measured them on and off medication to compare those two conditions. And this was also purely behavioral. So we do not have any neural data from them. So these are the response times from all groups. You can see that the healthy young adults are the fastest, and then followed by the healthy older adults. And the PD patients, either group of them showed the longest response times, which makes sense since bradykinesia is one of the core symptoms of Parkinson's disease. Let's have a look on the individual group comparisons. So since response times tend to be not normally distributed, we calculated permutation tests for each of the conditions or for each of the comparisons, and we obtained for this mixed design, a main effect of group and a main effect of condition, which means that we have age-related slowing in the older group. They tend to be slower in total than the younger group. And the pairwise comparisons showed that both groups, the healthy older and the healthy younger adults, speed up their response times when anticipating positive and negative feedback as compared to neutral. This is against our hypotheses. We would have anticipated an interaction here, but I will talk about the interpretation later. For the first PD group, where we could record intracranial EEG from the human SN, we conducted a time frequency analysis on the data, and you can see the results on the left-hand side. So these are the power spectra from the Q phase, and we performed a cluster-based permutation test in field trip, which yielded a significant cluster ranging from alpha to beta frequency range and extended from roughly 210 milliseconds to 600 milliseconds. In a second step, to assess the differences between the conditions, I averaged the cluster power. You can see the cluster outlined in black in the plots, and compared them pairwise. And on the right-hand side, you can see the cluster averages or the cluster power averages. And we again had significant differences between the positive versus neutral and the negative versus neutral condition, but not between the two incentive conditions, so positive and negative. We also have the reaction times or response times from these Parkinson patients. There were no significant effects in this sample, which is what we hypothesized, but on the other hand, it's rather unstable or inconclusive since we have a very low sample size. And we were wondering whether this is the absence of the effect is grounded in medication withdrawal or due to small sample size and low power. So the last study, and I'm almost through, is the on and off comparison of the Parkinson patient sample, the second one. And here we can see that we have a group main effect and a condition main effect, which is significant, meaning that the Parkinson patients are slower off medication, which makes sense. And both of the groups, again, no interaction, show faster response times when anticipating positive and negative feedback. To sum everything up, all our participants showed the RT effect. So they increased efforts to obtain social approval and avoid social disapproval. The performance benefit was not attenuated due to neuronal degeneration. This is likely to interpret it as, they did not have to learn the stimulus outcome association in our task. It was presented and explicitly stated beforehand. They might experience problems when having to learn this association. The same applies to the PD patients. And the most important finding here is that we have activity in the substantia nigra, differentiating between neutral and positive cues, and also negative and neutral cues. Thank you very much.
Thank you. You were perfectly on time, by the way. That was some good timekeeping there. Thank you so much, Alexandra, for your talk. That was very interesting. I do invite any attendees, but also the other panelists, if you have questions or you want to follow up on anything, this is like your moment. Oh yeah, please, Yelena.
Yeah, I'm interested. Can you extract dopamine signal from non-dopamine signal in substantia nigra? So I think that there is like a populational neuronal activity.
So you mean, can I differentiate between non-dopamine and dopaminergic activation?
Pars reticulata, pars compacta, something like that, and there is different types of neurons there.
That's true. For that, I would have to look at the firing pattern, which I did not do. I only looked at the summation of the potentials. But I think it's possible.
Yes, you haven't control for some movement, maybe represented. So there is like substantia nigra, pars reticulata. It's for movement planning and outputting movement programs, something like that.
And they should not have moved during the Q phase. it should be enough time free of movement. But of course, there might be some movement planning since they had to respond right afterwards.
Okay.
I haven't controlled for that.
Yeah, thank you.
Thank you very much. If we don't have any other questions coming in, no, it doesn't seem like it. I'll be happy to introduce our second speaker for this session, which is Jelena Belova from the Semenov Institute of Chemical Physics. And she will be talking about oscillatory activity in the SDN under PD. Do we need to account for a periodic broadband signal / activity and how it affects our data? Jelena, are you okay to share your screen? Does it work? Awesome. Off you go.
Yeah, can you hear me? Yeah, okay. So I'm glad to present here my study data, and I would talk about local field potentials and a periodic signal and how it affects our data. So one moment, I just check how can I, okay, so yeah. I study Parkinson's disease and basal ganglia activity, and Parkinson's disease is a hyperkinetic movement disorder that is caused by dopamine deficiency and subsequent basal ganglia dysfunction. And deep brain stimulation is a procedure that is proved to be effective to treat motor signs of PD. And the target for DBS is not always, but often the subthalamic nucleus. Neurosurgeons commonly use micro and macroelectrode recordings to define the borders of the subthalamic nucleus during the surgery. And these recordings provide very important data about neuronal activity in the affected structures in Parkinsonian patients. The first model of basal ganglia network was proposed about 30 years ago by Albin and DeLong, and it states that imbalance between a direct and direct pathway may prevent normal movement. But current model of Parkinsonism pays specific attention to the patterns of neuronal activity. And they say that extensive synchronization in several frequency bands lead to the emergence of pathological oscillation in the basal ganglia structures. And here you can see the picture from the review of Peter Brown about oscillations in basal ganglia. Peter Brown states that lower frequency oscillations in theta alpha and beta range are antikinetic, while higher frequency gamma oscillations are prokinetic and facilitate the movement. So how can we study these oscillations? Local field potentials are commonly used as it represent populational neuronal activity, and we can apply fast Fourier transform to plot the power spectral density, and the oscillations are shown as peaks on this spectra, and the amplitude of these peaks correspond to the power of these oscillations. However, aside from periodic oscillatory components, there is also aperiodic activity that is often referred to as noise. It is easily seen when we plot the power spectral density in a log-log scale. But these aperiodic features of LFP signals are generally ignored because it's difficult to interpret them. But recently, Richard Gao and colleagues proposed that this aperiodic component may provide some valuable information about the state of populational neuronal activity. They suggested that this slope in the spectra may reflect the ratio between excitatory and inhibitory currents in the site of recordings. They modeled LFP signal as a linear sum of inputs from excitatory and inhibitory neuronal populations. They derive inhibitory input from the current from GABA A receptor that is much slower than AMPA receptor that was used to model excitatory inputs. So they state that a periodic slope of LFP in their model may reflect the balance between these inputs and change with excitation and inhibition currents. So we decided to test this hypothesis using LFP recordings performed during DBS surgeries. It is challenging to compare the LFP spectra recorded in different patients and even in various recording sites. So we search for a task that allows us to compare LFP recorded in short period of time in the same site. We thought that if the slope indicates a shift from excitation to inhibition, we would see the changes in the slope when switching from rest to moving condition. So we collected the data in 2017-2018 in Burdenko Center for Neurosurgery during 22 DBS surgeries in Parkinsonian patients in the subthalamic nucleus. And during recording, patients were asked to contract the fist and move foot on the contralateral side of the body. There were in total 116 LFP recordings that contain rest condition and fist contraction, and 48 recordings from this set was also containing leg task condition. Here you can see the steps of analysis, and we segmented our LFP recordings at rest and during hand and leg movement and analyzed them separately. We plot power spectral density in alpha, low beta, high beta, and gamma bands, and use two different approaches to estimate power oscillation in given bands. First, using conventional approach, we estimated power of oscillations using mean value of power spectral density in the given frequencies. And then we used the novel approach when we extract this aperiodic signal from the spectra in log-log scale, and then account for oscillation based on the amplitude of peaks after the extraction of this slope. And in some cases, there were no oscillatory peaks in a band after this procedure. So here are the data that was obtained using two methods. We compared power, and you see on the top the data using a conventional approach, and on the bottom there is the data using novel approach accounting for aperiodic component. And we see that conventional approach gives us a very significant increase in gamma oscillations, while accounting for the slope, we see a very significant decrease in the slope component, but no change in gamma oscillation score. Also, when analyzing leg movement task, we see also increase in low beta range for both methods, and p-values are not similar but comparable for these methods. So here are group spectra for conventional approach, and you see a clear increase in low beta and gamma range. And on the right side, there is individual data. Each point is individual site of recordings. X-axis show value of power at rest, and Y-axis show power at movement condition. And we see all the dots above the diagonal. They show increase in the power, and below the diagonal there is decreased values. So we see significant gamma and low beta increase for these conditions. And here are the spectra that were obtained using a novel approach, and we see clear decrease in the slope during movement, and also we see not very clear, but still increase in low beta band during movement. And here also to the right are individual data for all our data set. So if we consider classical boxes and arrow model here, we may state that changes in the pathways may be reflected in the movement state, and we expect increased inhibition toward the ST end and corresponding decreased excitation from the ST end to internal segment of globus pallidus. And these change when switching to the movement condition would lead to decrease in the balance between excitation and inhibition, and decrease the slope in the slope that we observed in our study. So in the right, you see the part of the picture that I have shown you earlier. And here we see prokinetic gamma oscillations that probably is not oscillatory activity, but maybe reflect the changes in the slope in a periodic movement, in a periodic broadband signal. So let me read the conclusion of our work. The slope of a periodic noise in LFP spectra probably reflect the excitation inhibition balance of synaptic projections to the site of LFP recordings. At least our data confirm this hypothesis. The slope decrease in STN during movement may reflect dynamic changes in motor processing through basal ganglia structures and probably display the shift in activity from an indirect pathway toward a direct pathway. The visible increase in LFP gamma power during movements may arise from a periodic source rather than oscillatory one. And in general, oscillations in lower frequency, like low beta band, were not affected after extraction of a periodic slope. I would like to thank our team and say special thanks to my friend and colleague Yulia Semyonova. She contributed extensively to this study and performed a lot of computations on spectral features. And thank you for your attention.
Thank you very much. Thank you very much, Elena. We do have some amazing timekeeping in this session. So time for questions. Again, I invite any of our participants. You can raise your virtual hand or use the Q&A or also use the chat to pose your questions. And I'll be able to unmute you so you have the option to ask the question in person. Also, yes, panelists, Sandeep, you have a question.
Okay, I have a very small basic doubt. That is, Elena, it was a nice presentation. And in one of your slides, you mentioned there is a shift in STN, that power spectral density slope, STN slope, when we are moving from the shift from the direct to the indirect pathway. So can we also associate that particular shift in the frequency ranges, that is from the beta ranges to the gamma ranges, to the pathological outcome? That maybe in case of Parkinson's disease, we do we see in one of the study which I was referring, there is an increased frequency, the activity in the higher frequency ranges. Can we relate that to this experimental outcome that you were seeing, that is shift from the direct to the indirect pathway?
I think that we need to revise this data, and maybe we will see this change in the slope. And according to the proposals of the Richard Gao and team, which I'm referring to, they think that yes, of course, this shift reflect these changes, and maybe we can estimate the pathological imbalance in the circuits using this metric. But I think that it's not very easy, as we need to estimate the slope, we need the recording of some duration. So we cannot estimate a very tightly, very short shifts in the slope now. So maybe when accounting for long periods, we don't have very short data, but we can measure some state. Yes.
Okay. Yeah, thank you, Elena.
Thank you. We do have a question in the Q&A, which is from Sanka, and they ask, gamma still seem to have a change, although not statistically significant, even when accounting for slope. Also, there has been evidence for gamma in dyskinesia, right? So how much can we attribute to the slope and how much can we attribute to gamma?
So I have shown here on the slide, it was not very, I haven't mentioned it, but you see n number for the aperiodic component method. So there is only 14 cases from 48 that were shown any gamma oscillation after extracting of this slope. So there was very few recordings that still have some gamma oscillations after extraction of the slope. Maybe in some other conditions, or maybe for the longer recordings, we will estimate these gamma oscillations and can distinguish this aperiodic slope from the gamma oscillations. In our study, there was a very few peaks in gamma range after extraction.
Thank you very much. If there's no further questions from panelists or attendees, I'm just waiting a moment. All right, looking good. I would hand over to our third and final speaker, who is Sandeep Nair from the Indian Institute of Technology, Madras, and he will speak on a multi-scale cortico-basal ganglia model for arm-reaching task in normal and Parkinsonian conditions. Sandeep, you're up.
Yeah, thank you, Ellie, for the introduction. So let me start sharing my screen. So you all can see my screen. Okay, thank you. So good day, everyone. Today I'll be talking about our work, the systems neuropharmacological model of corticobasal ganglia circuitry for arm reaching. So let me start with a brief introduction. Parkinson's disease, the second most neurodegenerative disease after Alzheimer's, is caused to the death of dopaminergic neurons in substantia nigra pars compacta subsystem. And over the last couple of decades, the number of people that are severely impacted by this disease is on an incline. The exact cause of the cell death is not yet known. However, various hypotheses link the cell death to metabolic deficiencies, metabolic vulnerabilities, energy deficiency, oxidative stress, Lewy body formation, and alpha-syn aggregation. So in order to comprehensively understand the mechanisms leading to the cell death and associated pathological symptoms, it is important to have an integrated model of various subsystems at multi-scale level. So this is the outline of our model. So the essence of our model is to explain how cell death leads to the behavioral abnormalities and explore various therapeutical outcomes. So let me use the pointer. So this is the model consists of an arm model. So interface with the motor neurons. There is a corticobasal ganglia circuit and the basal ganglia and the substantia nigra subsystems. So let me zoom into the corticobasal ganglia circuitry. It is the arm is trying to reach the target, and the end effector position of the arm is detected by the proprioceptive cortex and passed on as a signal to the motor cortex. The motor cortex also receives a signal from regarding the goal information from the prefrontal cortex. It combines the signal and it will send a signal towards the basal ganglia, and basal ganglia with after its internal mechanisms will send after the internal dynamics, it will send a corrective signal back to the motor cortex, which will further issue motor commands and then drive the arm further. So inside the BG, the striatum, based on the sensory feedback error, it does a value computation, and corresponding to the value difference signal, it will send a GABAergic current towards the substantia nigra pars compacta, and together with the GABAergic NMDA and AMPA signals that is receiving, it will send a dopamine. It will release dopamine into the extracellular space. So the BG subsystem, it consists of an input nuclei. So one second, let me remove the laser pointer. Okay. So the BG subsystem, it consists of the input nucleus striatum, which consists of the D1 and D2 MSNs, which are differentially activated in the presence of dopamine. Initially, the GPI will be inhibiting the thalamus, and under normal condition, the dopamine level is high, and on reception of the motor command and with high dopamine availability, the direct pathway is activated. The D1 MSNs will be projecting to the GPI. It will inhibit the activity of the GPI, which will disinhibit the thalamus, and thereby facilitating the movement. On the other hand, under Parkinsonian conditions, the dopamine signal is due to the death of substantia nigra cells, the dopamine signals are less in availability. So the D2 pathway will be activated. The D2 projections to the GPE will inhibit the GPE, which will reduce the inhibition towards the subthalamic nucleus, which will overexcite the GPI, and which in turn increase the inhibition towards the thalamus, thereby inhibiting the movement. So the dopamine plays a crucial role, and it is modulated by the molecular mechanisms of substantia nigra pars compacta. So we have also, therefore, we have also tried to integrate a detailed biophysical model of substantia nigra pars compacta, which explains all the molecular level mechanisms like glycolysis, mitochondria, the calcium buffering mechanism, the levodopa uptake mechanism, etc. And the dopamine released from a network of such substantia nigra dopaminergic neurons will be released into the extracellular space. So this dopamine is released based on the GABAergic input. The amount of dopamine is controlled by the GABAergic signal that is received from the striatum based on the sensory feedback signal that it is received from the cortex. So the EDA that is released, now the Parkinsonian condition happens due to the deficiency of the dopamine that is released. Now levodopa therapy is one of the first line mechanisms, medication methods for the Parkinsonian patients. Levodopa being the precursor to the dopamine. So we have tried to integrate a two-compartmental model of levodopa administration. So the levodopa is orally consumed. It reaches the gut, and it is absorbed into the bloodstream at a rate I. The C1 and C2 compartments represents the central and the peripheral compartments. The central compartment is where the levodopa is administered and the plasma concentration measured. And the peripheral compartment represents the interaction between the plasma and other bodily fluids. So a portion of the levodopa crosses the blood-brain barrier, and it reaches the blood-brain barrier, and it gets combined with the L-dopa that is synthesized from blood tyrosine. So, and this L-dopa is packed into the vesicles. It is converted to cytosolic DA using the enzyme aromatic amino acid decarboxylase, which is then packed into the vesicles, transported towards the synaptic cleft, docked to the proteins, and released into the extracellular space. So let me show some of the initial results from our model. So we for the testing strategy, we follow two types of testing. One is the impact of cell death, the percentage of cell loss impacting only the striatum, and in the second case, the cell loss is impacting both the striatum as well as the subthalamic nucleus. So for the first case, under the normal condition, we can see that the arm is reaching the target. It reaches pretty quickly, around 0.6 seconds. The movement speed, it follows a perfect bell curve where the velocity increases in the start up to a certain point, and then as it approaches the target, it slowly comes down. And the STN population activity, which is the time frequency analysis, shows higher powered activity in the 0 to 10 hertz spectrum. So as we keep on increasing the cell loss, for the 25% cell loss, we can see that the trajectory is smooth, the reaching is perfect, and but it takes a slightly higher time. And with even higher cell loss, we can see that the trajectory is wavered. It doesn't, it is, and the velocity curve is not smooth. It increases a bit, then it comes down, and then it further increases. And the STN population activity shows higher powers in the mid-beta regions. With a further increase in the cell loss, we can see that the arm fails to reach the target even after the maximum allowed time of 5 seconds. That is what we configured in our model. And for the second case, where the dopaminergic loss is impacting both the striatum as well as the subthalamic nucleus, in the normal condition, the arm reaches the target pretty quickly. With increasing cell loss, the time taken is more. And with the 50% cell loss, what we observe is kind of a akinetic rigid. It is a kind of a cogwheel rigidity, where the arm slightly starts moving, and then it stops for a certain amount of time, and then we can just see the velocity curve, a movement speed, and then in middle it picks up the speed, it moves towards the target, and then there it gets stuck again, and then it hovers around and reaches the target, taking considerable amount of time. With even higher increase in the cell loss, what we observed is that arm fails to start from the starting position, and it is some kind of akinetic rigidity. With levodopa medication, the performance is considerably improved, and it is seen during the performance metrics that we recorded in terms of average time to reach and the percentage of successful reaches. And here are some of the cardinal symptoms of PD that we simulated using our model. So in the first case, under normal condition, the arm reaches the target pretty quickly, and a point, I'm not sure whether it is visible, it reaches the target around 0.6 seconds. And then second case is the bradykinesia. It takes considerable amount of time, however it reaches the target and around double the time. And the third one is the reaching tremor. It moves towards the target, and as it almost approaches the target, it hovers around, and it takes considerable amount of time reaching the target. And the third one, which we saw in the earlier plot, the cogwheel rigidity, where the arm takes some time, initiates movement slightly, then it gets stuck, and it moves towards the target, and then from there it gets stuck again, and then finally reaches the target after considerable amount of time. And the final case, that is the akinetic rigidity, with increasing cell loss impacting both STN and the striatum. Here the arm hardly moves from the initial position, and it never reaches the target. So let me fast forward this one. Okay, yeah, I'm almost winding up. So I'm not sure why this slide is not moving. Okay, so all these are a small part of a bigger picture, that is to build a patient-specific test bench. So here, using various activities performed and the interaction with the cortico-basal ganglia circuit, the meta parameters are derived using targeted outputs and the therapeutic interventions. And then fine-tuning the same, the optimization of the medication can be done in terms of dosage and the frequency of dosage. So here are a few of the selected references that we use for our work. And, that's all from my side. Thank you for your participation and patience, and looking forward for your questions and valuable suggestions. Thank you.
Thank you very much, Sandeep. We do have a little bit of time left before we have to vacate the room. Oh, I see. Yelena, is that your hand?
Ah, yes.
Yeah, can you explain what is the difference between single units and populational activity on your slides? So what assumptions do you have about single activity and populational activity? They are not the same.
Okay, so the single activity is that in our model we used the subthalamic nucleus is model using a 15 by 15, 15 cross 15 neuron element. That is a total of 225 neurons are in the cluster. So what in the single activity, what we did is that the frequency response we analyzed for a single neuron, and the STN population activity is the one that we did for taking the local field potential. That is the average of all the neurons, all the 225 neurons. So that is how it is represented. The single activity I just showed for reference. That is what is the activity of a single neuron, and what is the.
So there is a single activity from Parkinsonian patients or animals,
No.
or there is model?
No, this is just a purely model. So this is just showing what is the power recorded in a single, that power that is observed in a single neuron during that, because a single neuron, it will be anyway showing higher power, and the population activity, because during the averaging out, when it is a very asynchronous, when the asynchronous oscillation occurs among the neurons, the peaks will cancel each other, and then the power will reduce. So that is why we see a difference in power with respect to single neuron and the population activity. So ideally, we have to take the population activity only, and the single neuron activity, I just showed it for a comparison. Because in reality, we will be recording the population activity only.
Thank you, Sandeep. If there are no further questions, that is the end of this session. Thank you again to all the speakers. Well done, and that's some really impressive research that you presented, all of you. And hopefully see you around in other talks and future online, or hopefully one day real life events.
Thank you very much for the session.
Thank you.
Bye, everyone.
Bye, everyone.
Bye.
Bye.
Processes and the role of AI in teamwork. So welcome, Kristen. The stage is yours.
Thank you. Hi, everyone. Let me share my screen. Okay, so hi, I'm Kristen. In this presentation, I will discuss the study on neural flexibility and the propensity to change opinions following online media consumption. Our team's main research aim was to investigate the dynamic coactivity of neural correlates of opinion change as prompted by online persuasive news articles or social media posts. Our research team became interested in this topic because, as we witnessed today, persuasive media on Facebook, other social media platforms, or news sites has a profound effect on individual opinions and important decision-making. Information that you see and consume online has neurological and social implications, not only on the individual level, but also on the societal level. Digital media consumption may impact voting behavior, political partisanship, and even policymaking. Previous communication neuroscience research shows a significant relationship between frontal brain regional activity and opinion change. We further investigate this finding to explore the nuance in this dynamic neural activity within frontal brain regions using data that closely resembles a real-world scenario. Our main method, dynamic community detection, serves as our novel contribution to this literature. I will provide an overview of this method in a few minutes. We conducted an experiment in which EEG activity was collected from 240 healthy and diverse participants while interacting with biased content, such as inflated news articles or social media posts within a social media environment, which was actually created and programmed by our research team. Before and after interacting with the platform, we collected participant opinions via surveys on various sensitive issues such as vaccinations, capital punishment or the death penalty, corporate investment practices, and politics. To determine behavioral change, we calculated individual opinion change scores before and after using the SMART platform based on their survey responses. Individuals who changed their opinion had a larger difference in their pre and post platform responses. To determine neurological changes, we estimated the statistical relationship of the EEG data between electrodes using WPLI, and we then input these matrices into the dynamic community detection algorithm to extract network metrics that quantify the probability of nodes, or electrodes in this case, to change their network affiliation across time. So the novel approach that we contribute to this communication neuroscience literature is a network science approach that determines a node's likelihood to join distinct communities over a certain time interval. These communities can be defined as groups or neighborhoods of nodes that are connected at some point within the whole defined interval. While many network metrics can be extracted, using this technique, we examine two key network metrics to describe the change in dynamic neural activity. We look at network flexibility, which is the average flexibility of all nodes, propensity to join different communities over time, and network promiscuity, which is the average promiscuity of all nodes, or of nodes to join each community at least once over the whole interval. And we further use these metrics to explain a node's high or low flexibility within the larger network. These allow us to investigate the nuance in dynamic neural activity within the frontal brain region. From our analysis, we find that neural flexibility and network promiscuity of frontal nodes, specifically within the gamma and alpha band, during engagement with content online, are significantly associated with opinion change. The interesting and potentially surprising finding is that neural flexibility and promiscuity are significantly higher for participants who did not change their opinion. This means that network promiscuity and flexibility within these nodes in those participants who did not change their opinion join only a few network communities in the brain during opinion change. These results indicate that coactivity of frontal brain region or other network communities explains an individual's likelihood to alter their position on a sensitive issue. This work confirms prior work showing frontal nodes are significantly involved in this process. However, these findings are a little surprising because we would think that a neural flexibility would be associated with a more complex cognitive process like fluid intelligence or something like that. But we have to further investigate these findings, and our team is currently doing so. And so, yeah, Aditi's work, I actually work with Aditi, who's also here and will be presenting shortly, but thank you.
Thank you very much for the talk. So we're still waiting for questions from the audience. Please don't be shy. Use the Q&A tool. Yeah, in the meantime, while we get some questions, I'm wondering, I'm very curious, what is the application of so how these results can be brought into the public, or I don't know, or eternally in the army? How is this used?
Yeah, so I mean, this is in terms of army relevance. When we apply these findings in terms of we think about individual soldiers in teams working together, or potentially using some sort of information online, or like working with people who might be influenced by biased content. So the way that, by understanding how opinion change works, we can potentially mitigate malicious intent to change someone's opinion online. So our work basically just shows that it gives more detail into the neurological framework of opinion change. And I think there's probably many applications in psychology and other, but yeah, I think in general, it's more about potential interventions that could be developed to study terrorist organizations or extremist groups, and like how that content could trickle into certain communities online and affect various organizations and groups in the army or in civilian circles.
Yeah, thanks for the answer. I think Sophia is asking if you can explain how all this may interact with fluid intelligence a little bit more.
Yeah, so we are investigating that now just because the finding was surprising. So fluid intelligence research shows that neuroflexibility and network flexibility and network promiscuity are higher in individuals who show higher fluid intelligence abilities. So comparing neuroflexibility to opinion change, which are both cognitive processes that incorporate metacognition, we would expect some sort of similar neural underpinnings in both of those processes. And so we anticipate that that potentially what's happening is that individuals who don't change their opinion may be spending more time thinking about or more time analyzing content that disconfirms their original beliefs. So if you see something that if you see something that you agree with already, you're not really thinking about it that much. But you see something that you don't agree with, you have to like assess your own belief while processing that. So that involves some more complex process, such as similar to fluid intelligence.
Mm-hmm. Yeah, thanks.
Can I ask a question? Sorry.
Yeah, sure. Please go ahead. Yeah.
I because I'm nervous about my talk, I wasn't paying attention to the beginning. When did you measure the EEG? Was it while they were doing the task, or was that just like offline when they were just sitting, or was it resting state or something?
So it was while they were doing the task. Okay. So there's resting state, and then we have while they were using the platform, and while they were doing the surveys and using the platform, but we only looked at their survey, the differences in the EEG recordings of individuals while using the platform who changed their opinion versus not.
Okay. And do you think that it's a state or a trait-based thing? Like, do you think there are just people who are going to be one way, or do you think you can apply your measure to say this person is particularly in a mood to not change their opinion or something?
I definitely would say that it's probably, assuming that it's a trait is hard because, or I think just not reasonable. I would think that that this opinions and the responses that the neural responses and data that we collected would probably vary depending on certain underlying beliefs and conditions in that scenario. So we actually have more data actually that was not included in this study, but we also recorded EEG data of these same participants discussing the content that they saw together in a group. So from that, we can compare that data to see if somehow, because of those social interactions, their neural activity changed from when they were using the platform individually. So that's something that we're going to explore soon.
Cool. He's mute.
Thank you, Zita. I was typing. Yeah, no, I'm wondering if you know what are the proportions of people who are more prone to change their opinion than not.
Yeah, I don't know I don't remember the exact numbers off the top of my head, but there definitely are more individuals who didn't change their opinion than did from this experiment. But there's also some other members of our team are also investigating if the type of content. So the scenarios were corporate decision making, politics, your opinion on vaccinations and the death penalty. So obviously some of the information in those scenarios incites a different level of attention and emotion that depends on the individual. So someone might care a lot about vaccines while someone might be very anti-death penalty. So it definitely, I think it depends on the scenario as well. There's probably a lot of factors that we are investigating right now, actually. So. Yeah.
Okay. Very interesting topic. Good luck with the future work, and thanks for presenting to us. I think we're going to move on now to the next speaker. She's Zita Patai. She's a postdoc at the University College London, where she's investigating the neural correlates of navigation in humans. She uses fMRI and MEG, among other tools. And today, the title of her talk is Conflict Detection in a Sequential Decision Task is Associated with Increased corticosubthalamic coherence and prolonged subthalamic oscillatory response in the beta band.
Well, thanks very much, Alex, and thanks for everyone in the NeuroMatch community for making this possible. My title is a little bit of a mouthful, but it kind of summarizes it. So if you don't get my talk, you can read the title and get the gist. And normally I would freestyle this, but given the time limitations, I've stuck to a little bit of a script. So no jokes for now. Okay, so we usually explore conflict in sort of these sort of simple contexts. Like here you can see this is called a flanker task. And what you can see here is that the peripheral stimuli are facing the opposite direction than the middle stimulus. And this is what you would call an incongruent trial. And these are sort of termed conflict trials. They're basically when your prepotent response needs to be inhibited to some extent. So these sort of conflict trials have been associated with an increase in theta power. And that's true for the frontal cortex and the subthalamic nucleus, which is what I'll be talking about today. So the subthalamic nucleus has been proposed to be this sort of decision gatekeeper via sort of changes in oscillatory power. Recently, however, there has been some evidence that theta power might also provide information about conflict, but sort of across trials rather than within trials. And there is an increased coherence between frontal cortex and the subthalamic nucleus, but the locus of this cortical coherence is not clear yet. Additionally, most of these studies, and what I'm trying to show here on this little image here, kind of look at conflict on a sort of moment-to-moment basis, rather than looking at it over sort of a long period of time when we accumulate evidence and multiple bits of conflict may come and go, much like in real life. So to study this in more detail, we developed a sequential decision-making task, and it's very simple. Participants are shown cues. There's only two of them. There's either a left-facing mouse or a right-facing mouse that you can see here. And essentially, they're presented these mice in a sort of pseudo-random order, and they have to make a decision about whether they think the mouse will run to the left or the right. So it's just a plain evidence integration task. But importantly, you need to accumulate the evidence over these discrete time steps, and you terminate the search by yourself. So if you think you've accumulated enough evidence, you can make a response. So the reason I've included these ... dots here is because you can take as many cues as you want. You can sample as many as you want. And to study the neural correlates of this sequential decision making, we used 13 patients implanted with deep brain stimulation electrodes in the subthalamic nucleus and whole head magnetoencephalography, as well as 13 match controls who obviously only were in the non-invasive MEG scanner. Quickly, the behavior. Patients obviously worse than controls, but still above chance. More importantly, the interesting thing about patients was that here I'm plotting a histogram of the number of cues they sampled, and you can see on the simple bar graph above, patients numerically do sample more cues, but they're also sampling them very suboptimally. So for example, here in red, you can see some patients are sampling a lot of 1 or 2 cues. You can't really integrate evidence then, and they're sampling a lot more cues sometimes. And nevertheless, compared to a Bayesian integrator, an optimal Bayesian integrator, they were suboptimal. And in terms of strategy use, it seems that most people, patients and controls, are sort of integrating evidence, but some of them are also relying on the fact that they respond when they get 2 matching stimuli at the end of a sequence. So if you see 2 left mice, you're more likely to make a response than if you see left-right. So sort of kind of ending on a same response, a non-conflict trial. Okay, and so for the For the LFP and the MEG recordings, I'm just going to go over how I've defined the conflict. I'm calling it same and different. You could call it match, non-match, low conflict, high conflict, depends who you are and what you want to call it. But essentially, within the sequence, for every single cue, I defined whether it's the same or different as the one right before it. So here you see two right-facing mice. I would call this cue, which I call QI, a same trial, and this would be a different trial. And remember, I'm doing that at every single time point, except the first and last trials, obviously, where I can't define that. And I'm just going to look at what's happening in the brain. Okay, so first let's look at the subthalamic nucleus. I'm plotting beta power, and these vertical lines represent the onset of the cues. QI is the first point where I define same or different. And what you'll notice is that very early after the onset of this cue, there's a difference between these same and different trials. And what you'll also notice is that even though at first, strangely, beta power seems to decrease, it eventually does increase. So it does seem like this conflict information is carried over to the next trial. And that's more visible here when you look at the subtraction. So there's this first kind of dip in beta power, but then it increases. And we know that beta power increases can be associated with response inhibition. In the cortex, it looks exactly the same. So just for the interest of time, you can just look at these two difference waveforms. They look very similar, but you'll notice two things. In the cortex, the signal starts a little bit earlier, so it seems like the conflict signal may originate in the cortex, and it also kind of completes itself within that first match or non-match cue, rather than carrying over. And when we look at where the signal comes from in the brain, we find that there's occipital, temporal, and frontal sources where this sort of conflict signal arises. And then in order to kind of understand the interaction between these brain regions, we looked at coherence. And here I'm just plotting time frequency plots with coherence frequency on the y-axis, and then 0 is the onset of the cue. And you'll notice very nicely that the frontal cortex and the right subthalamic nucleus show an increase in coherence when there's a conflict. So that means these two brain regions are communicating when there's a sort of conflict detected. And if you want to look at it more specifically, you can see it's in this kind of low beta band. You get a really nice peak and difference in coherence. And some of the more keen listeners will think, why? She said that there was a decrease in beta power. And yes, I did say that. There is a decrease in beta power, both in the cortex and the subthalamic nucleus. Nevertheless, in beta power, there's also a coherence increase. So summarizing that here, this is extremely busy and we don't have time to go over it, but just the gist of it is coherence between the areas increased, and power itself, both in the frontal cortex and the subthalamic nucleus, is actually decreased during this initial time period when the cue comes up, when you would have that conflict. Some of you may be wondering why I banged on about accumulating evidence and then looked only at local conflict, which seems really boring. The answer is because we actually did look at different models of evidence integration, and we found no evidence that the subthalamic nucleus does any sort of normalization, evidence integration, or any sort of stimulus number, which I call here urgency signal. So basically, there was no evidence that there was any sort of surprise signal or sort of expectation violation in the subthalamic nucleus. Obviously, we do integrate evidence as humans, and we know the patients and controls did it as well. So we also looked at our whole head MEG data, and we found that indeed in the beta band at the cortical level, there is evidence of some sort of evidence integration. So we are integrating evidence, but maybe not in the subthalamic nucleus. So quick summary, patients sampled more cues, but they were still suboptimal. We found beta power to be the signaling of conflict. I didn't show any theta results because they weren't there. So the kind of classic idea that theta is signaling conflict, we were not able to replicate that in this sort of different task that looks at evidence accumulation across time. Beta power starts earlier in the cortex for conflict. It's more prolonged in the subthalamic nucleus. There's an increase in coherence, and there's no integration, evidence of this integration or surprise signal in the subthalamic nucleus. And kind of really just being hopeful, we think that this pathological amplification that we saw in the subthalamic nucleus, so this carryover of this conflict signal, might be something that leads Parkinson's patients to have more impulsivity or motor impairments. Very speculative. Nevertheless, thanks very much to my supervisors, Vladimir and Rafal, and to you for your attention.
Thank you very much for the talk. There was a lot of information there.
Oh, sorry.
No, that was cool.
I have loads of extra slides with other data if people want to see it. So.
I mean, I would like to, because I'm not an expert in the topic, but it was very clear, I think. So yeah, we don't have questions from the audience yet. I found particularly interesting. There we go. We have a question, so I keep mine for later. So this is a question from Alisa Moore. Does the data on cortical STN coherence point to involvement in the hyperdirected basal ganglia pathway as opposed to the indirect pathway in this conflict behavior? And she's saying both have been implicated in motivated behavior and impulsivity in animal models. Can be the question.
So great question. And in the preprint, we do have a little discussion about the hyperdirect pathway and how that might be involved. Indeed, there is some evidence that sort of this lower beta, which is kind of what we're finding, might be indicative of this hyperdirect pathway. I don't have any sort of DTI data or anything else on these patients in order to kind of look at sort of connectivity and things like that. But in the little QR code in the corner, you can maybe go look at our preprint and see what we said about it. But this isn't really my background, so I'm not going to say anything more than that because I'm not sure I can answer it properly.
Cool, thanks. And I think if you are not scanning the QR code, it's the preprint on bioRxiv, right?
Yes, yes.
I'm copying that again in the chat. So I was about what I found most interesting. So this connection between the coherence between the subthalamic nucleus and the frontal cortex. Could you go back to that slide?
Yeah, sure. This one. Sorry, this one.
Yeah, exactly. So could you say a few more words about this, or how
yeah,
that?
So there is like really a handful of studies, obviously, because these are patients. You can imagine there aren't that many studies on the subthalamic nucleus invasively anyway. And a couple of studies have looked at sort of what's actually happening, and because a lot of the sort of healthy neuroscience shows that the cortex does all this great Bayesian integration, but then there are all these models of the subthalamic nucleus that claim that it does some form of normalization or something along those lines. So that was why we designed this task. But it turns out that really the only thing we can find is that the frontal cortex, which by the way, it's MEG data, so pinch of salt, but it actually was localized to BA6, so supplementary motor area, rather than the inferior frontal gyrus, which is often more involved in these stop signal go, no go tasks. So that was a bit surprising, but again, no one has ever looked at it with sort of MEG where we could actually source localized. So this is the first bit of evidence that maybe that like a more superior frontal area would be involved. And indeed, what we find is that when there's conflict, these two areas are communicating more. So I think that kind of makes sense. It's a little bit disturbing that it happens in that time window when the queue is actually up, but we'll see what the reviewers say about that. But we couldn't really find anything that would indicate that that would, diminish the results necessarily.
Thank you. Maybe there are questions from the other panelists? No? Okay. So what do you keep working on this or?
Yeah, I mean, my background's actually the hippocampus and memory. And this was sort of I've always really wanted to work with deep brain stimulation patients. So this kind of happened randomly, but it actually has led me to want to study the subthalamic nucleus a little bit more, not just from this sort of decision-making or conflict detection purpose, but maybe its involvement in other circuitries in sleep and memory consolidation. So that's why I kind of said, I'm sorry about the question about the hyperdirect pathway. I just don't feel comfortable enough talking about that necessarily. But this data is actually available online, both the MEG data and the LFP data and all the scripts. So if anyone wants to play around with it and find something else, that'd be really cool. I did really beat the horse or whatever you call it. Really did try to get everything out of this data that I could, but there's probably a lot more in there to look at. And I didn't show any of the behavioral modeling we did on the patients and the controls, but essentially the idea was that we would look at this sort of testing this model of Bayesian normalization in the subthalamic nucleus. And as I show here, there is really no evidence for that. So I think one of the benefits of, science, or rather this study specifically, is that the senior author was the one who came up with the model, and we didn't find evidence for it. So it's kind of good to update those models of decision making, I think.
Mm-hmm. Cool, thanks very much,
Thank you.
Zita. Anybody said anything? No. Yeah, thank you very much. And yeah, data available, that's great. Open science. So any attendee here, keep on working on that. So yeah, we're going to move to the last speaker today. Thank you very much, Zita. The next speaker is Aditi Perijanan, and she's a third-year undergraduate student at Tufts University and a researcher as well at the U.S. Army Research Lab as our first speaker. Yeah, looking forward to listening to her talk as well, which is on neuroprocesses underlying contextual influences on decisions. Welcome, Aditi.
Thank you. So my name is Aditi, and I work with the U.S. Army Research Laboratory. And today I'm going to present on the social and cyber effects of decision making. So our everyday interactions are affected not only by our own decision making processes and actions, but also the spread and evolution of information. So as social interactions become increasingly online through social media, information consumed through this medium shapes how we think. In this study, we examine the neural underpinnings guiding the impact of information and social versus cyber context on decision making, where information credibility varies greatly. So in the study, participants went through 4 scenarios on various topics related to current issues where they interacted with a simulated social media platform that had real and fake news articles relevant to the topics. And then they also discussed the topics with other participants. They were given a questionnaire at the beginning of each scenario after interacting with the simulated social media platform and after the discussion group to measure how their opinion changed after these interactions. And throughout the entire process, they were also wearing an EEG headset so that we could measure their neural electrical activity during opinion change. So I can explain one scenario as an example. The scenario is about vaccination. So the participants started by answering an initial questionnaire that asked whether they would vaccinate their hypothetical child. And then they read articles on the social media platform about the effects of vaccination, some of which were in support of vaccinations and others were against them. And then the participants answered another questionnaire repeating the same question of whether or not they would vaccinate their child. Finally, they discussed it with other participants who also read these articles, and then they filled out a final questionnaire on whether or not they would vaccinate their child. So in order to quantify opinion change, I assigned each participant a score for each scenario from 0 to 1, with 0 being they did not change their opinion at all, and 1 being switching to the opposite opinion. And then I added this up for all 4 scenarios to get a score from 0 to 4 for opinion change. So on the x-axis is their opinion change due to the simulated social media platform, the difference between the first 2 questionnaires. And on the y-axis is their opinion change score from 0 to 4 after the discussion, so based on the difference between the second and third questionnaires. And when I plotted this data, I noticed a slight linear trend, which might tell us that some people are simply more prone to opinion change, regardless of whether they were influenced by social media or in-person social interactions. After this, I divided the participants into groups based on whether they were more cyber influenced, socially influenced, both or neither, so that we could evaluate whether individuals in these different groups also differed in their neural activity. So the hypothesis that drove our analyzation of the neural data was that variability in opinions would correlate with variability in neural activity. So data was captured throughout the study using an EEG headset that placed electrodes on the participants' heads and measured the electrical activity of their brain. And then we separated that data based on frequency into 5 different bands. And once the data was filtered, we analyzed using 2 different methods. The first was traditional power analysis, which measured how each frequency band varied in electrical power. And the second method was the relatively newer networks approach, which measures the variation in connectivity with a statistical relationship among nodes across time using graph theory. And using those 2 metrics I just described, I created box plots of the coefficient variation for each group of participants to determine if these metrics could be used to differentiate groups defined by how they change their opinion. So there's also that showed that the respective neural pathways of information processing versus digital interaction and in-person interaction were not statistically different, suggesting that similar neural processes serve both types of decisions regardless of whether they were influenced by social media or an in-person discussion. So our results suggest that different scales of neural processing underlying decision-making are at play when individuals are presented with a variety of reliable and unreliable information in different contexts. So going forward, we hope to consider different neural metrics, such as the one that Kristen talked about earlier today, as well as looking into if spatial or temporal activity varies across different opinion change groups. And we also want to further consider how people's opinion change was affected by how much their discussion group varied from their own opinions. And we hope that better understanding opinion change and people's influence by in-person interactions and social media will help us potentially protect people from being affected by misinformation and fake news. And I'd like to thank everyone that worked on this study at the US Army Research Laboratory, DARPA, and Advanced Brain Monitoring. Thank you.
Thank you, Aditi, for the presentation. It was very interesting, and that touches a very relevant topic as well, especially currently with so much social media and discussions and so on. So again, we are welcoming questions from the audience. You can use ideally the Q&A tool in Zoom, or you can ask directly as well in the chat. While we get questions, I have one. So it's not clear to me. So from the presentation, how maybe you mentioned this, but how do you exactly are measuring? How are you exactly measuring the changes in neural activity? Can you say a few more words about that?
So we had them wearing an EEG headset the whole time. Mm-hmm. And the two metrics I looked at here were power. So that was like electrical power of EEG in each of the 5 frequency bands. And then the second one was network strength. So that's based on the connectivity of nodes using graph theory.
So, and I guess from the EEG, you could localize at least roughly the location of the changes in the neural activity. But is there, so because my broader question is, so what would be the interpretation of the changes of the correlation between the change in opinion and the effect on the neural activity? It would be very interesting to know more about all that. So can you say?
So for these particular metrics, it looked at activity across the whole brain rather than like specific regions, but that is something we definitely want to look into to see if there is a difference between groups that change their opinion influenced by different contexts, if they also differed in like which areas of their brain behave differently.
Cool, thank you. So you actually collaborate with each other, Kristen and Aditi?
Yeah.
Okay, that's cool. That's very lucky we have you both. And so what are the directions for future work regarding these studies that you both have presented?
So there, we did like capture more data related to the discussion as well. So like both Chris and I looked mostly at neural activity while they were interacting with the social media platform, but we do want to look into how they interacted with other people in person as well and see how that differed.
Mm-hmm.
Do we have time for another question from me? Just
absolutely, yeah.
I have 2 kind of technical questions. One is, how do you guys make sure that the people? Okay, I'll ask it differently. Did you look for eye movements, and is it the case that, you know, because if people are looking at those questionnaires, some people who may be more anxious or something, I'm just making this up right now, but eye movements could have a huge effect on those sort of neural measures, especially if you're just looking at power and it's not really time locked to anything. So have you thought about that?
We have not considered eye movement data.
just maybe consider like, you know, how like maybe moving around and stuff may actually change those measures. And I don't know anything about, you know, how people change their opinions and stuff, but it might be worth noting that there aren't any other just random physiological things going on that changes that. And then my other question was going to be, you showed the, like your 4 groups. So have you looked at just a correlation rather than trying to group people? Maybe you mentioned that and I didn't hear it, sorry.
A correlation of what?
So, you know, you had your plot and then you made those 4 groups, 1, 2, 3, 4, and then you looked at the changes in brain activity. And I was just wondering, because people are kind of on a spectrum rather than in groups, have you actually just tried to dump everyone together and see if you can see changes that correlate on an individual level with changes in beta and alpha and whatever?
I did try that. I haven't found any significant results
there. Cool, thank you.
Okay, we have one question from the audience. Alisa Moore is asking about if you analyze the specific content of the articles and also the discussions between the participants to see what is the type of information that might be driving the change in opinion.
Yeah, so opinion change across the 4 different scenarios did vary greatly. Like some scenarios, people were just like less likely to change their opinion. So I think we should go back and look at like how the particular context of the scenario and the information they were looking at affected their general opinion change.
Mm-hmm. But so is it like, do you have like different categories of the, you know, of social media articles or is it kind of general?
Yeah. So like each scenario of the 4 had a lot of articles associated with it, about half of which were like in support of the topic and half which were against it.
Mm-hmm. Okay. Thank you very much. Yeah, I think if we don't have more questions from the audience, we have like 4 minutes more. Let's stick around for 1 more minute or so to wait for questions from the audience. You can also ask questions for Zita or for Kristen if you like to make use of this few more minutes that we have. And otherwise, it always takes some time to type questions. Yeah, so Sophia is saying that these have been great talks. I agree. And thank you very much, you all 3, for your presentations and the discussions. And yeah, and enjoy the rest of the conference at Neuromatch, and hope to hear more about your research in the future.
Thank you very much.
Everyone in the audience. The conference at Neuromatch, and hope to hear more about your
Very good. Oh, hang on. I only saw the not the presentation mode. I actually saw the software.
Yeah, that's correct. It's with for some reason at the moment with me with Keynote, I have, when I do full screen, I cannot switch anymore between programs. So I'll just do it full screen when I'm.
And then Arthur, hi Arthur, very welcome.
Oh.
Hello, could you just quickly share your screen?
Sure. Can you see it right now?
Yeah, perfect.
Yeah.
Okay.
Is it full screen?
Yeah, it's full screen. Looks very
Perfect.
good.
Thank you.
Okay, and Melvin, I think we can get started, huh?
Yes.
Okay, well, hello everyone. Welcome to this session here in room 4, and overall theme is cognition, motivation, and emotion. And I'm accompanied here by 3 amazing speakers. First speaker for today is Sophie Falk, and Sophie is from the Max Planck Institute of Human Cognitive Brain Sciences in Leipzig. And she is very curious to understand what is human about the human brain and to contribute the neuroscience community in a positive way. and her talk, is on social gradients. Please go ahead, Sophie.
Let me share my screen in full screen. So thanks everybody for being here. I will speak about how to organize our social brain, and specifically how the intrinsic connectome gradients govern the organization and plasticity of social brain functions in humans. So I really like this photo, but this is not the only reason why I included this, because what is so cool about like being social in humans is that we can like exchange thoughts and we can share our minds with other people. But how we do this and what is the biological basis of this, that is not exactly clear. And this is also behind this like cloud here that is like visualizing it quite nicely. And I have to also refer to Philip Kanske talk, who would be later today, and he will speak in much more detail about the theory and background and all the studies on the social brain. And today I will only speak about the empirical results of my study, but for more background, please also go to Philip Kanske. So the social brain can be divided in multiple subsystems, and social processes are facilitated by both network segregation, like going apart, and network integration, when networks are more closely connected and integrated together. And on the other hand, large-scale gradients reflect the organization of brain function. However, the relationship between the social capacities and large-scale brain organization remains quite unclear, although steps have been made to like make some assumptions and understanding. So to move forward, I use this gradient approach and this analysis approach to look at this integration and segregation and this embedding of social functions within a larger scale brain organization measured by functional connectivity. So what I did to embed the social brain, on the one hand, I had functional localizers of social affective, social cognitive, and attention processing. And attention was measured with a cued flanker task, where you see a cue and a target. Compassion was measured with a video narrative where people spoke about an emotional or non-emotional event, and then people have to give a compassion rating. And theory of mind was measured by a video, and then people have to answer a theory of mind question about the mind and what the person was thinking about, or a more neutral question about some fact about the story. So these contrasts in the scanners, which is published also by Philip Kanske and also Mathis Trautwein, resulted in 3 different fMRI activations, as seen here in the 3 brains. And when you put it on top of each other on the surface, as you see here, you see it's also a certain gradient. So the colors always go from yellow, red to green, like at multiple places in the brain, which means maybe that these functions are also embedded and organized in a way in our cortex. So this was the start of our study, like these task-based contrasts. So to look further at the organization, we took resting state data of the same participants, and we created 3 gradients, like depicting its organization, aligning also with previous work on large-scale organization of the brain. So the first gradient went from unimodal to higher order default regions. The second gradient switched between sensory, motor, and visual regions. And the third gradient was to the multiple demand system on the one hand in yellow versus default mode on the other hand. And if you then embed the 3 colors, so yellow is attention, red is compassion, and green is theory of mind, along these gradients, first in 2D space, you see that the gradients all have their special place along this organism. The functions all have their special place along this gradient. So it's again yellow, red, green, no matter what contrast you see at. So this is interesting, and it might also mean that there's a special embedding of each of these functions, both in the integration and segregation of these cortical regions within their larger scale organization. So to measure integration and segregation, we also created a measure of eccentricity in the 3D manifold, which was made by combining the first 3 gradients and their Euclidean distance from the imaginary null. And it will be clear in the next figure. So here you see that this eccentricity, so to what extent a function is more integrative and more segregated, like it differed also depending on the task at hand. So the overlap between attention and compassion here in pink shows most integrative functions, which also relates to dorsal anterior insula and more mid-cingulate regions, whereas the overlap between the social functions here in light green on the left you see that it's more segregated, maybe because this is also the most distant function from outside processing, such as primary cortices. And then we also took network-specific metrics, and this is all to build up on to understand like how the social brain is embedded and also can change with training. We also see that in this 3D space, the green, yellow, and red all have their special place. And we also have different network metrics, namely the between network distance and the within network distance, sort of designating how they're spread. And also these features, they differ a little bit depending on the task-based network. So this is cool. We show, okay, these task-based networks are sort of embedded in a different way in the functional connectome. But this is really meaningful. So we did various checkup tests to see, okay, do we also see this with other metrics and approaches? And there you see also that the functional communities that can be assigned to each of these functions, they differ. So you see attention is mostly frontoparietal control, whereas affect and is mostly default mode and frontoparietal control, and theory of mind is mostly default mode. So it seems to be like a bit of a shift between these frontoparietal networks versus default mode networks. Second, also meta-analytic decoding using neurosynth, and we use social and some cognitive terms, shows a dissociation between inhibition, executive function, attention, working memory on the one hand for attention functions versus memory, but also some emotion and mentalizing and theory of mind for the theory of mind functions, whereas affect seemed to include a little bit of both, but also goal-directed and affect aspects. So it seems also that on a meta-analytic basis, these task-based activations are distinct. And also we did genetic decoding with the Allen Brain Atlas, where we also see that there are different genetic patterns associated with each task-based activation, and also different trajectory when this expression takes place. So emotional task-based activations, they are associated with expression mostly with early fetal age ranges, whereas both reorienting and executive control, as well as theory of mind, relate more to these later expressed genes, which is quite interesting. And you see also the patterns in the regions, it differs quite a bit. And then the last thing to see if it was really meaningful, if this embedding really made some sense to behavior, we correlated also the embedding with behavior. And what you see is that attention, so individual differences in attention score, they related also to the average embedding of the attention network here in yellow on the top, and the distance between the attention and the theory of mind network. Conversely, perspective taking was associated with the average embedding of the theory of mind, but also a little bit with the social affective network, as you see in dark red on the right, as well as the distance again between this attention and theory of mind network. And compassion did not show a relationship. So I hope you understand, it seems that there is some meaning to this embedding in this functional network of these task-based activations that are associated with these three functions. So moving forward, we were interested specifically whether these functions can also change independently when you train them. And for this, we had a complex training design, which was part of the research study by Tanya Singer, where we trained on the one hand attention through mindfulness, attention, breathing exercises, affect, to talking about emotion and thinking about your own emotions, opening the heart. And on the other hand, theory of mind by thinking about yourself and your perspective and also discussing them with a partner. I sadly don't have the time to go deeply into this, but please look also at the research study books and publications to further understand what we trained here. But all in all, it took 9 months, and each training block was 3 months. And this was done in a crossover design in 2 groups of people, as well as a third training cohort to control for the first 3 months with affect, as you see here. So what did we found? Like, we used this model. So before and after, there was also MRI measurements and also behavioral measurements. So first to the MRI measurement. So what we found is that with presence, so when you do this mindfulness attention, you segregate the brain. So brain regions become less similar and then networks become more decoupled. Whereas with perspective taking, it becomes more integrated. And affect seems more in the mean, like balancing it out. It's more like not giving a significant change. And this was happening in both training cohorts, as you see below. Also, within network, you see again the same pattern of average embedding increasing versus decreasing in perspective change. And last, we also looked at the behavioral relevance because we had behavioral measures measuring change of attention, compassion, and theory of mind. And here we could show that with how this integrative and segregation changed, you could also predict attention, compassion, and theory of mind independently, and the wave maps could not predict any other function than the function that it was trained upon. And what is also sort of interesting is that you see that those people that change most showed most positive weights or most segregation within their task-based networks, sort of aligning with theories about segregation and meaning automation, meaning that you're better at a certain task. So behavioral relevance, yes, was my answer, but it's obviously more complex than that. So to summarize, social capacities are uniquely embedded in the intrinsic functional organization of the brain, and longitudinal analysis underscore that there are different functional alterations depending on what you train, and behavioral relevance has a both topological embedding and change. So our findings shine a light on the relationship between the intrinsic topology of the brain and the dynamics underlying social skill in adults. And a preprint will be out very soon, and I hope maybe even next week. So with this, I want to thank Tanja Singer, Boris Bernhard, Seyuk, Boyong, Mathis, Philip, and Anne for helping me with this study and supporting me. And then I also want to shout out to my lab, and if you're interested in this line of work or broader, follow my Twitter or DM me at any time with thoughts or comments. So with this, I want to say thank you. And yeah, this is the gradient connecting for you to relax a little bit.
Thank you so much, Sophie. Thanks for the interesting talk. Well, for the audience now, you have the chance to ask Sophie as many questions as you want. Well, we've got only 3 minutes, to be honest, but Jannis Bielefeld has a question here. Do you want to ask the question yourself, or do you want me to read it?
No, I did not parcellate it. So,
okay.
Jannis, sorry.
Yeah. The question is, how did you parcellate the brain initially?
I did not. It was a vertex-wise analysis, but we also redid analysis with the Schaefer parcellation, 400 parcels.
Okay. Are there any more? Does that answer your question, Jannis? Do you want to follow up there, or any other questions? Okay, so then I've got a question, which is not necessarily related to the technical part, but I've recently submitted a paper and I talk about theory of mind, and in theory of mind, which is kind of the ability to put yourself into and to interpret and understand other people's intentions. And I put in beliefs, and a reviewer said, I'm not so sure about the beliefs. What do you think about that?
Yeah, interesting question. I guess to capture what is theory of mind really, and whether beliefs would also, I would also agree with you. I would also say beliefs that it also needs to have some inference about the minds of others to know what they want, right? But there might also be a more effective component. So some people also makes distinctions between more effective theory of mind and cognitive theory of mind. And I think that also later Philip will also present there's so many seeing the minds in the eyes, there's so many tasks and formulations of theory of mind that it's also hard to find this common overlap of the process that is underlying it. So, but yeah, to me, I think I've also written in my paper beliefs as part of theory of mind. But
okay, thank you very much. There is another question here. However, we have only 1 minute left. Sophie, can you read this question and maybe write something in so that we can continue?
Yes, I can type the answer.
Okay, perfect. So I would, Yamini, so this answer will be, or the question will be answered by Sophie directly, and we go on, and I'm going to introduce Arthur to you. Arthur is currently a PhD student at the University of Osnabrück in Germany, and his work focuses on the neural basis of joint action and social interaction. And yeah, Arthur, go ahead. We are very excited to hear about your research.
Unmute. So welcome everyone. Sorry for a short delay with the microphone problem. So as Gunnar mentioned, I'm currently a PhD candidate at Uni Osnabrück, and I'm focused on joint action and social interactions. And when one thinks about social interaction in the context of neuroscience, the method that was developed less than 20 years ago, hyperscanning, come to our mind. And at the beginning of my PhD, I had a look at the literature and I decided to do the review. So I decided to bring all the possible papers that existed till last year and try to review to have a look whether all of this makes kind of sense. So let me tell you where did I start it, because if one think about neuroscience, we have people like Hasley Hodgkin who did amazing job defining all parameters related to action potentials. But if you think about explaining cognitive functions on the level of action potentials, we are still really far away from having proper answers. Since around 30 years, we are putting people in the scanner, and you just heard amazing talk by Sophie, where we also get answers about single brains and what's happening there. However, what's missing is the interaction between people. And for that reason, we need to develop new methods. We come with methods as mobile brain imaging. So on the right side, you can see example from our lab. There was a talk a couple hours ago by Annalisa Gerd, where we actually measured real life situation in the supermarket, face perception, and compare that with lab experiments. On top, in recent years, technology develops, hardware develops, and we can build our experiments in virtual space. So we have examples of huge cities where you can not only interact with a screen, mouse, and a keyboard, but actually interact with everything that is possible. And for neuroscience, this is a step we have to go. But all of those methods that I just mentioned have one missing point. What is there missing, in my opinion, is the bringing second person. So bringing more people to the lab or bringing the lab outside. So for many years, we investigated single brains. We closed the door in our lab for our participants, and we don't know what's happening there. And we want to understand the brain. And actually, this brain that we want to investigate is always used in the interactive situation with other people. We are social animals, and we excel in social interactions. And that's why I believe we have to bring second person to the lab. And if we bring a second person, and if we measure two people at the same time, that's what we call hyperscanning. So we have a method of simultaneous brain imaging with different methods. So first of all, we have fMRI. Right now, as far as I know, there is only one facility trying to do that. It's in Japan, trying to do fMRI simultaneously for two brains. On top, we have methods like EEG and FNIRS that you can see on the screen. Both of those methods are becoming more and more mobile. So it's not only, as you can see on pictures, using this second person neuroscience for two brains in the lab, but also one can bring that outside of the lab. And when it comes to FNIRS, it is right now the most often used method for the hyperscanning, because it's really not that prone to motion artifacts as other methods. And that's why it's becoming more and more popular to study social interactions in real life. And when we already measure, when we already make that step from the hardware side, as I presented, there comes another question. What actually do we measure? Why do we need hyperscanning? And what are the statistical methods that are applied in the field? So I try to review this 20 years and put them into some kind of groups. So we have coupling and connectivity measures. So if you think about two brains having certain activity, performing a task together, one can try to think about how those two brains synchronize and try to define it. So example of such a method is a phase locking value, where you can when you can look in the different frequencies, how the phases of different frequencies actually relate or synchronize between two brains. On the other hand, we have correlation and dependence analysis. So really often I mentioned this facility from Japan when they do dual fMRI scans. When you think about regressing everything out that relates to your experiment and thinking what's left there in two brains that still correlate, this is another group of methods used in the hyperscanning. And on the right side, we have two other methods of graph theory measures and information flow. I would like to say that graph theory measures are right now not that explored. There are a couple papers, and I actually believe that that's the way to go for hyperscanning in future, because the same way as we are developing and expanding the field of functional connectivity measures, we can do exactly the same for two brains. We do not have to limit ourselves to think about a network only existing within one brain, but actually about networks that exist between two brains. And at the end, we have methods that relate to information flow. So causal methods that actually try to use Granger causality or partial directed coherence to think about how one neural activity influences neural activity in the other brain. And here I would like to make a point that every time there is a new field, we need time to define And when it comes to a neural synchronization, we already know so much about neural synchronization within one brain. And I talked about those 4 groups of methods. And right now, all of them are referred to as synchronization in our hyperscanning papers. And actually, I believe that in upcoming years, we have to redefine that. We have to think what statistical analysis method actually, what they bring new and how to separate those concepts so we are not sticked into performing absolutely different type of analysis and actually calling all of that interbrain synchronization. And at the end, I would like to talk about cognitive functions that are used within hyperscanning method. And right now here, each of those topics that I mentioned could be a talk in itself. But I just want to make a picture for all of you who are interested about what already happened. So within cognitive science or within cognitive neuroscience, we have so much research on language, on how do we produce language, but we produce language to talk, we produce language to communicate. And right now there is a new dimension of studying that communication by bringing 2 brains to the lab. On the other hand, decision-making. We've studied so much about decision-making in relation to psychophysics, but we forget that every time we make a decision, that decision can influence other people. And already knowing that there is a second person sitting next to me while I make my decision, this influences my decision. And we also have to study neural basis of that. Our life consists of many different cooperative and competitive situations. There is a whole spectrum in between those two sides. And we also have to study that. So hyperscanning already tried. Hyperscanning gives us first insights, what's happening on the interbrain level when we cooperate or compete. And I believe this is a really important dimension. On a lower level, we have coordination and synchronization. So every time we try to do something together with another person, our brains also synchronize. And studying this, it's also part of hyperscanning. And when I talk about synchronization, we can think about joint action. And here we can talk about representations that we create, a representation not only about myself, but representations of the person I collaborate or the representation of a person I work with. And neural aspect of that is a still totally open question. And hyperscanning is the method to answer that. From a higher level of cognitive functions, we have emotion and affect. One can think about synchronization between brains or interbrain networks happening between people in a romantic relationship or people in a relationship between patients and a therapist. Those are really important parts of everyday life. And we already know that behavioral synchronization between patients and therapists is helping the therapy. Right now we can think further and we can think about it in a space of neural activity. We also have studies over 2 heads, so involving more than 2 brains. There is amazing work of Susanne Dicker where they actually measure the whole classroom in relation to the teacher. So those are the things that can happen in the framework of hyperscanning. And the last, we have also intervention methods. So giving people oxytocin and looking how this actually changes interbrain networks is another step, or using THCS, so transcranial current stimulation, to see whether we can causally influence synchronization. And at the end, I also want to mention music as a great space or a great dimension of our everyday life that can be useful to try to understand those interbrain networks. So I try to picture you there's many different cognitive functions that are studied with hyperscanning. And with all of that, I would like to come to a little bit controversial claim. So this year, there was a paper published by Ana Lucia Valencia. It's a philosophy paper. So I'm trying to point that this is an open question, really radical and controversial, but in a framework of embodied consciousness or embodied cognition, one can think whether our consciousness is not only bounded to one brain, but it could be something that is also extended and always happens between brains. So with this point, I would like to start a discussion with you, because it's not that I'm trying to say extended consciousness exists. I'm saying that we have a lot of results coming from hyperscanning pointing that interbrain networks or interbrain synchronization exist. And maybe we can redefine or rethink how we consider our future studies on cognition in that kind of framework. So that would be it from my side. I know that we all live right now in a really difficult times when all of us are staying at home. So you can hit me up on ResearchGate on Twitter if you want to talk about social interaction on hyperscanning. I would love that.
Fantastic. Thank you so much, Arthur. A very interesting concept, to be honest, completely new to me. And I'm pretty sure there must be some comments here from the other panelists. Sophie, what do you think? Do you have thoughts on this? Or, of course, the audience. Please feel free.
I was muted, sorry. But yeah, it's definitely interesting, but it also like is a challenge, right? To measure and also like to know the time lag, because sometimes like for me now, it took me a while to unmute myself, but I guess sometimes when social interacting, it also takes me some time to understand what a person means or the context. So there's also this time lag going on. And sometimes I understand only what people meant like a day later. And their emotions, because it's also very complex, right? Or what would you say about that, Arthur, about these time differences?
I absolutely agree. But I also believe, as a really young person right now, starting my career in science, that picking up on the biggest challenges is the way I would like to go. So I love all work on the single brain. I learned so much from it. And I realized that from learning on the work on the single brain of social interactions, we can move forward trying to improve it. So improving our ecological validity by bringing more people. When you talk about the time lag, so the change, I understand what you actually meant day after. Yes, of course, we can think about months later when our emotions settle, when our emotions in relations to someone are changing over time. And still we cannot study that easily, or at least neural substrates of such a changes, because we cannot measure our brains constantly. I believe that hyperscanning is a way to start right now, to try those really simple paradigms to measure those two brains and to not limit ourselves to measuring only one brain. I believe both sides can profit. So our understanding or between brain networks can actually improve our understanding of single brain, and other way around. Everything we know already from a single brain can actually give us new hypotheses about how to understand those between brain networks. So it's only a new dimension within a neuroscience that I believe still has to be explored. It's around 120 papers, and I believe in each of our single brain neuroscience fields, we have thousands or a couple thousand papers in every field. So that's why I'm trying to bring that more to the public, that it exists, and it's sometimes worth it to think about also those interactive paradigms and include measurements from both participants.
Yeah, definitely. I fully agree. It's yeah.
There's actually a second question here from the audience. Shanhuang asked, do you know what sensory modality raises the highest synchrony? Auditory, visual, somatosensory interaction? Arthur, do you have any thoughts on this?
So when it comes to what sensory modalities, so right now we live in a really visual world. We all interact right now only via our cameras. We only interact with that. So definitely there is a lot to talk about synchrony between brains when it comes to vision. However, this was a really big point for the starting point of hyperscanning field. There were many claims that actually this whole synchronization comes from identical visual, auditory, or somatosensory stimuli. So we overcome that by applying certain statistical methods of randomizing our trials, of randomizing how do we analyze between brain networks to understand what's there apart from those modalities that exist in our everyday life. I hope this answers Shan Huang question.
Yeah, thanks, he says. Okay, it is now past. That means I have to go to our third and last talk for this session. And this talk will be given by Camila Pinto, who is originally from Brazil, and she is now at Northwestern University at the Feinberg Medical School. And she's going to talk about neural and genetic basis for human ability traits. The stage is all yours.
Thank you, Gunnar. 1 second. Can you all see my screen? Yes, okay. So as Gunnar just said, my name is Camila Boni Pinto. I'm a postdoctoral fellow at Northwestern University at Dr. Vania Pikarian Lab. And I'm here to talk about our study on neuro and genetic basis for human ability traits. So we all know, and we saw in the previous two talks, that we judge people based on their abilities. We use tasks to evaluate behavior, and we know that these behaviors can be correlated with different clinical outcomes or even prediction to some diseases. But so far, we don't have a common framework to evaluate ability traits. We usually use individual tasks, as Sophie mentioned, the flunker test to evaluate some cognition and attention, and specific other tests for motor or sensory. Here we choose a framework that is the NIH toolbox that assess these 4 different domains and draw ability traits based on the NIH toolbox. Why we chose the NIH toolbox? This is tool that was developed by the NIH and has been using for more than 10 years now. It's used both in the research settings and in the clinical settings. It's very easy to be performed, and it comprises about 38 different individual tasks to evaluate these 4 domains: motor, cognitive, sensory, and emotional. So in this study, what we did, we used 2 big samples. One of them is the NIH normative data set, which has 1,369 subjects, health subjects with different ages, and the Human Connectome Project data set, which has 778 subjects. As you see, these are big data sets, and we use those to draw 4 ability traits using those individual tasks. Right? First, we did that on the NIH normative data set, and we used the Human Connectome Project data set to validate these ability traits. Also in the HCP data set, we have data on brain imaging, and we use that to draw correlates between brain networks and the specific ability traits. And lastly, also the HCP data provides us with family structure in which we have data from monozygotic, dizygotic, full siblings, and no related patients. And with that, we could calculate heritability based of our ability traits. So as I mentioned before, first we draw the ability traits. We use a common data reduction analysis. Here we use factor analysis. We also evaluate the number of optimal factors that could explain the variance of these traits. And we end up with 4 traits: motor endurance, emotional processing, executive function, and cognitive ability, and social interaction. Here you can see the individual loadings of each one of these tasks, but for now I think we should focus on the 4 ability traits. As I mentioned, we did this on the NIH normative data set, and we applied the same model in the HCP data set, and then we compare individuals in these 2 samples to see if our ability traits were generalizable. To compare, we built clusters using only the ability traits and not using any other characteristics of these individuals. When we compare the proportion of patients between the 2 samples in the clusters, we can see here in the pie plots that they are fairly the same proportion, especially when we go for age matched in the samples. Why? The toolbox normative data sample has much older individuals than the HCP, and this help us to see that these patients end up clustering at the same rate in each one of these types based only on the ability traits. We also compare their demographic and socioeconomic characteristics, and they were fairly similar. This means like they were at the same age and fairly similar gender distribution between the clusters when we use the ability traits to separate these patients in different groups, including also level of education and income. Okay, after that, we ask ourselves to what degree can brain properties predict disability outcomes. So we again focus on the Human Connectome Project data. The Human Connectome Project data give us brain properties in forms of correlation matrix between large brain networks, ICAs, and we regress these brain properties against our 4 latent ability traits. Only 2 of them show significant results in the out of the sample regression, and I'm going to show in the next slide. So one of them was the motor endurance connectivity. We show here that 4 pairs of large brain networks connectivity could predict the performance in the motor endurance ability traits. In blue, we have the model coefficients, and this means that higher connectivity between the default mode network and the visual spatial network correlates with indicates a higher value of a specific patient in this ability trait. And the opposite for the sensory motor and language. Just like to point out that sensory motor here is more related to coordination and speech production. And we show here that the negative relationship between these two would correlate with this specific ability trait. Interestingly, if we go to the literature in research trials that focus on understand motor performance or focus only on motor tasks, these are the same large brain networks that are involved with performance in motor tasks. So we believe that this helps to strengthen the validity of using NIH toolbox as a framework to evaluate ability traits. We also found significant results for executive and cognition and cognitive function trait. And here, 8 pairs of large brain networks entered in our final model. And the same we can see in blue, the blue bars corresponding to the model coefficients, so showing that higher connectivity between the precuneus and the left executive control network indicates higher values of a specific participant on that trait. And again, the same here, the networks involved in this were also involved in other papers that shows that this executive control and salience network are important and associated with working memory in health subjects and subjects, for instance, with schizophrenia and depression. Finally, our last question, also using the Human Connectome project data, is what if we could estimate heritability of these traits? As we know, we all are better in specific behavior or specific tasks, and this has some contribution from our genetic background. So heritability, it's a way for us to evaluate a proportion of a variance of a determined trait that can be related to genetic factors. There are several models to evaluate heritability, and here we use a simple model that is the ACE model, in which tells that heritability is just a sum of additive genetic variance, common environment variance, and idiopathic environment variance that it could be, it's hard to measure because it's something that varies a lot from individual to individual. And we use data from the monozygotics and dizygotic twins. This model tells us that both monozygotics and dizygotic twins share 100% of the environmental variants since they were born at the same time, and these patients live in the same family. But the only difference here would be in the additive genetic variance, since monozygotic twins share 100% of the genetic factors, and the dizygotic twins will share about 50%. So when we correlate the pair of twins between themselves, the correlation values, it's an indication of the sum of the genetic and common environment variance. And with this, we can calculate a broad sense heritability. I put the formulas here just for reference, but we can go back if you have any doubts. And we were able to calculate the heritability only when that regression is significant, right? And this was true just for the 2 same traits, motor endurance and executive and cognitive function trait. This value here varies from 0 to 100, and we show that for the motor endurance, 30% of the variance of these individuals in that trait is explained by genetic background, and for executive function and cognitive function, about 49%. This is very interesting because if you look to other papers evaluating how much of the variance in executive function or in, usually they use working memory tasks. The number is fairly similar. They see that about 40 to 50%, some papers say 70% of the variance can be explained by genetic factors. So again, it validates the use of the NIH toolbox to evaluate abilities. And lastly, we also went to the factor space and measure the distance between a pair of twins or a pair of subjects in this space, meaning that when we measure the distance of monozygotic twins, they are more close to each other in the trait space, meaning they perform much similar to each other than when we compare with dizygotic twins, full siblings, or non-related family members or non-family members, showing again that there is a contribution of genetic for the variance in these traits. The same thing in the brain space, when we measure the distance between monozygotic twins and compare with dizygotic and full siblings, we see the same trend, showing also for the connectivity between these large brain networks. It's more similar when you have a closer genetic background. So in summary, we use the NIH toolbox to find these ability traits, and we could find 4 of them, and they validate in another sample. That was the Human Connectome Project sample. 2 of them, motor endurance and executive and cognitive function, were associated with specific between large brain networks function, and they were significant influenced by genetic factors. So thank you for listening to this presentation, and a special thank you to everyone on my lab.
Well, thank you very much, Camilla. Thanks for the interesting presentation. Are there any questions from either the audience or from the panelists? Any questions from Sophie or Arthur? Any questions?
Yeah, I have a question. Like, given that I actually work on a similar but different project, but I wonder, did you also consider like genetic correlation across traits? And like, are you also planning to look at this, or have you already written it up? Or
no, we did not look across traits, but that's an interesting question. We look within traits, yes.
Okay. And it was not like the emotional stuff was not heritable?
No, it was not. Also, the sample, if we compare the sample of the HCP data that we use for calculating the heritability and the sample that is usually using specific big data genetic papers, the sample is small. We have about, I think we have about 120 pairs of monozygotic twins and less from dizygotic twins. So we speculate that this could be also a reason why the correlation was not significant. It was kind of a lack of power. But yeah, in the sample, they did not.
Yeah, it would also be interesting to see the genetic correlation within each domain to see if actually these tasks, what extent they do correspond or not. Because yeah, that's also always a bit tricky, but like very cool work. Cool.
Okay, nice. Thank you for your comment. We're gonna definitely look into that. Yes.
All right. If there are no further questions, it's almost 45 now. If not, then I'd like to thank all the speakers, and I would like to encourage the audience, if you have questions, please get in touch with the speakers individually. We have actually provided the Twitter accounts as well as the Google Scholar accounts here in the chat box for each of the speakers. Otherwise, I'd say thank you very much for the nice session, and I wish you all a very nice day. Bye-bye.
Thank you so much for hosting.
Yeah.
You will recognize what those are when in the middle of my talk, I guess.
I'll start in a few seconds, see if anyone else joins. All right, welcome to room 4, Neural Match day 4. So in this afternoon session, this afternoon on the East Coast of the United States, good morning, good evening to everybody else. We'll be discussing things that go on below the neck in neuroscience. Not my area of expertise. I'm usually used to above the neck neuroscience, but we have a set of 3 interesting talks on the spinal cord. We'll start with Felix Federling, who is a postdoc in the Dodd and Mason labs at the Zuckerman Institute at Columbia University. You can find him at Felix Federling on Twitter, which we'll put in the chat box if you want to go ahead and follow him. He'll be presenting his work on 3D mapping of the whole mouse spinal cord. Take it away, Felix.
Cool. Thank you very much for the introduction and also being involved in organizing this meeting. It has been very much fun so far. Yeah, and as you said, I will talk about tools that we made to analyze the spinal cord in its entirety. Okay. So the spinal cord is a quite critical part of our nervous system and allowing us to engage with our environment by relaying and processing somatosensory and motor signals. And research on model systems like the mouse has identified major classes of spinal neurons that establish these functions. However, as you heard throughout this meeting and with recent advances in transcriptional profiling of spinal neurons, it has become clear that we have massively underestimated the complexity and heterogeneity of the spinal cord. So to achieve a better characterization of spinal cell types and the detailed functional circuitry, we need to map the spatial relations of neurons and projections in the spinal cord with better precision, and most importantly, in relation to other neurons. So ideally in the context of the entire spinal cord. However, currently we don't have the tools to do this, and classical histology approaches are quite impracticable because the spinal cord is so long, right? And we also lack still a common coordinate framework for spinal cord analysis, making it complicated to map data with precision and to compare data from different samples and across groups. So while we have powerful tools today at hand to identify spinal cells and to trace their circuits, there is a need for an easy to use and high throughput method to analyze the whole spinal cord and to map the positions of cells and projections into a common framework. So and today I will talk about how we address these issues in 2 basic steps. So first of all, how we created tools to make the collection of data for the whole spinal cord more efficient. And then secondly, about how we develop software to automate the analysis of spinal cord sections in 3 dimensions and to map the data to a reference atlas. And so starting with the first problem, we first thought for ways to make the sectioning of whole spinal cord more efficient. And one way to reduce the work and time involved in this process is to split the tissue into multiple tissue segments and embed them in parallel in the same block for synchronous sectioning. So as shown here, by splitting the cervical to lumbar region of the spinal cord into 9 tissue pieces, we can bring down the number of actual sections that we need to cut from over 1,000 to about 100. However, now with these small tissue pieces, it is quite tricky to embed them in the same block in a perfect orientation without confusing their identity. So to help embedding, we developed spine racks, and spine racks are 3D printed support structures that can fit into an embedding mold and help guide tissue pieces into place. And so similar to a wine rack, they hold objects of interest in a preferable orientation. These spine racks, we made them from PVA. This is a polyvinyl alcohol, a water-soluble material that can be softened through incubation in OCT embedding medium after the tissue has been placed and before freezing the block for smooth sectioning through the material. PVA is actually a component of OCT, and after sectioning, any remains can simply be washed away by washing in PBS. This material is commercially available as filament for many 3D printers, and so it's very easy and cheap to produce spine racks in large quantities and also in different geometries for other applications. So using these spine racks, we can collect all sections of the spinal cord within only 1 hour of sectioning time and on only 15 slides, which means a massive improvement in time for both sectioning and also subsequent immunohistochemistry compared to traditional approaches. And in addition, the precise arrangement of tissue sections on the slide also makes it possible to image the sections automatically on a slide scanning microscope like this, while keeping track of the identity of each tissue section. So to address the second issue and to analyze the section data now in 3 dimensions, we developed a processing pipeline for Fiji that we call SpinalJ. And this pipeline follows these basic steps shown here that I will explain in a minute, and with exception of the first step, runs fully automatically after initial setup. The overall hands-on time, including the first step, is currently a half an hour to an hour, depending on the size, the section quality, and also the size of the data set for one animal. So I can't go into much detail, but this first step basically of the pipeline is required to bring the individual tissue section images into a rostrocaudal order, as shown here, and then to sort out empty and damaged sections that are still included, and to come to this and to orient the sections so that we end up with a continuous stack of intact sections that is ready for image registration and 3-dimensional reconstruction, as shown on the right here. And for this registration process, either NeuroTrace, which is a fluorescent Nissl stain, or DAPI can be used. But besides of that, you can, of course, stain for whatever you like. So now, after we have created this 3-dimensional reconstruction of our section data, let's see how we can bring this data into a common coordinate framework and to analyze it in the context of anatomical annotations. And because there is no 3-dimensional atlas for the mouse spinal cord, we have built one from the 2-dimensional Allen spinal cord atlas that consists of 34 annotated sections, one for each spinal level, that were drawn based on Nissl stainings. And so to do this, we first digitalized these annotations of the Allen atlas by filling all the regions with a color code, and then we built a 3D volume from both the Nissl and the annotated sections, simply by stretching the sections of each segment to match the rostrocaudal length of each spinal level, as has been measured by others. So this new atlas does not add any rostrocaudal resolution compared to the Allen atlas, but rather brings the segment annotations into rostrocaudal context and into a 3-dimensional format. And this now allows us to map our experimental data first to the Nissl reference using either the NeuroTrace or DAPI channel, and then overlay the 3-dimensional annotations for analysis in atlas space. So, and as shown here, SpinalJ is able to map sections shown in magenta to the Nissl template in green with very high accuracy using either NeuroTrace or DAPI on the right for registration and mapping. We have also quantified the mapping accuracy of NeuroTrace signal to the gray matter annotations and find across multiple animals an average accuracy of about 90%, confirming the visual impression of good mapping. So after establishing the basics, let's talk about some experimental data. And for this, we first labeled corticospinal neurons by injecting a tomato AAV into one side of the cortex and then mapping their projections into the spinal cord. And using SpinalJ, we can quickly reconstruct the sections and analyze the mean intensities of signal per atlas region, as shown here in the middle. And this is a summary of the data for each segment. And this allows us to quickly assess the distribution of signal across the entire spinal cord. And this on the right is assuming. We can also combine individual atlas regions into more intuitive groups of regions, for example, gray matter laminae or white matter tracts. And this allows us to compare the distribution within these broader regions, for example, also across hemisegments, left versus right. And in our analysis, we see that within the white matter, the signal is mapped to the dorsal funiculus, and within that region, specifically to the corticospinal tract, as we would expect. And in the gray matter, the distribution of signals matches quite nicely to what others have reported at cervical level using manual approaches, with the difference that, of course, now our data set can also be analyzed at different spinal levels. In SpinalJ, we also have options for automatic cell detection and detection of projections, including a tool for machine learning pixel classification that is called Elastic. And as shown in this example here, we where we genetically labeled FOXP1 expressing motor neurons, we can train Elastic to recognize either the soma or the projections, as shown in red and green respectively. And we can then use these probability masks to separate cells and projections from a single image channel and analyze their respective densities in each atlas region. This segmentation approach is also particularly useful when dealing with signal in a high background, as shown here in this example of an IB4 staining of non-peptidergic DRG fiber terminals. And as you can see, compared to the intensity analysis shown on the top, the analysis of the segmentation of the segmented projection densities gives us a much clearer result, showing that these fibers innervate lamina 2. So in my last slide, I want to focus on how spinal J can be used to compare data across multiple samples. And for this, we mapped FOXP1 positive cells from 4 different animals, and we plotted their positions in a different color for each animal here on the left side. And you can see that the distribution of cells from the different animals aligns quite nicely without any obvious color shifts that would indicate major mapping offsets. And when we look at the number of cells in each spinal segment, we see that the cells are equally distributed across animals and that they concentrate at limb levels as we would expect for this type of motor neurons. We also calculated the distances between the centers of mass of the individual cell distributions in each animal and within each segment, and find that the average mapping offset across all segments is around 30 micrometer. So very, very small offset. And this really shows that we can map cells in different animals with high accuracy, and that allows to directly compare these samples. So, and with this, we now have quite a powerful tool, I think, to analyze the positions of all different types of cells, projections, and even synapses in relation to each other. And this allows us, maybe as a community even, to build a detailed map of the entire spinal cord. We're currently finishing up Spinal J and make it available for everyone shortly. But in the meantime, please also feel free to contact me if you're interested in using SpineRex or Spinal J. And so with this, I want to end by thanking the lab, my mentors and collaborators, especially Luke, who did most of the coding for Spinal J, and of course, all of you for your attention. Thank you.
Thank you, Felix. Nice talk. And right on time, great timing. And so feel free to ask questions in the Q&A. I'll start. So in your last slide, you showed very good consistency in terms of registering across individuals. What strain of mouse did you use, and did you expect there to be differences across different strains?
So we looked at some cell populations that we labeled across different strains, and we don't see major differences, but maybe this is also because if there are any differences in size, maybe of the spinal cord, these will get compensated by mapping it to this common framework, right? So yeah, so the problem at the moment is that the atlas is not very accurate. This atlas is built from a single mouse, right? So the annotations, I think, have to be improved by building an atlas that it comprises several animals, and then you can talk more about inter-animal variability.
Thanks. Any other questions?
Can I ask a question?
Of course, speakers are free to ask questions as well.
Yeah. So Felix, awesome work. I have been missing this for a long time.
Yeah.
Is it possible to use Spinal J to align one individual section? I mean, I have so many pictures of sections of the spinal cords. Can I just use the program to find where it would sit?
So for robust alignment, at the moment, it's still the best if you can tell it what the rostrocaudal extent is of the volume that you want to map. In regions where we have a lot of morphological landmarks, so if you have like motor neuron clusters that clearly identify a certain level, it can do it automatically. But for example, in the thoracic cord, where even the annotations for several segments are the same, there's no way really to map it precisely unless you tell them where it should go.
Okay, thank you.
But and we did this, the spine regs, you can certainly use them not only for the analysis of the whole cord, but sectioning maybe from 9 animals, only a lumbar fraction of the cord, and then map those volumes.
Yeah, nice.
Thanks again, Felix. We'll move on now to our next talk. You can continue the discussion in the chat box. If you have other questions for Felix, feel free to ask them there. And now, our next presenter is Sebastian Choi. He is a postdoc in the lab of David Ginty at Harvard Medical School. You can find him on Twitter at @SebastianChoi, which we'll put in the chat box. He just had a paper published yesterday. Congratulations. So you can also check out a link to that paper in the chat box below. Sebastian, take it away.
Okay. Thank you for the introduction today. And my name is Seung-won and Sebastian Choi. I'm a postdoc in David Ginty's lab at Harvard Medical School. And today I'll talk to you about the project that I've been working on the past few years, which was focused on the ascending spinal pathways that convey effective touch and pain signal to the brain. Every day, we experience a myriad of somatosensory stimuli. The hugs from loved ones, warm shower, and mosquito bite, and sore muscles after a workout. All these tactile, thermal, and itch and nociceptive signals are first detected by primary sensory neurons, and then propagated into the spinal cord where they're processed. And then these signals are transmitted to the brain through ascending spinal pathways. The primary sensory neuron subtypes that innervate the skin and detect a wide range of somatosensory stimuli have been identified and characterized. So here are examples. There are low threshold mechanoreceptor neurons that detect light touch and proprioceptors and nociceptors, and thermoreceptors that detect thermal stimuli, and proprioceptors that detect itch stimuli. And in the field, we have the genetic access to almost every single subtype of primary sensory neurons. In contrast, however, very little is known about how these peripheral signals are integrated and processed within the spinal cord, and how these signals are conveyed to the brain by a spinal cord projection neuron to generate our somatosensory perception and behavior responses. So the goal of my study was to better understand the circuit logic of these ascending spinal pathways. And I've been focused on this anterolateral pathway, a major ascending spinal pathway that is known to convey mainly the temperature and pain information from the periphery to the brain. And these neurons receive nociceptor inputs, and their axons cross the midline in the spinal cord and innervate the contralateral side of the brain. And I was particularly interested in this one region, lateral parabrachial nucleus, because this region receives the most dense innervation from the spinal cord. And this ascending pathway is termed spinal parabrachial or SPB pathway. And again, as I said, we have in the field genetic access to all these, most of the primary sensory neuron subtypes. In contrast, we almost completely lack the mouse genetic tools that label different subtypes of projection neurons in the spinal cord. So we started this project by generating mouse genetic tools to label different subtypes of projection neurons in the anterolateral pathway. And we generated first the TACR1 positive tool because TACR1 positive neurons were the only known population that label a major subset of SPB neurons. And then we also took an unbiased screening approach to identify unknown and novel subtypes of projection neurons. So we conducted in silico screening of over 1,300 the back GFP transgenic mouse lines from the genset. And we identified this one line, GPR83-GFP, labeled a major subset of SPB neurons. So these neurons are mainly located in the superficial lamina of the spinal cord dorsal horn, where the projection neurons are enriched. And with the retrograde labeling experiments, I was able to show that these neurons label a major subset of SPB neurons. And with the mouse genetic tools that we developed, we were able to visualize both of these populations at the same time. So here in these images, TD tomato neurons represent the TACR1 positive projection neurons, and green neurons represent GPR83 neurons. So as you can see here, these two neurons are largely non-overlapping populations. And then we looked at the brain target innervation of these two SPB subtypes. So interestingly, it turns out that the TACR1 positive neurons mainly form synaptic terminals in these two subnuclei of the PBN, whereas GPR83 projection neurons uniquely innervate DL and EL subnuclei in addition to the IL subnucleus. These results suggested these two SPB neurons innervate the PBN in a zonally segregated manner. So now we have these two anatomically distinct subtypes of SPB neurons. So next we wanted to investigate the function of these two projection neurons, particularly what somatosensory stimuli or signals they convey to the brain. So before I go into the detail of the data, I wanted to give you some background. So textbook view of ascending touch pathway mainly emphasizes the dorsal column medial lemniscal pathway, where the touch signals are first detected by mechanosensory neurons, and these signals are transmitted to the spinal cord and brainstem and to the thalamus, and then somatosensory cortex. So this is a classical touch pathway. In contrast, as I said earlier in the introduction, anterolateral pathway, mainly known to convey the pain and temperature information to the brain. However, we think that this newly identified GPR83 positive SPB pathway form dedicated and unique touch pathway within the anterolateral tract that convey tactile signals from the spinal cord to the lateral parabrachial nucleus. And the PBN in turn broadcasts these tactile signals to other regions of the brain, including the amygdala and hypothalamus. So I'll walk through some of the data that support this idea. So we first wanted to look at the physiological response properties of these two projection neurons. So we collaborated with Sarah Ross and Rick Corber's lab, and they used this ex vivo skin spinal cord preparation. And Junichi, the postdoc at the time, performed wholesale patch clamp recordings on these targeted projection neurons while giving different types of stimuli onto the skin. So he measured the change in the firing rate upon the application of different types of somatosensory stimuli. And one of the most unique properties of GPR83 neurons was that compared to the TACR1 projection neuron, they're highly sensitive to mechanical stimuli. So here in this summary rate of plot, the left panel is a percentage of responders and right panel is the firing rate. As you can see here, the GPR83 projection neurons are they're very responsive to the wide range of foam-fry filament skin indentation. Now, when they respond with much more higher firing rate. And whereas the TACR1 projection neurons are more responsive to thermal stimuli, especially in the innocuous range. Here's a summary plot. So to define the spinal cord circuit mechanism underlying this mechanosensitive response properties of GPR83 projection neurons, we performed the channelopsin assisted circuit mapping experiment using acute spinal cord splice preparation, and also this complicated mouse genetic strategy that allowed us to specifically stimulate different types of sensory neurons while recording from specific types of the projection neuron in the spinal cord. So axonal terminal stimulation of mechanosensory neurons, including the MRGB4 positive non-peptidergic neurons and TRAC-B positive a delta low threshold mechanoreceptor neurons, evoked large light activity currents and also action potential firing in GPR83 projection neurons, but not in the TACR1 neurons. So this suggested GPR83 neurons receive strong synaptic inputs from the mechanosensory neurons, and this can explain its mechanosensitive response properties. And then we turn to the anatomy of the spinal parabrachial pathway. What is really interesting about this pathway is that they have a unique organizational property. So in this experiment, I injected a cre-dependent alkaline phosphatase virus unilaterally into the right-hand side of the spinal cord and performed tissue clearing and the whole mount AP staining to visualize ascending axonal projection to the brain. And as I said earlier, the ascending pathways within the enterolateral tract, they mainly innervate the contralateral side of the brain. For example, here in the midbrain, PAG or superior colliculus. However, the PBN receives predominantly bilateral inputs from the spinal cord. Another interesting anatomical feature of this pathway is that they lack somatotopic organization. So in this experiment, I injected three different colors of synaptophysin viruses into cervical, thoracic, and lumbar spinal cord, and I examined their synaptic terminal distribution within the PBN. And surprisingly, these three different colors of synaptic terminals were intermingled within the PBN, suggesting that there's no clear somatotopic organization in this spinal parabolical pathway. So these unique anatomical properties led us to suspect that maybe this pathway convey affective component of somatosensation rather than discriminative component of somatosensation. To directly test this idea, we performed this optogenetics coupled instrumental conditioning assay where animals receive selective optogenetic stimulation of either of these SPB pathway when they press the active lever but not inactive lever. So we tested a different optical strength, and we started with the high power, strong activation of either of these pathway and then dialed down. And down to the 1 milliwatt strong activation of either GPRE3 or TECR1 pathway reduce the lever presses over time, suggesting that a strong activation of these pathways is aversive to the animals. And surprisingly, when we stimulated the GPR83 with the low intensity light stimulation, it promoted positive reinforcement. In other words, animals increase the lever presses over time, suggesting that low intensity stimulation of GPR83 pathway is appetitive to animals. So these results suggest that this GPR83 pathway can be associated with either positive or negative valence in a scalable and stimulus intensity dependent manner. To summarize, I didn't have much time to show you this part today, but we showed that these two pathways form parallel ascending circuit modules that cooperate to convey nociceptive signals to the brain. And as I described today's talk, this newly identified GPRE3 pathway constitutes a mechanosensory limb of the enterolateral pathway that conveys an effective component of touch. So I showed you these neurons are highly sensitive to mechanical stimuli. They receive strong synaptic inputs from the mechanosensory neurons, and they form bilateral and non-somatotopic organized synaptic inputs into the PBN. And they can induce the behaviors associated with either positive or negative valence in a stimulus-dependent manner. So overall, our study reveals these two anatomically and physiologically and functionally distinct spinal parabolical pathway that underlie effective touch and pain. So with that, I'd like to thank David, his support, and all the people from David Gintis who contributed to this work, and a collaborator, and the mouse lines and the viruses and the funding source. And with that, I'll take any questions.
Thank you, Sebastian. Very nice talk. We have a question from Asaf. In intact animals, is it rare to record responses of PB neurons to tactile, non-noxious stimuli? Can you comment how this relates to your findings?
So that is a good question. So I think one of the issues that mostly in the field, people have done recordings in the PBN, anesthetized animal. So I think one possibility is that maybe anesthesia can affect the activity of these neurons. And also, as my work sort of suggested that probably depending on the stimulus intensity, it'll recruit. So if you increase the intensity of the mechanical stimulation, it will recruit more and more neurons activated by mechanical stimuli. So I think one might want to have really sensitive physiological measurement to really detect the units that are responsive to the innocuous mechanical stimuli. But at least in our ex vivo preparation, we see pretty strong response to the mechanical stimuli.
We have another question from Rebecca Bohm. Great talk. Is the SPB pathway in mouse comparable to that in human? Do you believe we can expect this pathway to perform the same function in humans?
Oh yeah, that is a really good question. Actually, in humans, this spinal pyruvate pathway has not been much studied. People mostly focused on spinothalamic pathway that goes to the signal goes to the somatosensory cortex. But there are a few the human studies. Actually, some of the human patient studies suggested actually this anterolateral pathway can convey both negative valence associated with the pain signals and also the gentle touch, or pleasurable touch information associated with the positive valence associated with the gentle touch. Because those studies, what they showed was, for example, the patients with the terminal cancer, they have really extreme pain. So one of the strategy to reduce the pain in those patients was to transact the ascending the anterolateral pathway. So basically cutting the anterolateral pathway. And these patients have reduced the pain sensation, but also oftentimes they also show they lose the kind of pleasant feeling, the present feelings associated with the gentle touch, while maintaining the ability to locate or identify the source of the sensory stimuli. So I think those human studies kind of suggested maybe in humans also have conserved roles in terms of conveying this affective component of somatosensory stimuli.
Thanks, Sebastian. Again, we can continue the conversation via chat or Twitter. Sebastian, you have a question in the Q&A that if you can answer in written form, that would be great. We have to move on to the next talk.
Okay, can you see the presentation?
Yep, so your presentation. So Augusto Escalante, he's a postdoc in the group of Herrera lab in the Neuroscience Institute in Alicante, Spain. He'll be presenting his work on how the spinal cord gates self-generated mechanosensation.
Yeah, thank you for the presentation and for the opportunity to present here. So I'm going to be telling you about the population of spinal inhibitory neurons that are characterized by the expression of PTF1A in controlling self-generated itch. So thanks to the talk of Sebastian, I can go a bit quicker on the organization of the spinal system. I will just start by introducing you how the overload of information that we are receiving in a similar fashion with all the social media we are being bombarded nowadays. The nervous system is also continuously receiving stimuli, external stimuli that has to process. Not only that, the nervous system also has to cope with self-generated sensory information that is generated while we move, right? All of this information goes to the nervous system and it needs to develop a way to distinguish between both. So I'm interested in somatosensation, and as you have seen in the previous talk by Sebastian, this somatosensory modalities starts at the skin, which is not only a barrier to protect us from the external world, but is also our largest sensory organ, given that it's covered by different types of receptors that transmit all this information, like the ones cold, heat, pain, touch, or itch, just to name a few that we are all familiar with, to the dorsal horn of the spinal cord. Now, the dorsal horn of the spinal cord is composed by two types of neurons, like the rest of the nervous system, excitatory and inhibitory neurons. For the purpose of this talk, I'm only interested on the inhibitory component. And thanks to the work of many labs across the years, we know that inhibitory neurons that populate the dorsal horn are specified by the transcription factor PTF1A, and the mature neurons express the marker PAX2, another transcription factor. We know also that the mutants for PTF1A lose all inhibitory neurons in the dorsal horn, so that's pretty well established. And we also know where and how these populations are specified and evolve and migrate through embryonic development. Now, until you can see that in the adult, they populate the superficial layers of the dorsal horn. Now, to start to unravel the function of these neurons in somatosensation, the first thing I did was to perform monosynaptic rabies tracing and to in a specific PTF1A Cree mouse line, right? Here you can see how sensory neurons in the DRGs are labeled after injecting the modified rabies virus in the spinal cord. Using a battery of markers that we're not going to spend much time, we saw that most of the inputs that the PTF1A cells are receiving are related to touch, so to light mechanosensory information, and not much pain or proprioceptive sensory information. They also receive motor related information coming mostly from the motor cortex and from the gigantocellular nuclei. But we also saw that these neurons make around 86% of all of these special type of synapses that many neurons establish in the dorsal spinal cord, that it's directly on the terminals of the sensory afferents before they reach their target neurons, which is a way to control the somatosensory input information into the spinal cord. So in order to work with this population specifically in the spinal cord, we have used an ablation strategy that has been previously published by the Goulding lab, in which cells that express both Cree and another recombinase FLIP leads to the expression of the receptor for the diphtheria toxin. Here we cross these animals with double PTF1A Cree CDX2 FLIPO. This mouse line allows us to gain a spatial specificity, given that it's only expressed in the caudal part of the body, so that we can get expression of the diphtheria toxin receptor only in our PTF1A Cree cells in the spinal cord, and not, for example, in the cerebellum, where PTF1A is also expressed. Then, upon delivery of the diphtheria toxin, we can eliminate those cells. Now, what we got was an ablation efficiency of around 40% of all the PTF1A Cree-derived neurons when we evaluated using a tomato reporter, and a similar decrease of around 36% in the number of PAX2 neurons in the dorsal horns of the spinal cord. Now, what happens with these animals? You can see in this video very clearly, they develop a very intense itch phenotype. So these animals scratch all the time, which is around 22 times per minute, which compared to a control that scratches around 0.2 times per minute, it's really crazy. They scratch around 100 times more than control animals. However, and interestingly, what is known as chemical itch is not affected. So if you inject any pruritogen into the skin, the animals will not scratch more than controls. Also, no other somatosensory modality that we could test for was affected, except for the sensitivity of the hairy skin. So if we apply very light air puffs into the hairy back skin, we see that PTF1A ablated animals respond more strongly than control animals. So their hairy skin's sensitivity was higher than in controls. So to gain a better understanding of how this phenotype works, we video recorded animals continuously through several days, both during the dark and light phases, and we calculated how much these animals were scratching and when. So as you can see here, animals were more active during the dark and during the light phases, both for controls and ablated animals, as is expected from mice that are nocturnal animals. But we also observed that both controls, but also the PTF1A ablated animals, scratched more during the dark phase. That gave us an idea that probably the scratching phenotype was correlated in some manner with the level of activity of the animal. The more they would move, the more they would scratch. In order to test this, we just did a simple experiment that was to restrict the ability of the animals to move. So we just put them in this plastic enclosure that we have used before for the air puffs delivery. And as you can see here, the animals can move, can change size, they can groom, they can do stuff, but they cannot walk. And as you can see, the same animals before and after putting them in this enclosure, the scratching goes down almost to control levels, giving us the idea that probably it's actually the mechanical stimulus that they encounter in their home cage what is triggering this, this scratching phenotype. To test this, we just did a very simple experiment that was to reduce the amount of mechanosensory cues that they find in their own environment, that it's the laboratory home cage. So we just remove all the bedding material from the cages and we quantify the scratching before and after. And as you can see, we found around a 40% reduction in the amount of scratching that the PTF1A ablated animals experience when you reduce this, what we can call mechanosensory landscape to which they are exposed. To confirm this idea, we explore the possibility that this kind of stimulation would lead to more activation in PTF1A ablated spinal cords than in control. And for that, putting the animals under anesthesia, I stimulated just the hairs on the back of the animal, trying to avoid touching the skin. And we kept the animals under anesthesia to prevent any other kind of stimuli to affect these animals. And then we performed CFOS analysis to have an idea of the activation of neurons in the spinal cord. As you will see in these images, but probably better in the higher magnification ones, the dorsal horn of the PTF1A ablated animals were enriched in CFOS inactive neurons, while the ventral horn that does not receive this type of light mechanosensory stimuli was unchanged. So in order to test gain of function approach, we reasoned that increasing the activity of an inhibitory population in otherwise part of the spinal cord that it's normally under very strong inhibition would not lead to a clear phenotype. So we decided to use the GRPR population of spinal neurons that since many years has been recognized as the population of neurons that are both necessary and sufficient to signal itch in the spinal cord. So we used GRPR-Cre animals combined with an intersectional genetics chemogenetic activator. So the cells that are both FLIP and Cre positives will express the channel HM3D that are activated upon delivery of CNO. And we would measure the scratching in these animals. So in control animals, of course, there was no change between vehicle and CNO activation. The same was true for when we activated just PTF1A cells. When we activated GRPR-Cre cells alone, we saw a high increase in scratching as was expected. And when we activated at the same time in the same animal PTF1A and GRPR cells, what we saw was a clear reduction in the scratching phenotype, confirming what our hypothesis, that is that PTF1A Cre cells are in place contacting presynaptically the mechanosensory inputs that come into the GRPR itch circuit in the spinal cord. So upon ablation of these cells, the simple movement of the animal around during their normal life will lead to activation of GRPR cells that will lead in turn to scratching. With this, I just want to thank the Rudiger Klein's lab in the Max Planck Institute of Neurobiology and all the people that contributed to mouse lines, and of course the funding and you for your attention. Thank you.
Thank you. Also, very nice talk. So again, place your questions in the Q&A. I sort of had a question about the phenotype. It seemed very interesting to me when I'm thinking about sort of this cancellation of self-initiated sensory inputs in other domains. Why is the animal interpreting this as itch, right? As opposed to another sort of somatosensory sensation that's not, itch.
Yeah. Well, the short answer is we don't know, obviously. The long answer is GRPR neurons receive, we have quantified that in the paper, receive abundant input from mechanosensory mechanical sensory afferents. So if those afferents are disinhibited, that is going to lead to activation of the neuron, and then of course to what GRPR neuron signals is itch. So that's the behavioral consequence. Then I need to stress out, we are removing only like 40% of the PTF1A cells, and this is the phenotype we see. So the other 60% of PTF1A neurons probably are targeting other types of fibers, right, of sensory fibers. So I would not be surprised if in that case we would see a different type of behavior as the output of losing these neurons.
So how dependent do you think this is on the mouse generating the movement? So if you force the movement on the mouse itself or impose the movement, you think you would see the same phenotype?
Yeah, I was trying to perform that kind of experiments, like putting the animal on a treadmill and that kind of stuff. I never managed to make it work because, of course, the animal are anxious for other reasons, the apparatus, everything. So it was pretty tough. So the best that we could come up with was just to record the animals in their natural environment, that is their home cage, and just see how they behave there. And then we see this correlation between movement of the animal, as you see, natural, spontaneous, and scratching. If I could find a way, well, there would be a way like stimulating optogenetically some regions in the brainstem that are correlated, that have been demonstrated to induce walking, then we could perhaps answer your question better. Yeah.
Any other questions from anyone? One last one for me then.
Sure.
What would happen if you shaved the mouse, removed its hair?
So I did that experiment actually, not shaving completely the animal because that's tough. So I was doing an experiment in which I would shave 1/2 of the animal, of the back of the animal. And surprisingly, what happens there is that the animals scratch exactly at the interface between the region that doesn't have hairs and the ones where the hairs are starting. So that region would develop quickly wounds from scratching of the animal, just in wild-type animals. So apparently having part of the skin in which you are having this interface between no hair input and hair input, it's enough already to trigger this scratching response. Probably the animals are detecting their own hairs now contacting more the now naked part of the skin. But we stopped the experiments when wild-type animals behaved exactly the same as already developed this phenotype. I mean.
Thank you. Very, very interesting finding. I want to thank again all our speakers for the nice session today. Hope everyone enjoyed it, and please enjoy the rest of NeuroMatch. Thank you, everyone.
Thank you.
Thanks.
Thank you.
The other room, if the live stream on the other room is finished, yeah, we will go live immediately. But we will get queued if there is another webinar running at the same time.
So each room has their own Zoom, right?
Right, yes. Oh, actually, you were previously in the same room, I think. I just saw you in the link. Let me just paste the link as well. Okay. So I guess I should go off screen now and wish you best of luck.
You can stay with us.
Really? I mean, it's not protocol, you know.
Yeah,
maybe I can join if nobody else shows up.
When the other speakers don't show up, then protocol go out the window.
Yeah, I know. Okay, so let's start.
Did they have
now.
a social last night? When was the social? Is it tonight or was it last night? They were watching some movie for one of the socials.
Oh, it was yesterday.
It was yesterday?
Yeah, I guess. All right, so
wait, before you go, before you go, you have a presentation tomorrow, right?
Right.
Are you going to wear a costume?
Really? Am I supposed to?
Yes. I think Conrad sent an email saying that there was going to be like a costume contest.
Oh, okay. I'll figure out what is it. Okay, I'll prepare. Thanks for letting me know. Krishna, can you hear us?
Yeah. Hello, Harry. Hello.
Hi. How's it going?
Hi. Hi, everyone.
Okay, great. So we should start now because time's up. So hopefully everything's okay and your screen share is okay. And I'll try to add the others, the other speaker from the attendees list if they joined late. So I'm off screen in the chat. Good luck and have fun. See you.
Thank you. You want to share your screen, Krishna?
Yeah, sure. Just give me 1 second.
So getting that, I'll welcome everyone joining us for another great afternoon for us here. Maybe good morning or good evening to the rest of you. Day 4 of NeuroMatch. Have a nice series of talks on some theoretical computational neuroscience. First up, we have Krishna Katya. You are in Devi University?
Pardon? I'm from Srimata Vaishno Devi University.
Yes.
I'm part of DevoWorm Open Science Group.
Right, and he'll be talking to us about the differences between biological and artificial neural networks.
Yes.
All right, so you can go ahead and get started. You can put your presentation in presentation mode.
Is my screen visible?
You can see your presentation, but it's still not in presentation mode.
Is it? I think it's now. Is it being visible?
It might be slow. Can you advance the slides?
Yeah.
Yeah, I think it might be stuck. So try unsharing and sharing again.
I'm changing the slides. Can you see the changes?
No. So if you can stop sharing and then try again.
Yeah, I apologize for.
That's all right. I mean, it's yeah.
Yeah. Now, can you see the changes?
Yep. You can go ahead and get started.
Okay. So first of all, greetings everyone. I'm Krishna Katyal and I'm going to present a contrast between biological and artificial neural networks. And this presentation is co-presented by Bradley Ellis, who helped me and acted as my mentor. So first of all, that I would like to talk about what are ANNs. ANN are artificial neural networks. They are the mere imitation of the biological neural networks that we have. So ANNs are they are unlike biological neural networks in many ways. So and they are alike biological neural networks in many ways because they both help in computation. They both can be help in learning. They provide animals and machine the ability to learn. So how much complexity do we need to capture the computation power of the nervous system? First of all, I would like to so for biological neural networks, we have different kind of neurons that help us in different kind of muscles. We have different kinds of neurons for muscles and for sensory organs. And same do we have different types of neural networks. For example, if we want to compute something that has a relationship with its previous values, then we have LSTMs. If we want to do a computation with the help of images, then we have convolutional neural networks, so on. So with different problems, we have different type of neural network, and with different things, we have different type of biological neural networks. So here is a diagram of a biological neural network. Here we have dendrites, axon, we have synapses. The dendrites act as the input, the axon is the main cell body, and then we have synapse that act as the output. So there is C. elegans. C. elegans is an organism that has much of its counterparts to humans. So research of C. elegans helps in You can say it can be in a broad way or layman language, it can be cited as a mere prototype of human body. It's not, it's far less complex than human body, but a lot of things of C. elegans and humans have a great resemblance. For example, here we can see that its ion channels, they are interconnected much like the human brain. So artificial neural network in the year 1943, when Mckesson did their experimentation and found that the neuron impulses are somewhat chemical and electric in nature, this gave rise to the ANN. Then we have Madeline and Adeline, one of the very basics artificial neural networks that were formed in early 50s. So if we, so what is basically an ANN? It is in somewhat representation of a BNN that is done by a computer. So much like BNN, we can do a lot of things that many of us have used Siri, that is developed by Apple, or we have used a lot of things like object detection and object recognition, natural language processing. All them are achieved by the means of ANNs. So here is an example of an ANN. So how it is not similar to our neocortex, because you can see that it's in 4-layer network. It doesn't make a mesh-like structure. The topology of a biological neural network is very different than an ANN. Here we have just a simultaneous layer of networks, but in case of BNN, we have a huge interconnection here. Every node in a BNN is someone connected to the other node by a huge mesh. You can say a huge crumbled mesh of connection. But in case of ANNs, you can see that it's far more simple. And the difference in ANN and BNN also occurs in many ways, like fault tolerance. Since the BNNs are interconnected with one another, it is decentralized. It's decentralized, so it has better fault tolerance capacity. For example, even if 1 or 2 layers are not working well, we still have other connections, which is unlike in case of ANN. For example, in this picture, if this layer is not working well, my whole of the network become redundant. But if we take a ANN here, if some nodes in the center pass, I guess my cursor is visible. Hello. Am I being audible?
Yeah.
Oh, okay. So if in the central part of this huge mesh-like structure, if 1 or 2 nodes are not acting well, it doesn't make much of a difference. Hence, with the help of its topology and asynchronous triggering of nodes, we have better fault tolerance. Now, if we go, okay, so power consumption and topologies, as earlier mentioned by me, due to the due to its due to the asynchronous and synchronous difference in triggering, they have a lot of different functions. So even though the ANNs are go much in a, you can say, straightforward simultaneous mode, they sometimes can have loops that are known as recurrent feedback. As mentioned by me in my first slide, we do have some nodes that we have that will take the value of its previous input and will make a loop-like structure. But that's not even the 1% of the complexity that occurs between the BNN. Okay. So power consumption, and human brain is round about 20 watt of power. It you can say it consumes 20 watt of power. It is different between genders and between the age of people, but the somewhat value ranges between 20 to 25. But in case of a GPU, that's a graphical processing unit that we used for training neural networks, take around about 150 and 250 watt of power. So why is the human brain more energy efficient? It's because of its topology. We, for example, if you want transmission between a node A and B, then we choose the you can say the route, or you can choose the communication pattern that has the least power demand. But that's not the case in the ANN. That's why it gets more and more power consumption. So, like earlier mentioned by me, if we want to go from here module this one to this one, we will not take the longest route. We will take the most you can say most energy efficient route. So thank you. This was all. So I would like to sum my my talk with that the main difference between ANN, BNN are in their topologies. One is having a huge mesh-like structure, and the other is quite simple with feed forward and a backward propagation, and the asynchronous and synchronous triggering of the neurons. And the third one is the power consumption, and the fourth one is the ability to tolerate fault. That's all. Thank you, everyone.
Thank you. So please feel free to put your questions in the Q&A. So I'll start with a rather naive question, given that I'm not a computational neuroscientist. So for the biological neural networks, does everyone start with cortical-like networks? I mean, there's different types of biological networks. Do we think that this is the most flexible one?
Can you please repeat the question?
So when, the comparison is usually made to cortical networks, right? But there are different types of biological neural networks, right? You can think of the spinal cord as one that has a very different topology than the cortex. Does the field usually just work on sort of cortical-like networks because we think these are the ones that can facilitate the most flexibility or learning?
Yeah, the comparison here is between the cortical-like networks and regular ANNs. With BNNs also have a lot of different type, as mentioned by you, that spinal network, spinal cord has different kind of network. Even the ANNs are also not same. Here my comparison was between a simple feed-forward ANN and the neurocortex network. We have different kind of ANNs, and so we have different counterpart of different kind of BNNs also. Like for example, if we are going into vision, we will have CNNs that are convolutional neural networks that are somewhat different to our basic neural networks. But yeah, here the comparison was between what we get in our brain and between a simple feed-forward artificial neural network.
Any other questions from anyone? Our speakers are free to ask questions as well as our attendees. So I have another question then. So in comparing the differences between the biological and artificial neural networks, looking towards the future, do we want the artificial ones to be more biological, or do we thinking biological networks, we want them to be a bit more artificial-like?
Yeah, that's a really nice question, and it can be made into script of a sci-fi movie. So I would like to say that the artificial one would be get more into the biological one, because the brain, the neocortex that we are having, it's having, you can say, infinite potential. Humans can be artists, they can be engineers, they can be poets, they can be anything, and all that is because of the what part of brain and how we are using that brain. So yeah, as we can see from last 50 to 60 years, the ANNs are getting more in like the BNNs. Like for example, when it all started, the ANNs were just used to compute the OR and AND function. For example, if you are computing the OR function, you know that when we have two on values to one values, the output is 1, and if we have 1 0, then it will also be 1. But if we have 2 zeros, then it would be 0. So when the ANN started, they were doing it pretty well. But then the question is XOR, if a selective OR came, and then we had to you can say improvise it and add a further layer for an complex decision. For example, now when we are moving up with further when we are developing further computation methods, then we are having the ability to make it more like a mesh-like structure. For example, the human brain is said to have 10 to the power of 7 neurons, whereas the maximum that we are having in an artificial neural is 10 to the power of 3 or 4. That's a lot of difference. So yeah, we are going in the processes of making ANNs much like the BNN, but we are still a lot of, you can say, years behind that. But I guess one day it will come that when we will able to replicate the brain, not wholly, but yeah, in a partial manner. We are doing it in a partial manner, but we will able to do that asynchronous and synchronous triggering of ANNs, much like the BNNs that we have. And we'll be able to make it much more fault tolerant and much more energy efficient. And you can say brain can do a diversity of tasks. A person who is an artist can also be a logical person, can be a mathematician. But the ANN that we are developing are not much versatile. They are, for example, if we have CNN, they can only do the things that are required for the images. And if you have, for example, an attention layer for NLP tasks, it's only good for understanding the language part. So yeah, I think in coming time, maybe in 1 or 2 decades, or even less, we will able to have versatile kind of neural networks, because neural networks also have more than 15, 16 varieties. We have perceptron, we have Adeline, Madeline, convolutional neural networks, feed-forward neural networks. We have our residual block networks. So yeah, one day I think there would be a mixture of all of these networks that would be a lot more similar to the brain.
Thank you again.
Yeah,
nice thorough answer. So we'll go ahead and move on to our next presentation from Eric Wong at UCSD. He'll be presenting work on neural attractors and cognition. So Eric, take it away.
Thanks. So I'll be presenting a simple high-level model for the core computational architecture for learning and cognition in the human brain. There you go. Okay, so the model is guided by some behavioral considerations and also some biological considerations. So to start off, we note that human intelligence, at least modern human intelligence, is mostly learned. Collective human intelligence is driven by the accumulation through language, symbols, and diagrams of knowledge from all of our human peers. We now spend the first several decades of our life learning a distillation of this accumulated intelligence. And we also note that the learned associations from all these lessons remain explicit, that is reportable, explainable, and teachable. And we would suggest that these features suggest like an associative network as an underlying architecture for most of this type of intelligence. Associative in the sense that we directly connect some, object A to object B. But we also note that parts of human intelligence are implicit, so that is not explainable or teachable. So examples of explicit and implicit might be, explicit knowledge would be whales and dolphins or cetaceans. The Pythagorean theorem would be explicit. Implicit is things like, is Fred trustworthy? Which of these two chess positions is stronger? Implicit thought usually has explicit components in it, but the hallmark is that they're incompletely explainable. And we suggest that the presence of that type of thought suggests that there are both associative and also deep components to the underlying network. Deep in this case meaning specifically that there are elements or layers in the processing that are hidden to the owner of the brain. We also note that anything can be input or output to the network for our brains. So the example, like what animals have feathers and what are birds covered with, suggest two ways of asking the relationship between birds and feathers that are opposite in direction. So they reverse input and output. But more generally, you can think of virtually anything you could think of can be either the input or the output of a particular question, either the question or the answer. In contrast, lower animals are more or less one-directional processing systems that map sensory input to actions. And also in contrast, in our own brains and most brains, examples such as the visual cortex actually are hierarchical and are directional. Even though they're recurrent, they're directional in their processing. In this case, processing input from photoreceptors to objects, and that suggests that the underlying structure is relatively unstructured, unlike a hierarchical network. Okay, so some biological guidance. We'll note that Hebbian, sort of synapse-specific plasticity, and neuromodulation, which is sort of less synapse-specific, are sort of the two classes of learning mechanisms that are well documented. We'll also note that this is less universally accepted, but something like attractors or cell assemblies or engrams, various other labels, represent things. So some set of cells in the brain represent objects. We also note that empirically, there is associative learning mechanism can connect attractors. So it's an example is classical conditioning, so we've shown empirically that just juxtaposing two things in time causes them to get glued together. Okay, and I'll go into this biological part in a little bit more detail. First, noting that the spike timing dependent plasticity, which is the main version of Hebbian learning, directly converts evidence into predictions. So if we take the example of some primitive organism that has a sensor, this is a neuron that gets input from some heat sensor, and then decides that you should move in that response. If that same organism has a light sensor and a neuron that receives input from that, if there's a synapse between the two that is subject to spike timing dependent plasticity, what that synapse does is it's on the lookout for asymmetries in the timing between the input to the light and the input to the heat sensors. So if light consistently precedes heat, then that synapse will convert that piece of evidence into a prediction by wiring this synapse and causing the light neuron to excite the heat neuron, and therefore predict that heat will come before the heat actually does come, and allow the animal to move ahead of time, making that a prediction. Let's see. Okay, and we've recently noted that this same principle of basic STDP converting observations into converting evidence into predictions scales up to the attractor level. So this is in a paper that we just uploaded to bioRxiv just last week. It's listed at the bottom of the screen. And we note that STDP can both create attractors in an initially random network, and it can associate attractors using the same, effectively the same sort of mechanism as this neuron to neuron association works attractor to attractor as well. So don't have time to go into that in detail, but so we would suggest that STDP and STDP-based mechanisms are actually sufficient to support all of these features. Okay, so to actually put together a proposed architecture based on these observations, we start with a random collection of neurons, randomly connected, recurrent, and into that we claim that these mechanisms can embed attractors from the existing pool of neurons. So on the left, they're shown as collections of neurons in green, but just schematically, for simplicity, we show them as attractors as pentagons on the right. So the left and right are the exact same thing, just depicted differently. And we note that attractors can be associated with each other, and that also these background neurons can be attached to either attractors or to each other. Okay, so then collecting that into a model for both explicit and implicit thought, starting with explicit thought. So if you have if Sally happens to know that that whales and dolphins are both cetaceans, then this set of attractors would be in Sally's brain with these associations. If Sally wants to convey that information to Bob, then she might use language to convert this particular attractor structure, linearize it into a set of words, and deliver that set of words to Bob. Bob would then parse that set of words And the understanding of language would cause Bob to say, oh, whales are cetaceans, dolphins are cetaceans, cetaceans include whales, cetaceans include dolphins, and just juxtaposing those, the excitation of those attractors in time causes those to be glued together by STDP. And then, and effectively Xeroxes this association from Sally to Bob. And so we suggest that in your first 20 or 30 years of life, you might repeat a similar process maybe 1 million times and pepper your brain with hundreds of thousands of these associations. And that actually just the simple operation of those associations is probably the bulk of what we actually do with our brains. But there are implicit functions as well. And we'll remember that this STDP mechanism is active at any synapse that follows that type. And again, it's looking for regularities, so for statistical regularities in the spiking patterns between pre and post-synaptic cells. And anytime it finds statistical regularities, will convert that into increased synaptic strength and convert that therefore into prediction. And that happens all over the network all the time. So if you look at how that might support explicit thought. So if you ask Bob, is Fred trustworthy? There may be some explicit components to the answer to that. So, so Bob might not know if Fred is directly trustworthy, but know that Fred knows Linda. Linda is trustworthy, so that might provide some evidence. That's explicit. But in addition, Fred might explicitly know Bob might explicitly know that Fred is tall. That also doesn't connect directly to trustworthiness, but in Bob's experience of all the people he's run into that are either trustworthy or not trustworthy, those interactions may build up an implicit bias for either for or against tall people. In addition, this synapse right here may have noticed that every time Bob runs into Fred, there's a car alarm going off in the background. And with or without Bob actually consciously realizing that fact. And that connection would feed into the network and may influence the level of excitation of the trustworthiness attractor as well. So by that mechanism, this whole web of associations that's learned by STDP-driven mechanisms might support implicit thought and learning as well. So this is just a very much simple core theory for human cognition. It's obviously surrounded by many specialized systems that are not included in this diagram, but we'll just summarize by saying that our hypothesis is that the core computational architecture of the human brain is a hybrid of an associative network driven primarily by human teaching, in addition to observations from the environment, supporting explicit thought, and that is embedded in a Hebbian network that's driven by STDP-based conversion of evidence anywhere in the network into predictions, and that supports implicit thought. And I thank you for your attention.
Thank you, Eric. Nice talk. Any questions you have, just enter them into the Q&A. So I'll start with a question. So for this sort of model, how can you incorporate sort of innate biases and association strength between different types of stimuli?
Right. So there's, obviously in lower animals, there's good demonstration that genes have a remarkable ability to actually pre-wire a bunch of stuff. So that's certainly not included. This model is more, the learning part after birth. But and I don't know how the genetic part is actually implemented, but clearly there are both very sophisticated mechanisms for imprinting of innate behaviors, and also for very early learning of some things. For example, like intuitive learning of of words and grammatical structures and stuff like that that need to support this kind of model.
So it would also work with sort of a semi-structured model that's not completely random to begin with.
Yeah, I mean, right. Obviously, this is very, very much simplified. There's, like lots of cell types. There's a bunch of structure that's in the cortex. There's layers and there's a lot of structure that is there. So what we're trying to ask is it's somewhat surprising that there's not really a well-established, really core computational model. And so this is obviously, grossly simplified. But it's asking whether, the pieces that are well understood, like STDP, can be cobbled into something that that can learn a broad range of, human-like learning activities.
So we have a couple questions in the Q&A. First one from Kirubel, great talk. How scalable is this model to more complex decision-making tasks? What other mechanisms could be added for greater performance?
Well, actually, to me, the most interesting question is what happens when you make this network big enough that you can actually put your million associations into it. So it's not actually obvious to me that this mechanism by itself is not sufficient.
Next question from LR. How does the separation between closely related concepts emerge? Do you need mutually exclusive subset of neurons to implement two attractors?
That's a great question. And bottom line is I don't know the answer to that. That was kind of the topic of a really nice general adversarial collaboration that was sponsored by this same organization that was talked about last week. Do you learn new things by activating old attractors, modifying them, and then repackaging them, or do old attractors stay separate and you just modify associations between them? I think that's a unresolved question. I think this architecture supports sort of either version of that, because it's like, I've drawn these pictures with associated with attractors separate, but the attractors could just as well be overlapping as well. But yeah, it's a good question, but I think it's unresolved.
All right, thank you again, Eric. Nice talk. We'll move on to our next presentation from Bradley Alicea, who is at the Orthogonal Research and Education Lab as a head scientist. And he's also a senior contributor to the Open Worm Foundation. You can find his Google Scholar in the chat box. And he'll be discussing how you can incorporate the beauty of development into some artificial neural networks.
Yes, so thank you. Jesse Parent, who's a co-author on this, is also in the chat. So if you want to ask him questions after, he might be able to answer some. Thank you to our co-authors listed here. And this is approaching artificial intelligence as an embodied developmental process. So these three GIFs here show different organisms in the process of development. Here we have some neural crest migration. Here we have a developing nervous system in the axolotl, and there we can see elongation of the zebrafish notochord. So we notice that all these things are very much morphological or involve the body in their development. And so we're going to take a little bit different approach than what we would have with biological organisms. We're going to use something called Breitenberg vehicles. So if you're familiar with this, this is a toy model that was developed in the 1980s by Valentino Breitenberg. Their first order stimulus response mapping. So you have this light source up at the top. The sensors are in the front of the robot. They get transduced to the wheels, and it drives it's called a vehicle, but it's really implemented usually in robots, and it drives it in different directions, and they exhibit all sorts of behaviors. So they yield these sort of tactic or reflexive behaviors. And so it's based on a very simple neural network. But we've taken this a step further in our group, and we've developed something called developmental Breitenberg vehicles. So these are embodied nervous systems in these vehicles, and the neural network develops from that simple sensor effector mapping to something more complex in the organism. So this is what we call network morphogenesis. So we're building a network from nothing. And the initial conditioning is that simple mapping, but then you get a complex network over time that then behaves in various ways. So this is a schematic of how this works. So we've used genetic algorithms to develop this network morphogenesis technique where we have layers of cells, which are like neurons. These serve as interneurons. So the sensor is up here, the effector is down here, and these circles are interneurons that are layered, and then they connect gradually over time. They connect, in different combinations of pathways. And we can add other types of models, such as a CGS, which is something that we use in our group as well. This is a more mature version of this developmental Bratenberg vehicle with a denser set of connections and maybe a couple more internal nodes or interneurons. And so we can do different types of behaviors with these. One example I use here, used it in a number of talks, is called chromotaxis. So the idea is these vehicles will be attracted to not only a certain color, but can differentiate between colors. And so as a phototaxis, this operation is simple attraction or repulsion. Are you attracted to this color or are you repulsed by it? As a preference, you have this separate continuum of emotional valence. So now it becomes important to differentiate between different shades of a color or different colors. And then there's a difference between congruent stimuli. So color, for example, and then maybe the shape of the color might become important. And we've simulated that using generalized Hebbian learning, which occurs later in development. And then we can also have differentiation between incongruous colors, and this enables spatial learning. And this is something we've done with a genetic algorithm, not only evolving these networks, but like differentiating between things in the environment as well. And actually, I'll send you this paper. This is a good source for this type of work. So the heart of this method is something we call connectivity activation encoding. And this is a simple notation for a matrix. And this is what it looks like. You have your cells and you have connections, and that's your matrix. Each developmental time point produces a connectivity matrix. But of course, as the size of the network increase, or the number of interneurons increases, the size of that network also increases. But it's basically over developmental time, you keep adding neurons and adding connections, and that's what you end up with. And then you have, from that matrix, you can get a corresponding determinant A. So then we have this relationship between the matrix growing over time and these different A's that are available to over developmental time. So in development, the number of neurons can grow and synapses can fluctuate in terms of their strength and efficacy. We think of this in terms of universal functions rather than as an actual transformation of WIJ. It's this idea that they're like these fundamental rules that govern these matrices no matter what their size. And so that's borne out a little bit in the deep learning literature. But this is very different from sort of like a reductionist representation. In that case, you have all sorts of different things going on in the brain. What we need to do is to make this WIJ sparse, but we also want to make it as biological as possible. We want to add biological sort of relevance to it. And so you can see how this kind of works in a C. elegans model. These dots, white dots, are the number of neurons that grow or that appear or born in the C. elegans over developmental time. I've mapped them onto an adult to show that you start with a single neuron in what will become the head, then you get a couple neurons in what will become the head, and then finally you get many neurons in the head, and then some starting to be born in what will become the tail. And that's how we envision our vehicles being very similar to that. But we also add something called critical periods, and critical periods are a window of developmental time that features an enhanced ability to learn. So critical periods exist in all sorts of organisms, and we usually associate it with like early visual learning or other types of learning and physiological reorganization, which is actually one of the hallmarks of critical periods. And so we are going in this direction where we're taking these vehicles, they grow over developmental time, but we're introducing these critical periods where the critical period can expand or contract, and that's when a majority of your growth and reorganization occurs. And so this is mapped out in the following way. You have morphogenesis, which is defined by slow growth in this network. So you add nodes and you add connections, but it's relatively slow. Then later on, you get this critical period, which is in the dark gray. And you can see the difference between this morphogenesis and the critical period, which is technically part of the morphogenesis, is where you get your number of nodes will grow at a higher rate, and you'll get a lot of reorganization amongst the connections between them. Then finally, you get developmental learning, where you stop adding nodes altogether, and then you actually modify the weights of the connections. You don't lose and gain connections so much, you just modify the weights. So in that kind of stable network, you can start to learn things. And so this is developmental, what we call developmental freedom, which is where you've fixed the network in terms of its morphology, but now you're learning on it and you're changing the weights with respect to like different sorts of stimuli. And so we can think of this as terms of early and late effects. So we have this contingency matrix here that we've made where we look at the critical period and then we look at developmental freedom. So an early critical period means that the adult neural network tends to be more homogeneous. There's less time for preliminary or auxiliary nervous systems or behavioral traits to develop. And there are fewer varieties of experience prior to maturity. Contrast this with a later critical period, which is what we see in the adult neural network in that case, is that it can exhibit a larger number of possible configurations across the population of individuals. Slower developmental periods afford more time for exploration and wandering. Then if we look at developmental freedom, we see that there's a low degree of overall developmental freedom when you have an early critical period, and then of course correspondingly early developmental freedom. And this early critical period allows for a faster ascendance to a fully mature suite of behaviors and capabilities. With late developmental freedom, which corresponds to the late critical period, you have a high degree of overall developmental freedom, and this allows for a slower ascendance to adult behaviors and capabilities. And so I'm going to also introduce, before I wrap up, this idea of Gibsonian information. This is the way we view the way we view information coming into this embodied nervous system. And so the key to Gibsonian information is that motion provides information. So what that means is it could relate to optical flow or relative motion between the object and observer. And I'll give a couple examples. So this is basic visual flow with motion and contrast cues. You can see this roller coaster. Imagine the agent is on a roller coaster and it's experiencing all these motion cues. But you can also do things like look at a disjoint distribution, and that is between a moving stimulus and a stationary stimulus. And in this case, you have this white noise embedding where you have just a stable white, static white noise background versus a dynamic white noise background. And you can see their motion cues in the ladder up at the top. Then you also have time series structure. So you have things that happen, events that happen in sequence. And so when they happen out of sequence, or you cut things that are expected out of the sequence, you get different types of visual scenes and different types of responses in the nervous system. And then finally, maximum versus minimum coherent movement. So this one's a little tricky, but this up at the top is the random movement of these balls. And so this is minimum coherent movement because while there's a lot of movement, it's not coherent. Contrast this with at the bottom where you have a lot of maximum amount of coherent movement. You have these flocks of birds that are moving in a very patterned way, and you can see exactly how they're kind of coming together and clustering. And so these are all the things that we want to be able to train our agents on and get them done, get them to differentiate between these different types of information, and then ultimately make that quantitative. We're also working towards reinforcement learning approaches, which are where we can develop discrete layers that will help the agents learn better policies. The growth of these nervous systems have to be countered by a potentially large and continuous action space. And we're thinking in terms of more specifically curriculum learning, where you give easy examples and then you give harder examples over time. And then you end up with, an ability to learn more and more over time. Finally, there's this issue of contingency in development. So going back to the scenarios for developmental freedom in the critical period, we have to remember that development unfolds in a certain contingent way. So each node is a developmental event. And so when you put a node in a certain place, you constrain the development of future phenotypes and how they can learn. And so you have these pathways that are contingent upon previous steps. And so when we build a nervous system, we have to consider that contingency is important. So morphogenetic events actually restrict the network. The more morphogenetic events you have, the longer your morphogenesis period goes, and the later your developmental freedom is, then you have this more contingent and more restricted network. The more changes will occur during the critical period, but it results in a network restricted to an increasingly smaller possible outcomes over time. And then finally, developmental freedom may allow for a limited convergent evolution of development. And this may have, there might be ways to investigate brain versus mind using that approach. Finally, thank you for your attention. Our work is sponsored by the Representational Brains and Phenotypes group, and we do this Saturday morning NeuroSim group every Saturday morning. We're open on the web here. We have a GitHub repo that if you want to contribute to or look over our work, we have that. We also presented something called the Psychophysics of Non-Neuronal Cognition earlier today, which might also be of interest if this talk was interesting to you. And you can read our preprint on this work on ResearchGate. Here's the QR code and the link. Thank you.
Thank you, really nice talk. So we just have a couple minutes for questions. We have one here from Drobomir. These were really fun examples of Gibsonian information. Could you please say how you would investigate specific forms of information in your robots?
I think presenting, like when I gave the example of chromotaxis, for example, that's where we're taking things from the Bradenberg vehicle and making it a little bit more complex, having multiple parts in the stimulus and then testing it in that sort of world. With Gibsonian information, we probably look at some of the things like, some of the examples that we used in there, like taking a sequence that it might learn like a sequence of events, and then chopping up the events, mixing them around, or taking key events out of the sequence, and then trying to get it to learn sort of how to infer things in between those in between those events and get a coherent sequence out of it. Or we could introduce, like, with coherent movement or coherent motion, get it to observe things like flocks and swarms, and get it to differentiate between that and something like Brownian motion, where you're introducing this sort of different degrees of like organization and collective motion, and seeing if it can distinguish between like some background where it's just totally random and some background where it's very coherent and structured. And so those are things that, those are more training environments, but then you can also measure the, when the agent is getting the information, you can measure how it's processing the information and things like that. So it's kind of how we're thinking about it. But
so just one last quick question. We'll be kicked out in a minute or so. What do you see sort of the application of these embodied neural nets versus a disembodied neural net in terms of what they can accomplish?
I think the ability is to like to get basic spatial information and other sorts of environmental context. So there are all sorts of cues when you put something in a body and you have it moving in different directions and sensing from different directions, and things are dependent not only on like inputs and outputs, but space and spatial analysis. That becomes important. And you could use that for a variety of things that are very spatially dependent, like, anything from like self-driving cars to like something like a robot that's trying to find its way through the environment. So there are a lot of potential applications for it.
Great, thank you. Thanks, everybody. It was a great session. I hope everyone enjoys the rest of NeuroMatch.
Thank you.
Thanks.
Oder starten wir dann
Ich glaube,
einfach?
du fängst einfach on time an.
Okay.
Da ist gerade noch was im Chat gekommen. Ah ja, YouTube is set up, so I guess we can start.
Okay, then hello everyone and welcome to the keynote lecture of Professor Philipp Kanske. I will host the session.
Wait. Sorry, I was too fast. Okay. Do you see the chat?
Yeah. Click the start webinar button.
All right. I think now we are ready to start.
Okay. Then again. Okay. Hello, everyone. Welcome to the keynote lecture of Professor Philipp Kanske today. I will host the session, and before introducing Professor Kanske and leaving the floor to him, I wanted to say that we will have time for about 10 minutes discussion after the talk. And if anyone has a question but does not want to speak up or is technically inhibited, you have the possibility to write your questions in the FAQs and I can read them out later. I'm very happy to introduce Professor Kanske, who is currently Professor for Clinical Psychology and Behavioral Neuroscience at the University Dresden in Germany. And he is an expert for emotional and cognitive processes that support human social behavior. His research targets many very exciting topics such as emotion regulation, metacognition, empathy, mentalizing, and social decision-making, of which he aims to understand the underlying neural correlates. Upon finishing his habilitation in 2013 at Heidelberg University, he was already announced by the Association for Psychological Science as one of the rising stars, and since then he received several national and international awards, such as the Hans-Meyer-Leibniz Award of the German Research Foundation or the Young Investigator Award of the European Brain and Behavior Society. And he has been member, and I think he's currently also the speaker, of the Young Academy at the German National Academy of Sciences Leopoldina and the Berlin-Brandenburg Academy of Sciences and Humanities. Today, Professor Kanske will talk about the social brain and in more detail about the interacting networks enabling the understanding of others. I think we're all excited to listen to your talk, and so without further words from my side, the Zoom floor is yours.
Thank you so much, Lisa. It's really great to be here. I'm very honored to be invited. It's my first time at NeuroMatch, but I'm a big fan already. So yeah, thanks a lot for inviting me here. The social brain. Why should you listen to me talk about the social brain? Why would you think about the social brain? I'm going to give you three very broad reasons to do so. The first one is that as humans, we are extremely dependent on others. From our birth until our deathbed, we really rely on social relationships with others. In terms of taking care of us, nurturing us, but also in terms of emotional and other support. The social interactions and social relations we engage in are also incredibly complex. In our everyday life, we kind of manage those social relations, even though they are so complex, relatively well, but it can also become difficult. For some people, it's more difficult than for others, and in some instances, it's more difficult than in other instances. And we know that people who have special difficulties in engaging in social relations, that this can be really harmful. Loneliness really is a killer. It's as harmful to your health as smoking. And we have some data that we collected during the lockdown in spring in Germany due to the coronavirus epidemic and pandemic, actually. And what we see there is that people who are especially vulnerable because of mental health problems in the past, these people, when confronted with, when having low social resources, develop especially strong mental problems, problems with their mental health. So the capabilities to build up close friendships, the capacity to engage in social interactions, even daily small social interactions, when you chat with your barista, when you get a coffee, for instance, these little exchanges and the close friendships we have, they really are important for our mental health, even for longevity, as has been shown. So what are the capacities that allow us to form these close social relationships and manage our complex social nets? What enables us to understand what's going on with other people? When you look at the literature, there's a broad range of processes that are discussed as being important, from affiliation, social attention, to agent identification, biological emotion, empathy, and theory of mind or action understanding. What I will mostly talk about today are empathy and theory of mind. Why? Because I think they are especially important, or they have a special capacity or a special characteristic that makes them very important. What is empathy and what is a theory of mind? They have been defined. Theory of mind is tracking others' intentions, desires, goals, but also emotions, and empathy is sharing others' emotions, yielding an isomorphic affective state in yourself. That means an emotional state that is similar to the other. If you tell me something that is very sad, that makes you sad, and I turn a little bit sad myself, that would be an isomorphic emotional state. So both theory of mind and empathy, what they give us is a representation of another's internal state. They really grant us access to what is going on inside another person's mind. And this is different from, I don't know, biological motion identification also. These are different important processes as well, of course, but what we also need to understand why people act in certain ways is to understand the driving forces behind their actions. And these are their intentions, goals, emotions, and so on, the inner covert mental states. Now, if you're interested in what the neural correlates of empathy and theory of mind are, you're lucky, because the research on these capacities has been going on for quite some time, for almost 2 decades. And there are now also really nice meta-analyses that you can look up. When you look at the meta-analyses on theory of mind, for instance, you see that these temporal regions are very much involved, lateral temporal regions, the temporoparietal junction, for instance, but also reaching up to the temporal poles, and these anterior and posterior midline regions. When you look at empathy meta-analyses, what you see is that the anterior insula seems to be very important, but also anterior cingulate cortex. Now, a problem with these meta-analyses that have been done so far is that these are really pooled meta-analyses across a wide variety of different tasks. And in a way, you can think of this as, because the tasks that have been used to study empathy and theory of mind are very, very diverse, what these pooled meta-analyses do is that they kind of throw apples and oranges into the same basket. And we don't really know what the specific processes are that might be specific to empathy and theory of mind. And also, we don't really know, when we look at these functions separately, how they work together, which they probably do in more complex social interactions. Another issue is that in the field, the terms are used quite diversely as well. You will not only find empathy and theory of mind as clear-cut as I have kind of pictured the simplistic picture now. You will also find terms like cognitive empathy, affective theory of mind, for instance. And they will also be used interchangeably. Sometimes empathy is used as an umbrella term for all kinds of processes. So what people do is they name their task when they publish something as an empathy task, for instance, or a theory of mind task. But it's not really clear whether the processes, and then they go into these meta-analyses, but it's not really clear what the processes are that are in these specific tasks. So Matthias Schutz and I and a couple of our colleagues, we set out to bring a little bit of order into this big picture. And what we did is we looked at the literature, and what we wanted to do is kind of sort the literature on empathy and theory of mind and include as many tasks as possible in an overarching meta-analysis. So let's start with the empathy tasks that we found. What we looked at here are really very different groups of tasks. For instance, tasks where people had to observe others in pain, observe others' emotions. They were really explicitly instructed to share others' emotions or others' pain, or to evaluate situated emotions, that is, emotions that are embedded in more complex situations, or reasoning about emotions, that is, situations where you really are supposed to deduce something about the emotion or some action based on the emotion of the other. And now, what you can do when you have these different task groups, you can do meta-analyses of the tasks that fall within each of these task groups. Instead of just averaging all of these empathy tasks together, you can look at the different task groups separately. And what this yields is a lot of brain images you basically get for each task group a meta-analysis. And now you can already see that there are some commonalities across all of these tasks. So they do seem to share something. For instance, activation in the anterior insula seems to be present across almost all of these tasks to some degree. But there are also differences. For instance, you can see that reasoning about emotions engages these temporoparietal regions quite strongly, while many of the other tasks don't really do that. You see some activation in these lateral, dorsal, and reaching into parietal regions here, but not so much in all of the other tasks. So there are some differences already here that you can visually see when you look just at the maps here. We did the same for theory of mind. Here we collected another range of different tasks: false belief tasks, trait judgments, strategic games where you engage with others in a dialectic or with other people in an exchange, social animations, rational actions, or reading the mind in the eyes that has been used quite extensively also. And again, you can do meta-analyses across all of these task groups. And again, you get a lot of brain maps that share some things. You see across most of these task groups, for instance, this anterior midline activation, precuneus activation, temporoparietal activation, but you also see quite a lot of differences between these tasks. You see anterior insular activation for reading the mind in the eyes, for instance, but not for the other task groups. And there are some other differences as well. So now we have all these different brain maps coming from these very diverse and different task groups that have been termed empathy and/or theory of mind. And what one can do now with all these maps is that you can use the data to actually find commonalities and differences between these task groups. You can kind of use the data, a data-driven approach, to group them together. And this is what Matthias did in a meta-analytic hierarchical clustering analysis. What it gives you is kind of a dendrogram like this one. On the axis down here, you see all of the different task groups that I've just presented to you. For instance, from the theory of mind group, the false belief task, or from the empathy group, the observing pain task. And you can see that they are based on the maps that I just showed you. They are grouped together very closely or further apart, depending on how similar the meta-analytic maps of these different task groups are. And maybe you can already, when you look at these, visually group them into bigger clusters. And this is also what statistics can give you. When you do so, one solution that statistics will tell you is a good one for clustering these is a 3-cluster solution. This would group together a couple of groups, tasks from the theory of mind group in a cognitive cluster, a couple of groups, tasks from the empathy group in the affective cluster. Interestingly, also one task from the theory of mind group, the reading the mind in the eyes. People who are familiar with that task might know that it really heavily depends on emotion. We are presented with just the eye region of faces, basically, and usually they express some type of emotion, and you're supposed to think about what that emotion might be. And this seems to be, in terms of what the activations look like, to be closely related to some other empathy tasks, rather than with the clear-cut theory of mind tasks here. And you can also see that there's an intermediate cluster here. In that intermediate cluster, you see situated emotions, reasoning about emotions, rational actions, and social animations. These are tasks from the theory of mind and from the empathy group that are a little bit more complex than the just observing pain in others, for instance. Interestingly, these tasks seem to be more related to one another than to the other empathy and theory of mind tasks. And what I'm going to do in the next for the next slide is explore a little bit what these different clusters are, what they look like, and also especially what this intermediate cluster might be doing. Is it a special type of social cognition, or is it something that is made up of the processes that are also engaged in the cognitive theory of mind and the affective empathy cluster? Before I do that, however, I want to tell you also that there's also an 8-cluster solution. This is basically a solution where you're almost on the task group level here. You have a lot of different clusters, and this just tells you that there's also a lot of variance in the data that is really specific to the different tasks, which is also interesting in itself, but a little bit a different story than what I have time to tell you today. All right, now when you have these different classes here, you can also look at the brain maps that you would get if you look at, for instance, a meta-analysis of the cognitive cluster. And this is what you see here. You have the brain map that would be associated with the cognitive cluster here on the left. You see anterior posterior midline regions and these temporal regions again. And you see the map for the affective cluster on the right, with the anterior insula, for instance, and anterior cingulate cortex. And what you also see is that the intermediate cluster seems to combine a little bit activations in regions that you also see for the cognitive and the affective cluster. You do have anterior insula on both sides here, which you get from the affective cluster, but you also have all these temporal regions that you would get from the cognitive cluster. So just from this view, one might generate the hypothesis that the intermediate cluster really is a combination of activation in the networks associated with empathy, the affective cluster, and this cognitive theory of mind cluster. Just for the sake of completeness, I'm also going to show you the 8-cluster solution, which just shows you that there seems to be really a kind of hierarchical organization in social cognition. And I'm also not going to go too much into detail right now. Now, people who are familiar with resting state data might already see that what is described as theory of mind-related activity is really closely resembling what people know as the default mode network. While the activation in the affective cluster resembles more activation in the salience or ventral attention network and maybe the frontoparietal network. And what we try to do is also to make use of resting state approaches to see where our clusters would be positioned and whether we can find commonalities and differences between the clusters. But before I move to that, we can also see here in the neuroscience decoding what functions or what terms in terms of neuroscience decoding the different clusters are related to. As you can see, the affective cluster is related to face, affective fear, pain. The cognitive cluster is related to theory of mind, mentalizing, and terms like that. And what you can critically also see is that the intermediate cluster is kind of a mixture of both. So as we already kind of generated the hypothesis from the maps, in terms of neuroscience decoding, you would also that would strengthen that hypothesis that the intermediate cluster isn't something entirely different, but it is a combination of what you see for the cognitive and affective cluster. Now we move to the resting state data. What we make use of here is data analysis from Daniel Margulies. What he did is he used the resting state data to come up with a principal gradient of a macro scale cortical organization. You get different principal components if you look at large scale organization of resting state data, and the main principal gradient would be this one. It ranges from sensory unimodal regions to abstract transmodal regions. And as you can see, these sensory unimodal regions are for instance, in the visual cortex, but also in the somatosensory and motor cortex. The abstract transmodal regions are in the anterior and posterior midline and also in the temporoparietal junction and the temporal lobe reaching to the temporal pole. So this is basically the default mode network. And we can now look at where, or that's what we did. We looked at our 3 clusters. Where would they be positioned on this gradient? And as you can see here, the cognitive cluster is mainly being positioned on the far right end of the cluster, on the abstract and transmodal end. The affective cluster has its main overlap with the middle part of the gradient, and the intermediate cluster kind of overlaps with both. It has particular overlap in the middle range, but also in the abstract transmodal regions. Again, arguing for this middle cluster being kind of a combination of processes that are distinct when we look at cognitive and affective functions so very separately. And this is actually a quite interesting finding, because from the resting state literature, what we know is that these regions that are positioned in the middle part and in the far abstract part here, they are more non or even anti-correlated. And for these more complex social tasks, it seems like they are nevertheless jointly co-activated. Now we took this one step further, and this is work by Lara Maliska and Matthias Schutz also. And we looked at when we try to see what regions are co-activated across many tasks with the different regions. What would we see there? Are there differences also between the different clusters? And I'm putting up the clusters here again. What we did here in order to do this meta-analytic co-activation mapping is to derive from the overall network. Basically, when you put all of the empathy and theory of mind tasks together and you create something that would be something like the social brain, when you use derive regions of interest from this, what you end up is the temporal-parietal junction, the insula, and also posterior cingulate cortex and anterior cingulate cortex. And for these two now, I'm going to show you what the co-activation maps would look like. For the insula first, as you can see here, here are the tasks in the cognitive cluster, and we can see what the tasks in the cognitive cluster, which regions co-activate with the insula in that cluster. And as you can see, these are regions in the anterior posterior midline regions and in the temporal pole, a little bit in the temporal parietal junction. For the affective cluster, it's a very different picture. You see the anterior cingulate, anterior insula, and these inferior frontal regions, anterior insula bilaterally. And now, interestingly, again, the intermediate cluster, as you can see here, the intermediate cluster is again a bit a mixture of both with these anterior posterior midline regions, but also the anterior insula. How does the picture look like for the temporal parietal junction? Here, tasks that fall in the cognitive cluster, for those, the temporal parietal junction co-activates again with anterior posterior midline regions, temporal parietal junction bilaterally. For the tasks in the affective cluster, the temporal parietal junction co-activates with different regions, namely the anterior insula again, for instance. And again, the intermediate cluster is a little bit in the middle. Okay, now this is very interesting, and it tells us that these networks apparently start cooperating, but that they can also be apparently separated in some tasks. If you really want to know how they cooperate and how they are linked together, what you need to do is you need to measure empathy and theory of mind within the same participants in the same type of task. And this is what we try to do in this task here. We named it the empatom because it's supposed to measure empathy and theory of mind. What does it look like? You're presented with a short video clip. In the video clip, narrator tells you a short story that can be either emotional or neutral. And after that story, you're being asked how you feel yourself right now. With the logic being that if you feel more negative after an emotionally negative video, you empathically shared the emotion of that person. And this is actually what people report. In that rating, they rate their own affective states to be subjectively more negative after the emotional videos than after the neutral videos. And this goes along with activation in a widespread network, including also the anterior insula on both sides and a couple of other regions overlapping with meta-analysis. As you can see in the white outlines, this is a meta-analysis on empathy tasks. The combined activation in these regions also moderately correlates with the ratings. Now the task goes on, and you're presented with a question, and the question comes in two flavors. Either you're asked what Anna thinks, so you're asked about the mental state of the narrator, or it's a factual question about the content of the video. This is kind of our control condition. This is the theory of mind condition. If we contrast the two, what we get is activation in a different network. You get all these temporal regions and anterior and posterior midline regions that you also saw before, and that overlap also with the meta-analysis on theory of mind. Again, if you combine activation here, it moderately correlates with behavior. The better you are, the stronger your activation moderately is going to be. Okay, now as we can apparently test both functions with this task, we can also look at the specifics, and this is what we did here. We directly contrasted empathy and theory of mind. And when you do this, you see very clearly and concisely the networks that we've seen already. For empathy, for instance, the anterior insula, empathy versus theory of mind. For theory of mind versus empathy, you get all the temporal regions and also these anterior posterior midline regions, for instance. We positioned these also in resting state, so seeded from the task-related regions here in resting state. And what we get is, irrespective of where we start to seed, is different networks with the typical default mode regions related to the theory of mind peaks, and the typical salience regions, including the anterior insula, related to the peaks that were derived from empathy activations. So also in resting state, it seems like we can really dissociate these networks. Now, there are also other ways to look at this, and we can, for instance, also directly correlate the two functions with this task as we assessed it in the same people. When we do this in terms of behavior, either within the task or when averaging across different empathy and theory of mind tasks, we do not see any correlation whatsoever. And also in terms of the activation, irrespective of how we derive it directly from the task, there's no correlation here. So it seems like also on an inter-individual level, these networks, at least they can be separate and they can vary separately. If this is the case, then what we would also expect is these networks to be differentially changeable. And in terms of inter-individual differences, we look, for instance, at people who are aggressive offenders. These are people who have all committed serious bodily harm to other people. And what we see here is that they are not different from controls in terms of theory of mind, but they rate, at least subjectively, that they feel less empathy with the person than in the control group. In contrast, in aging, we see a theory of mind deficit. So older adults are less good in answering the theory of mind questions compared to the young adults, but they are not different in terms of empathy. They feel with the other as much as the young adults do. So in terms of older adults and aggressive people, we can see that differential patterns of impairment in these two functions. We can also see that we can induce short-term plasticity. For instance, by music, if you play emotional music along, you see that people rate their emotions, they rate that they empathically share the other's emotions a little bit stronger than if the music that was played was neutral. And this has no effect on accuracy or reaction times in the theory of mind questions. So also in terms of short-term plasticity, you can differentiate the two, selectively target one. And you can also look at long-term plasticity. This is a study done by Sophie Falk and Mathis Trautwein, together with many other people, including me, where we did training people engaged in a 3-month module that focused cognitive functions, for instance, via an observing thoughts meditation, or that it had targeted affective functions, for instance, a loving kindness meditation. And after 3 months of doing this, we see after this cognitive module an increase in theory of mind performance compared to the other groups, the active control group, this task here, or a passive retest control group. And we see an increase in compassion, also compared to the cognitive module or the retest control group. And now, interestingly, this behavioral increase is also related to an increase in cortical thickness. For instance, in the anterior insula, the stronger the increase in cortical thickness here, the more the people have increased in their compassion scores after the training. Compassion being a social emotion like empathy, a little bit different, but also associated. And the more you increase in cortical thickness in this temporoparietal region, the stronger your increase in theory of mind performance after the cognitive module will be. So also in terms of long-term plasticity, it seems like we really can differentiate these two functions. Nevertheless, I wanted to highlight, a big shout out for Sophie Falk's talk. It's already happened. It was earlier today. Sophie Falk, who did the structural cortical thickness analysis here, also had a really nice talk on resting state data from the study. If you want to have a look at it, I can only recommend it. It's even though it happened, it's now, of course, online, and you can still have a look at it. Okay. So what we saw is that the networks really are differentiable. This is in line with what we also saw from the meta-analysis, the big one, that we do, when we look at all the tasks in the field, we do get a cognitive and affective cluster. So they are separable. And they are also quite independent in the sense that they can be independent. They can not, under some circumstances, they are not correlated. They can also be selectively impaired or enhanced through training or inductions like music, for instance. Nevertheless, you can also ask whether in some situations these networks start cooperating or in some way being related to another, interacting in some way. And as we tested in our task both functions at the same time, we can look at this, and we did this with dynamic causal modeling. We selected 2 regions of interest, the anterior insula as a kind of a prime part of the empathy-related network, and the temporoparietal junction as one of the prime regions related to a theory of mind. And when you look at the relation of these two regions, what you see is that in emotional situations, when emotion is high, basically, we see an inhibition of activity in the temporoparietal junction by activation in the anterior insula. So in these emotional videos, there we see such an inhibitory influence. And interestingly, this is related to reduced performance in the theory of mind questions, basically in questions like Anna thinks that ABC. So what this means is that in some situations in our task where we have very strong emotional situations, very strong emotional stories, it can be that the empathy related network has an inhibitory influence on the theory of mind network. But it also means that these networks start interacting under some circumstances. Together also with Lara Maliske, what we did is we looked, this is basically our study, together with Lara Maliske and Matthias Schutz, we tried to find as many studies that did this type of effective connectivity between different networks in social tasks, and kind of review what the connectivity really looks like. For our task, as I just showed, we found this inhibitory influence of the insula on the TBJ. The networks that interact here would be, in terms of resting state networks, be related to the default mode network and the frontoparietal network. Now, we found 6 other studies that also looked at effective connectivity between different networks during social tasks. And as you can see here, the pattern becomes relatively complex, especially the more regions are in there. It can be super complex. And I'm not going to be able to discuss all that in detail today. But my point, the point that I want to make here is that it really is the case that these networks start interacting in more complex social situations. What we saw from the meta-analysis is that there are a couple of tasks that are intermediate, where the networks are somehow co-active. We know that they can be jointly active. We don't really know how they are related. These studies here now give a first hint at how they might be related, but I think we're just scratching the surface here on how in more real life, more complex social interactions, these networks might start to interact. And this is, in my view, kind of what we also need to engage in next is have people do more complex social tasks, have different networks being active, and look at their interrelation to be better able to explain things. All right, so I'm coming to an end. I hope I'm still in time. To summarize quickly, on a meta-analytic level, within task and within subject, we can differentiate these two networks related to empathy and theory of mind. We see a non-correlation if you look at correlation. We also see that in development, for instance, or psychopathology, they can be differentiated. They can be selectively impaired, arguing for independence of these networks. And also in these functions. And also they can be differentially plastic in terms of what you train specifically. Nevertheless, we also see co-activation and even interaction of these networks in some tasks. And I think this happens in more complex, more naturalistic social situations. And if you really want to understand what's going on in actual social interactions in more complex situations when it becomes difficult, I think as I just said, what we need to do is engage in more realistic social neuroscience and look at the interaction of these networks also. With this, I'm coming to an end. I would like to thank all the people in the lab in Dresden. It's incredible fun to work with you. Thanks a lot. I would also like to thank Matthias Schutz, who is the best collaborator you can think of. And also Mathis, Anne Böckler, Mathis Trautwein, Anne Böckler, and Sophie Falk, who contributed a lot and who are also the best collaborators you can think of. And of course, also other people who contributed, especially also Tanja Singer. And I would like to thank you all for your attention.
Thank you for your really great and interesting talk, Philip. And yeah, I would open now the question session, and maybe I start with one question which has been written in the chat really at the beginning, because I think it's also a large question that is related to the bigger topic of the talk. And oh, it's already here. Yeah, the first question was, I don't quite understand why or whether one should divide empathy and TOM in cognitive and affective forms or not, just in general, not related to the talk, is meant by the comment. So why would we differentiate between those two?
It's an incredibly good question. I think this is, it comes a little bit from the terminological turmoil that is going on in the social neuroscience field, or in the field that looks at these processes in general, maybe. In my view, these terms have been used for very different things by different people, and I'm not attached to any particular usage. So my argument really is that we need to be careful in what we mean, and we need to differentiate some things, and for those, it might be helpful to use different terms. So for instance, empathy has quite often been used as an umbrella term, referring to perspective taking, theory of mind, basically, to empathy or affect sharing, and also to compassion in some cases. And from looking at the neural evidence and also the evidence I showed here, I think what is relatively clear is that we need to differentiate between functions like perspective taking, affect sharing, and also compassion. I didn't go into detail with that, but compassion is also a little bit different. And when we know that we need to differentiate between those functions, I think what it tells us is that it would be good to use different terms for referring to these so that we don't get this mix up. So in my view, in my ideal world, I would use a data-driven approach, like we did with the meta-analysis a little bit, and look at what functions group together, and then find terms that ideally describe these. And then if you call empathy affect sharing or empathy, I don't really mind. I just want to make sure that you differentiate it from something like cognitive perspective taking. And cognitive empathy, for instance, and affective theory of mind have often been used for cognitive empathy as a synonym for theory of mind, which is fine with me as long as you're clear on what you want to say with it. It might be easier to just use theory of mind or mentalizing to make it a clear distinction to affect sharing or empathy. And affective theory of mind has often also been used to refer to deducing others' affect. And depending on the nature of the task, I think you don't really need to engage into affective processing in order to deduce others' affect, but you might in some cases do so. In some of the tasks that fall into our intermediate cluster, these are tasks where you actually do that, where people are asked about the emotions of the others in a complex manner. And that seems to engage both networks, as I showed. But there might also be situations where you are on a very abstract level reasoning about the emotions of the others and not emotionally engaged yourself. And this should only activate regions related to theory of mind then.
Do you think we also need to differentiate between empathy in positive and negative emotions, and also in cognitive?
Yeah, absolutely. So I think what I spoke about today mainly is empathy for negative emotions of others. And as I said earlier, empathy is a function that yields an isomorphic emotional state to the emotional state of between the observer and the observed person. So if it is sadness that I share with the other, it's a negative emotion, and regions that are related to my own sadness will be active when I share the other person's sadness. If I share somebody's joy, the regions that are typically associated with joy will be activated. And these are different regions than the regions associated with sadness or grief, for instance. There's, for instance, a study where they looked at a game show, and the people either won the game show themselves or somebody else won the game show. And if it was somebody who was a nice guy, people started sharing the emotion with the other. They were happy for the other that he or she won the lottery. And this yielded activation in the ventral striatum, both when observing the other winning the lottery, but also when winning the lottery oneself. So for empathy or affect sharing, I think we really need to be clear on what the emotion is, and positive and negative might be relatively sensible categories here. For theory of mind, the knowledge that you create, or the representation, if you want to call it representation, that you create is an abstract propositional one. So in that sense, it should be independent of the modality. I can I can share your thoughts on the future, on the past. I can share your emotion. I can share your intentions. If they are represented in an abstract propositional manner, then the network that does that shouldn't be too different. And when you look at the network that enables theory of mind, it's basically the default mode network that is also engaged in episodic prospection and episodic memory, for instance, and also in mind wandering. So instances where your thoughts are decoupled from what is currently going on in the outside world. You're either mentalizing about yourself in the future, yourself in the past, or somebody else's mind. And all of these activate the same network, which gives to me the impression that it's really an abstract amodal thing that is going on, and you don't really have specific patterns. There might be some specificity within that network, but the regions itself shouldn't be too different.
That's interesting. So maybe many networks for empathy, then depending on the emotion. There's one other comment which I would want to read out. It's a question that you could comment on the similarity of the TOM and the default mode network with the semantic network, which is quite similar to the intermediate cluster network you observed.
Yeah, that's also a really good question. So the I showed a couple of data sets where we seem to show quite clearly that the regions engaged in theory of mind really are the ones that are also, that they fall within the default mode network quite relatively well. If you want to have a correct estimate, you can look at the Cortex paper that I cited, Matthias Schutz, Lara Maliska, and myself in Cortex this year, where we really calculated the overlap of different theory of mind task activations with the default mode network. It's quite extensive, especially for the false belief task or trade judgments, for instance. And as I just said earlier, I think it makes sense if you think about the default mode network as one that is on the abstract amodal end of this gradient, principal gradient of macro scale cortical functioning, cortical organization. And it allows this abstract representation probably of information, which is required for mentalizing about all kinds of things going on the other's minds. And in that paper, we also calculated the overlap of the empathy tasks with resting state networks. And the picture there is a little bit more diverse, but it's basically the frontoparietal network and also the ventral attention network, the salience network that are mostly maybe where the overlap is strongest. And these are networks also that we know are engaged in salience, and emotional is a signal of salience. And so I think it makes sense in that regard. In terms of language, I think it was semantic processing. For the intermediate cluster, when we did the neurosynth decoding, we did indeed get also some terms from the language field broadly. I'm not so sure if it's language specific. I think in terms of the neurosynth database, what one can say is that there are really many language studies in there. So you do get language terms also because there are just that many studies from that field in there. So that doesn't make it specific, but it may be that some things are shared. To be honest, I haven't thought that through yet to the extent that I would make speculations right now, but it's a good point, and one should probably do it at some point.
Maybe we have time for one last question before the time is running out. The question is, how do you understand the inhibition effect from anterior insular to TPJ in DCM analysis? Does it mean anterior insular increase or decrease activation in TPJ under emotion conditions?
Yeah, also good question. Thanks. It explicitly means that the anterior insula inhibits TPJ. It inhibits activity in the temporoparietal junction. And as we saw, this has effects on how well you mentalize. It decreases your performance in the theory of mind questions that come later. How you can interpret this is, I think, in a kind of emotion distraction sense, that if you are yourself in a strong highly emotional situation, as as we can see for many other emotional tasks, emotions can be distracting from other cognitive tasks. They can somehow deduct resources from if you want to use the term resource, and impair performance. And this is a little bit what we see here. I think an evolutionary function might be that if something emotional is going on in the outside world, emotion, as I said, means salience. It's a salient event that you want to orient attention to immediately. You don't want to mentalize too much about the other. In that instance, you want to be able to act fast to that salient emotional situation going on outside. And that's why maybe the TPJ is inhibited. It's a pattern that you also see in attention tasks, for instance, that and here Insula inhibits default mode network activity, for instance, in highly salient situations or task control situations. So in that sense, I think it makes sense to see that pattern here. But I could imagine that there are also other situations where the networks more work together, where, for instance, the default mode network kind of reads out the information from the insula to inform what type of emotion is really present in the moment. And as we saw from the overview of all the effective connectivity studies, the patterns are quite complex, and one really needs to look at the details of the tasks, what might be going on.
Thanks a lot. Actually, we are quite exactly in time, so I'm not sure if the session will end in a few seconds. I want to thank you a lot for your talk. I think there are many open questions which you will hopefully answer in later talks at some point. And I thank
also,
all
maybe
of you
just let me add that if you have any questions, feel free to text me, and I'm happy to respond also if I have the time. So that would be great. Get in touch if you're interested in our research or have questions.
That's a great opportunity. Perfect. Thank you very much. And thanks for all who shared the questions with us and the discussion, and have a nice evening. Bye.
Take care.
Action.
Okay, so we're live. And we start the session, start the recording, waiting for our attendees. And just to start saying that I'm your host for today, along with Isabella, who are backend and Zoom wizard. My name is Maria. I'm postdoc at EPFL Switzerland. And today we have exciting sessions with 3 speakers. Talks are 12 minutes long, followed up by a QA session. And we really need these questions. They need to be answered, and they can be posted in the QA. But we can start now with our first speaker, Andrew Matheson, a PhD student from Kathy Nagel Lab at New York Neuroscience Institute. You can find some information about his recent paper in the chat. We'll have a link there, and we're looking forward to hear your presentation, Matthew. Andrew, sorry.
All right. Up. All right. Well, hi, everyone. My name is Andrew Matheson. I'm a grad student in Kathy Nagel's lab at the NYU Neuroscience Institute. And today I'll be talking a little bit about the central circuit's for olfactory navigation in walking fruit flies. So olfactory navigation is a very ancient behavior, and it's the way that many organisms are able to find their food. It's been an informative behavior in elucidating how neural circuits function. So even in bacteria, which don't have neurons, we've been able to work out the enzymes of the circuit that allow them to move on chemical gradients, which is of course important to them. We have some understanding of how organisms navigate on chemical gradients that are on the substrate, such as C. elegans, but most organisms actually experience odor in a different way. And this is in a complex filamentous plume that's carried on the wind. So the conditions of the airflow will set the turbulence of the plume and dictate how much it fluctuates. This is a laser image plume of acetone from our collaborators at the University of Colorado Boulder. And you can see that close to the odor source, it's pretty consistent. But as you get further away, it's filamentous, it's uncertain, and it's fluctuating. So navigating in this stimulus is a difficult task for the nervous system to solve. But one group of organisms that's particularly good at navigating in this environment is insects. And it's been well established that the way that insects can navigate in this complex environment is by combining the direction of the wind, cues about that, with the presence of odor to move upwind towards the source. So we'll be using the model organism Drosophila to study the neural circuits for navigation. And this is a great place to do it because flies have relatively small brains relative to humans or mice, but they've got about 1,000-fold more neurons than C. elegans, and there's a wealth of genetic tools available in the fly to both label genetically defined subsets of cells, but also to manipulate their activity. To study navigation in the fly, I'm going to be using a behavioral paradigm that was designed in the lab by a former postdoc, Efrain Alvarez Salvado. These are miniature wind tunnels that allow us to get a fine-scale picture of the motor behavior that flies are doing in response to an odor while navigating. So what we do is we illuminate the flies from above with an IR LED plate and image them from below with a camera. The arena is made up of thin acrylic plates where we basically sandwich the fly in there so they can only walk and cannot fly. We have constant laminar wind flow coming through the arena, and we're able to inject a square pulse of odor into this to see how the flies respond. This is a very simple stimulus that we're using here as opposed to the complex one I showed you earlier. This allows us to correlate the fly's behavior with the stimulus. And one major benefit of the setup is we can see not just how the fly got there, not just if they made it to the source. So what does this data look like? Here I'm showing you the example of one video trial from a single fly. The box here is the borders of the arena. The blue dot is where the fly would start. In gray is where the fly walked during the period before the odor comes on. So you can see that the fly moves along the downwind end. They initially have a downwind preference in the arena. When the odor comes on, here it's apple cider vinegar, an appetitive odor to the fly. It smells like rotting fruit. The flies run upwind towards the source. When the odor goes off, the flies begin to turn and search, trying to re-encounter that lost odor. If they're in a real plume, it should be nearby. So Efrain showed that these two sensory motor transformations are how flies navigate in an odor environment. And of course, we can quantify this over many trials across many flies. And the two parameters we look at for this are upwind velocity for this upwind run. So we can see that the upwind velocity increases during the odor period as the flies head upwind towards the top of the arena. The metric we use for quantifying local search is called curvature. It's a measure of the tortuosity of a curve, how tightly wound and bound it is. And you can see the curvature rises at odor offset here. You'll also notice that the curvature rises initially, very briefly, and this is because the fly can be facing in any direction when the odor comes on, and then they'll run upwind towards the source. So the central question that I'm looking at is, what are the circuits that promote sensory-guided navigation in the fly, and where in the brain is this wind direction signal integrated with odor information? So today I'll be walking you through some of the circuitry in the fruit fly brain. I'll be starting at the sensory periphery and working my way to a central region called the fan-shaped body. Now, as a reminder, flies smell with their antenna, and in the antenna, their olfactory receptor neurons are housed. These are taken up to the antennal lobe in the fly, and here there's a glomerular organization, much like in the mouse olfactory bulb. So when we started this project, we wanted to design an optogenetic approach to be able to activate different populations of neurons and see how the flies respond. To do this, we expressed the red light shifted channelrhodopsin called crimson under a particular group of cells. And then we replaced our odor stimulus with 10 seconds of red light instead of the odor. So now we're going to activate the cells from above using red light and see how the flies respond. We started using the OrcoGal4 driver, which expresses in the majority of the fly's olfactory receptor neurons, though not all of them. We wanted to see if we could make like a fictive odor for the fly. And indeed, this was the case. So when we activated this group of olfactory receptor neurons, we saw that flies would run upwind in response to the activation, and they would produce local searches at odor offset. This is great. We have a tool now where we're able to go in and probe the role of a particular group of neurons in the brain and see how the flies respond in our behavioral arena. We next took this to some higher order olfactory regions, the lateral horn and the mushroom body. And these are two parallel higher order olfactory areas in the brain. Traditionally, it's thought that the lateral horn is important for innate olfactory behavior, while the mushroom body is important for learned olfactory behavior. Though this dichotomy has sort of been breaking down with some recent work that's been coming out. Now, some other groups have recently identified output neurons from these regions, lateral horn output neurons, mushroom body output neurons, that go deeper into the brain that were able to drive attractive behaviors in the fly. What we wanted to test is this attractive behavior correlated with our upwind runs in local search? So we took these recently identified attractive output neurons, and we saw that when we activated them in our behavioral arenas, we were able to produce upwind runs as well as local searches at odor offset, suggesting that these are correlated with that behavioral attraction. And I'm just showing one example line here each, but we did find a variety of output neurons from both areas that drove this behavior as well as others. Next, we wanted to ask, at the level of the lateral horn and the mushroom body, is this where wind direction and odor are integrated? And to do this, we used a 5-direction odor wind manifold where we're able to puff odors at the fly from different directions while doing 2-photon imaging that was originally developed by Marie Super in the lab. And what we found was that at the level of the lateral horn and the mushroom body, there's a non-directional odor signal that's encoded. So what I'm plotting here in the top in the colored traces is just the average of 5 different trials for an individual fly for each direction. And then below is a heat map that shows the individual trials. What we can see here is that they respond to every direction and that they're responding pretty evenly to the odor, which is apple cider vinegar in the little blue box for 10 seconds. So this suggested to us that odor and wind information must be integrated downstream. So where does this information go next? We did not notice at the start of the project, and we used a tool called TransTango, which allows you to label all postsynaptic cells to an individual GAL4 driver. And this identified the fan-shaped body as a region where the information was going. Specifically, the dorsal layers were labeled by these lateral horn and mushroom body output neurons. So the fan-shaped body is a region that's thought to be involved in navigation. It's part of the fly's central complex. The central complex is known to have orientation signals as to different types of stimuli and as well as encode the fly's heading. The fan-shaped body has 9 different layers as well as 16 columns, which may represent space around the fly. So the approach we took was to use an optogenetic activation scheme of different GAL4 driver lines of fan-shaped body neurons. I'm showing you here results from the screening the inputs, either to the dorsal or ventral layers, though we did look at a variety of other types of central complex neurons as well. And what we found was that these central complex neurons, when activated, were able to change the fly's motor behavior in a variety of ways. But what was of particular interest were these ones that were able to drive the fly upwind. And so we identified some inputs to the fan-shaped body. These go to the dorsal layer that encode odor and can switch the fly's wind orientation. So when activated, this dorsal fan-shaped body input can drive the fly upwind. And at this level, we're seeing an uncoupling of the upwind run in local search. Here, the flies are only doing an upwind run and not a search. We also saw that these neurons encoded odor. So much like the lateral horn and mushroom body neurons before them, we see that they respond non-directionally to the odor and that they're responding for every direction. So we have a pure olfactory input going into the dorsal layer of the fan-shaped body. And recent work from my lab mate Tim Currier showed that there is a pure wind signal going to the ventral layers of the fan-shaped body. So Tim identified this group of neurons called PFMs. They go between the procerebral bridge, the fan-shaped body, and the noduli, which are 3 regions of the fly central complex. And he showed that in this particular group, they're responsive to the direction of airflow. So these ones are ipsilaterally tuned. They respond to airflow from the same side. And when Tim presented either clean air or air with odor in it, he saw the responses did not change, which suggests that these neurons really only care about the direction and not the presence of odor. So we have pure odor input going into the upper layers, and then we've got pure wind information going into the lower layers. And using the recently published fly EM hemibrain, Aaron Lands, a rotation student in the lab, and I identified this group of local fan-shaped body neurons, which are downstream of both of these. So they receive input from these wind direction selective PFNs and odor information from these dorsal fan-shaped body input neurons. So this might be a site where wind and odor are integrated. So we hypothesized that these local neurons could couple wind direction to different motor programs, and that odor inputs could then gate these local neurons and how they respond to wind orientation preference. When we activated these neurons, we're activating them all at once, not in a particular column, but we saw that we were able to produce these sort of directed, sort of curling, searching trajectories going off in a particular direction, which was quite interesting to us. And when we imaged from them, we saw responses of sort of 2 flavors. The first was that we got column-specific direction-selective odor responses. So here I've taken an ROI around just 1 column in the fan-shaped body. And what we're seeing is that they seem to have the strongest response to one particular direction. We saw another type of response as well, which is where the flies seem to respond just to one particular direction and not the others. So what this says is that it appears as though these neurons are capable of integrating this wind direction and odor information and responding in a specific way, only to one direction, unlike what we've seen before. So in summary, today I told you about how we designed an optogenetic activation paradigm to identify the neural circuits involved in olfactory navigation. I showed you that higher order olfactory neurons promote both upwind runs and offset search, and that these neurons respond non-directionally to odor. We then traced this pathway deeper into the fox brain, into the fan-shaped body, part of the central complex. And here I showed that there's pure odor information as well as pure wind direction information going into the fan-shaped body, and that a particular subset of local fan-shaped body neurons are posed to integrate this information, picking up both wind direction and odor information to turn that into a directional signal for the fly. So thank you for listening. I'd like to thank a couple people. First, Kathy Nagel, my mentor, the people who worked with this on me, Aaron Lands, Tim Courier, and Angela Licata, as well as the rest of the Nagel lab, the Chopik lab, who we share space with, as well as the Staropoulos lab and the Sayed lab at University of New Mexico, and then my thesis committee, Dima, Niels, and Nick. And I can now take any questions.
Awesome, thank you very much for this great talk. Amazing data. And we are ready for questions. Panelists as well as attendees can ask any questions. Don't be shy. Okay, then I just wonder, what do you think would be the next step for you then? Because it seems like pretty much lined up everything. So what are the non-answered questions there?
So I think one of the things we want to look at next is, these responses are done in a fly that's basically hanging under a 2-photon microscope in the dark. So they have really no context of how the world around them is. So one of the things we want to do is put the fly on a walking ball and be able to have them see their real environment and then activate, present the same stimuli and see how the flies respond to that. We're also thinking about sort of wanting to activate individual columns at once. So here we're activating all the columns together, which is a bit confusing to the fly, I'm sure. That's not how it would naturally be based on the physiology. So we're hoping to use some genetic tricks to label either a small subset of columns or only single columns, activate and see how the flies respond.
Okay, great. Great, thank you. And I think we can start moving to our next speaker. And if you have any questions further on, please just post them in the QA and they will be answered. So our next speaker is Denise Weber, a PhD student working at Andres Trum Laboratory in Leipzig University. And we are looking forward to hearing from you. Go ahead.
Yeah, thanks, Maria, for the kind introduction and also for having me. And hi to all the other attendees today. So yeah, as Maria already mentioned, I'm a PhD student at Andreas Thum's lab at the University of Leipzig. And our lab is basically interested in the mysteries of learning and memory formation. And therefore, we are using the Drosophila larvae to understand those phenomena better. So some of you might wonder to what extent the ability of such a complex behavior like learning and memory formation is a simple organism can be applied to a higher organism such as humans. Well, not only the amount of neurons varies just a little bit, but also, let me shortly switch to the laser pointer. Yeah, the amount of neurons, but also the fact that only one structure in the Drosophila brain combines 3 unique features that also apply to mammal brains. The mushroom body, which you already heard about at Matthew's talk. So yeah, first of all, the mushroom body receives sensory information, mostly olfactory information, and this is similar to the piriform cortex in higher organisms. Secondly, the information is processed, evaluated, and converted into a response determining a signal comparable to the function of the cerebellum. Last but not least, the mushroom body serves as a storage and for associated memory and stores learned information, and molecules that are involved in this consolidation can be also found in the hippocampus of mammals. So the mushroom body is a really astonishing anatomical structure. And it also allows us to have a closer look how those phenomena are processed. And we are using, as I said, the larva as a model organism. And even here, compared to the adult fly brain, the mushroom body is way more simpler. So it doesn't consist anymore of the different lobes, alpha, alpha prime, beta, and so on, but just a calyx and the peduncle dividing into the ventral lobe and a medial lobe. And also anatomical analysis of the mushroom body revealed that there are only 11 compartments, all of which have a limited number of specific mushroom body input neurons, MBINs, and specific mushroom body output neurons, MBONs. So we are today focusing on the mushroom body input neurons, which can be dopaminergic, octobaminergic, or of unknown identity. So far, it has been shown that especially dopaminergic neurons in the medial lobe are necessary to process appetitive learning, whereas the function or the compartments where aversive learning is processed remains largely unknown. So the method we are using is quite astonishing. We are using split GAF4 lines, which allow us really to change the function or the activity of single neurons that are innervating the mushroom body in those different compartments, as you can see here. So we ordered or tested all available mushroom body input neurons and crossed them to an effector line. Here we used UA as a sweeper just to ablate those neurons and then test them for their function in aversive learning. The paradigm we used is a classical conditioning paradigm where we used a high salt concentration as an unconditioned stimulus and two different odors, amyl acetate and benzaldehyde, as conditioned stimuli. We also varied the amount of training cycles, one cycle just to trigger the short-term memory, or a freeze cycle training to have a closer look at the larva anesthesia-resistant memory. 12 minutes is not a long time, so I won't show you the data that we created for all the split GAF4 lines, and I will just focus on the two important ones. It's a combination where we are able to ablate down F1 and down G1, innervating the ventral lobe of the larval mushroom body. First of all, let's have a look at the data that we have for the reward learning. So we used fructose as a appetitive reinforcer. And also here we tested in the 3-cycle training and the 1-cycle training. And we saw that ablating those two neurons of the ventral lobe has no effect on their learning performance. Also, we did a gustatory preference test to see whether this ablation has an effect on the chemosensory perception of fructose. And also here we saw that larvae with ablated neurons are perfectly able to receive fructose as something appetitive. Then, of course, we tested for aversive learning because that's what we are interested in. Here again, an anatomical picture where you really can see that those two neurons are innervating in the ventral lobe of the mushroom body. Again, here, the data for the 3-cycle training that we performed, and we saw that even if we ablate those two neurons in the ventral lobe, they are perform not significantly different to the controls. But when we performed a 1-cycle training, we saw that there's a reduction in performance, which leads to the assumption that these multiple input neurons in the ventral lobe are necessary to process aversive short-term memory. Of course, here we also did some further controls just to check if the test conditions are fine for the larvae. And we saw that even if we ablate those neurons, that the larvae were still able to avoid salt, to perceive it as something really aversive. And also we performed some olfactory preferences for the odors we used, amyl acetate and benzaldehyde. And also here we saw that the larvae were still able to smell those odors. In a collaboration with Katrin Vogt from the University of Konstanz, we also had a closer look at the calcium activities and the ventral lobe dance in response to different gustatory stimuli, and they really verified our results that we had so far. So as you can see here, they really strongly responded to a high concentration of salt as well as to caffeine, which is just another aversive stimuli for them. And we also saw a low response to fructose, which fits with the vivo data that I showed before. Really interesting was the data that we got from her, where she tested the calcium activities for single dopaminergic neurons innervating the lateral appendix. And here we saw really, really strong response to salt, to a high concentration of salt, which already brings me to the conclusion that at least 2 dopaminergic neurons in the ventral lobe are necessary to reduce at least the aversive learning performance in a short-term paradigm. This brings now a lot of questions up. For example, is there a cluster of more than 2 dopaminergic neurons needed for a loss of memory? Based on the data that we got in the collaboration with Katrin, we assume that compartment D in the lateral appendix plays a really crucial role. Another question that is now that we are now trying to resolve is if this phenotype that we see is really specific for high salt or for general establishing a aversive memory. Therefore, we are trying to test or repeat everything that we did with caffeine as an alternative aversive stimuli, or quinine, as well as with optogenetics. And last but not least, of course, we want to figure out which mushroom body compartments are necessary for an aversive larval anesthesia-resistant memory. So yeah, that's the data that we got so far. And thanks for listening. Also want to thank, of course, all the lab members and yeah, Andreas Thum as a mentor, and Vincent for this really beautiful picture that is based on EM reconstruction data. Furthermore, I want to thank Bertram Gerber and Michael Steyer from Berlin and Magdeburg, and Katrin Vogt for the beautiful and interesting physiology data.
Great, thank you very much. Do we have any questions? If any panelists have questions, because we realized that for the previous talk of Andrew, I think they had a question, but it was missed, unfortunately.
Okay.
I could ask a question if that's okay.
Great. Sure.
I saw that in the imaging data, I don't know if this is real, but like, did it seem like the higher concentration of caffeine was actually activating the dopamine neurons less than the lower concentration? And do you know why?
Actually, we have no idea. We are right now trying to figuring out just to test for different caffeine concentrations to see when the threshold is switching from appetitive to, or yeah, aversive to not aversive anymore. Yeah, but we actually have no idea why this this change is there.
I guess a related question, if no one else is asking right now, is like, it seems like at least one of the dopamine neurons is integrating aversive taste and smell, right? Is that or is it just a different kinds of aversive tastes? I guess I'm just wondering whether you think this integration of different aversive stimuli is happening at the level of this neuron, or is it happening somewhere else and then just being passed along to this neuron?
That's actually a good question. So the hypothesis that we have so far is that we have at least those two compartments that are necessary for processing the aversive memory, and that there's maybe another, at least one compartment for storaging or the information, and that there's really then a complete loss of performance. Yeah.
Okay, thank you again. And we move forward to Anita Devineni, who is postdoc at Richard Alex Lab at Columbia University. If you check our chat, you will find there her Twitter tag. And we encourage you to follow her on Twitter, as well as check her blog post, where she promises to explain brains. And it sounds very exciting. She also does teaching, and you can also find in the chat a link for one of her latest papers. And I have to say that Anita is on the job market, so keep it in mind, please. Please go on.
Thank you, Maria. Okay, I'm unmuted. Yeah, so thank you everyone for being here. All right. In general, I'm interested in how the brain translates sensory information about the world into behavioral output that allows us to interact with the world, and how this transformation is modulated by things like experience and internal state. I've chosen to address this question in the Drosophila taste system. Taste is a really important sense because it ensures that we consume caloric or nutritious foods like sugar while avoiding potential toxins that are often bitter or sour. And these different taste modalities are encoded by distinct neural pathways. So the timing of neural responses is a key feature of encoding in many systems, but very few studies have investigated temporal dynamics in peripheral taste coding. So that's what I've set out to do. Flies can taste with many different organs, including the legs and the proboscis, which is the feeding organ. In this video of a fly feeding, you can see the proboscis being used to sample and consume the food. So I wanted to use calcium imaging to examine how different kinds of taste sensory neurons encode taste stimuli. These sensory neurons have their dendrites and cell bodies out in the periphery, but most of them project axons directly into the brain. So I can image sensory activity here at the axon terminals. I can genetically target GCAMP to specific classes of taste neurons, and I built a taste delivery system that delivers small drops of tastants to specific organs, such as the proboscis. Here's what I see when I image the sugar sensing neurons in the proboscis, and I stimulate the proboscis with sucrose. The GCAMP signal goes up when sugar turns on, goes down when sugar turns off. So this is exactly what I expected to see, and it's what other labs have published. When I image another class of taste neurons, the water sensing neurons, they also respond in the same way. But I see something different when I image bitter sensing neurons. There are two peaks of activity. The first peak occurs at bitter onset, so we call this an on response. The second peak, which is actually larger, occurs at bitter offset. So this is an off response. And this was surprising because no one has reported such large and striking off responses in the fly taste system. This off response is evoked by several different bitter compounds and by different stimulus durations. So it seems to be a general feature of bitter encoding, but not in all bitter neurons. So far, I've only shown you the responses of bitter neurons in the proboscis. If I instead image bitter neurons in the leg, now there's no off response, and you'll also notice that the on response looks much more sustained, whereas in the proboscis, it's more transient. This means that these two different organs are encoding the exact same bitter stimuli in fundamentally different ways. The leg encodes the presence or absence of bitter, whereas the proboscis encodes bitter onset and offset. So this two-peaked response profile is specific to bitter neurons in the proboscis, which suggests that it evolved for some particular reason that we'd like to understand. First, I confirmed that individual cells generate both of these two peaks, so it's not like there are separate on and off pathways. And additional experiments suggest that the off response is generated cell intrinsically, likely using the same bitter receptors that produce the on response. So next, I wanted to know how these dynamics influence the downstream neural circuit. I focused on a set of neurons called the PPL1 dopamine neurons, or DANs. These neurons generally encode aversive stimuli, such as electric shock or heat, as well as bitter taste. If I image one of these DANs and deliver bitter compounds to the proboscis, I see responses with two peaks, an on response and an off response. If I image the same cell and deliver the same compounds to the leg, now there's only one peak, only the on response. So the dynamics I observe in sensory neurons are conserved in these downstream cells, which suggests that they're probably important for something. So I next wanted to ask how this interesting two-peaked response might impact the function of these neurons. So let me tell you a little about their function. These dopamine neurons innervate the mushroom body, which you've just heard about in the last talk, and it's the learning and memory center of the fly. And these DANs are known to mediate aversive olfactory learning. Thanks to the work of many labs, we now understand how this works. If you pair an odor with a punishment, like electric shock, you activate some of the odor responsive cells of the mushroom body, called the Kenyan cells, at the same time as those PPL1 DANs, which encode punishments. And this coincident activation induces synaptic depression onto attractive output neurons. There's a balance between the attractive and aversive output pathways. So when you suppress the attractive pathway, then the aversive pathway dominates, which generates avoidance to the conditioned odor. So clearly, the timing of the dopamine neuron's activity is really important because it needs to coincide with the odor. But if the punishment I use is to deliver bitter to the proboscis, I know the DAN isn't going to show a smooth response like this. Instead, it will show two peaks, the on and off response. And in the paradigm depicted here, the first peak will overlap with the odor, but the second peak will occur afterward. And I wondered whether this might disrupt the synaptic depression that we would normally expect to see. So I did the experiment. I repeatedly delivered odor and bitter in this way to flies on the imaging rig. My readout of plasticity is to image this output neuron's response to the conditioned odor. So if there's synaptic depression here, then the response will decrease after pairing. And from these data, I calculated a plasticity effect, which represents how much the response to the conditioned odor changes after pairing. When I pair odor and bitter in the way that I just described, where odor predicts bitter onset and overlaps with the on response, normally we expect to see synaptic depression. But instead, I see that the response to the conditioned odor gets larger, which we think represents synaptic facilitation. In a second set of experiments, instead of pairing odor with bitter onset, I paired odor with bitter offset. So now the odor overlaps with the off response rather than the on response. And in this case, I saw the opposite effect, synaptic depression, which suggests that this synapse is capable of bidirectional plasticity. So what explains these results? Why does this first protocol induce facilitation rather than depression? And why do these two protocols have opposite effects, even though they both should involve coincident activation of the odor cells and the DAN? So the big clue came when I went back and did more experiments to image the DAN during the learning paradigm, which remember involves repeatedly pairing odor and bitter. I wanted to make sure that the DAN really does show two peaks in its bitter response. And I saw something interesting. Even in the absence of odor, if I repeatedly deliver bitter at 10 to 20 second intervals, like I do for the learning experiments, on the first trial, you see these two very nice peaks, the on and off response. But by the third or fourth trial, the on response completely disappears, whereas the off response remains. And this is actually true in the sensory neurons as well, which is very interesting. And this suggests an explanation for these results. The hypothesis is that it's only the off response that really matters in this paradigm because the on response goes away. And if we're just considering the off response, now you can see that only in the second protocol does the odor actually overlap with DAN activity, which is known to cause depression. In the first case, the odor and DAN activity are offset, which some studies have shown can induce facilitation. So to test this hypothesis that it's the off response that really drives these effects, I repeated the same learning experiments, but I replaced bitter with optogenetic stimulation of the DAN. So I can deliver odor in the same way, but now I can artificially stimulate the DAN to mimic having just an on response, just an off response, or both. And I'm not mimicking any habituation. So the question is, which of the two peaks reproduces the effect of real bitter? And we predict it would be the off response. So let's first consider the case where odor is paired with bitter onset, which, remember, with real bitter induces facilitation. If I get rid of bitter and mimic just the on response, I see the opposite, depression. If I mimic just the off response, now I see facilitation. And if I mimic both peaks, I see something in between. So only mimicking the off response reproduces the effect of bitter, suggesting it's what normally drives plasticity. And the on response actually pushes the synapse in the opposite direction. In the second case, where odor is paired with bitter offset, remember, with real bitter, this induces depression. If I mimic just the on response, I see the opposite, facilitation. If I mimic just the off response, now I see depression. And if I mimic both peaks, I see a weaker depression. So again, mimicking the off response alone best reproduces the effect of bitter. And again, the on and off responses have opposite effects. Together, these results suggest that when bitter is repeatedly paired with odor, it's the off response that determines the type of plasticity that's induced. And it can drive plasticity in either direction, depending on the timing of the stimuli. So just to be clear, I think all our results can be explained by the combination of two factors. First, the fact that the off response is really the dominant signal in the DAN because the on response rapidly habituates. And second, there are intrinsic timing rules governing mushroom body plasticity. The simplest interpretation, which I alluded to earlier, is that when the odor and DAN activity overlap, you get depression, and when they're offset, you get facilitation. So I actually made a computational model of this circuit to test whether this coincidence rule alone could reproduce my experimental results. And I also tested other kinds of timing rules, like rules that depend on the order of the odor and DAN activity and the delay between them. And I don't have time to go through the results, but what I found is that there are probably multiple timing rules at play there, including this one, as well as one that does incorporate the order of the stimuli. So stepping back, these two factors explain why using bitter in what looks like a classic aversive learning paradigm here leads to the opposite plasticity effect than usual, facilitation rather than depression. So of course, one intriguing question is whether the behavior is also opposite. Do flies actually show attraction to an odor that predicts bitter is coming, which would be weird? That's something I've been trying to investigate, but I don't have the results now. So in conclusion, I've characterized some interesting temporal dynamics in the fly taste system, and I've shown that the off response is a prominent feature of bitter encoding, and that it can drive synaptic plasticity during learning. I'm currently investigating how the off response might also influence innate behaviors like foraging and locomotion. More generally, off responses are observed in many different systems, but people often tend to ignore them. And my work suggests that maybe we should stop doing that because they might be pretty important. And I have a preprint of this study on bioRxiv, so you can check that out if you're interested in learning more. And I'd just like to thank Richard for his support, as well as everyone in the Axel Lab, but in particular, the people who are listed here. And thank you for listening.
Fascinating. Thank you very much. It's a great talk. So, ready for questions? Do we have any questions? Panelists, attendees? Okay, we have one question. So, awesome talk. It's from our Zoom workshop. So the question is, I'm not familiar with Drosophila, and if it is known how the off part of the response is encoded in the peripheral sensory neuron, would that be determined by a local circuit, or maybe the biophysics of the channel's receptors activated by adverse stimulations?
Yeah, that's a great question. Like, where does the off response come from? And that's a different section of this study that I didn't have time to go into at all. But one question is whether it's a cell intrinsic mechanism generated by the bitter neuron's own receptors, or whether it's a circuit mechanism that relies on synaptic connections or other kinds of communication. So we think it's the first one. We think it's cell intrinsic, and there are a few reasons for that. Like, for example, if I shut down synaptic transmission in the entire brain, just shut down all the synapses, I still see the on and off responses in those sensory neurons. So that suggests that it doesn't rely on sensory and synaptic input. Another piece of evidence is the fact that different bitter compounds actually can elicit stronger or weaker off responses in the same neuron. So the same neuron might respond to one compound with a strong off response and another compound with a weak off response or no off response. And it's the same neuron that's still showing an on response. And so given that we know that different compounds activate different receptors, that really suggests that it's generated at the receptor level, that it's the receptor that's determining it. But how that actually works, like how does the receptor open and close, and what makes it like maybe open again at the offset of the stimulus, that we don't know. We actually don't know a lot about the molecular biophysics of the receptor at all.
Great, interesting. So sorry, but we have a question. Yes.
Yeah, so I had a question, sort of on the same vein. Do you think it's post-inhibitory rebound generating the off response, like the same way it is in the early olfactory system where the off responses are generated? And if so, how do you think that you have the on response that stays initially and then dwindles down still?
Yeah, I don't think it's a general consequence. I don't think the off response is a general consequence of depolarization, which is how post-inhibitory rebound usually is. It has to do with just the membrane potential of the cell becoming more positive and then going back, and that's what triggers the rebound. Because, like, for example, if I activate the bitter neurons with crimson, you don't see an off response. Sorry, crimson is like channel rhodopsin, like optogenetics. So optogenetic activation, bypassing the bitter receptors, depolarizes the cell, as we know, but doesn't generate an off response. So that's also some of the evidence we think that it has something to do with receptor signaling, rather than at least like a classic post-inhibitory rebound effect. Mm-hmm. Thanks.
Okay, thank you. That was awesome. And if you have any more questions, we encourage you to ask it, because soon we have to go off, like in 1 minute or so. So I have questions about the behavioral part. Like, how would you test it even? What would be the setup for the experiments? To look at yeah, it's fascinating.
Yeah, like you mean for the learning stuff, or just in general?
For the learning stuff, yeah.
Yeah, well, for the learning stuff, right, we want to associate odor and bitter in these different ways that I've done, and test whether the fly then shows attraction or aversion to the odor. So it's like the real way you want to do it is some kind of head fix setup, because like to do this learning experiment, it's not like a classic experiment where you can have freely moving flies, because I have to stimulate flies with bitter on their proboscis. So I have to ensure that the bitter touches their proboscis at a certain time. So the fly has to be head fixed, and that's kind of what has been one of the limiting factors. So what I would do is do fly on a ball. So have the fly walking on a ball like they do with mice as well. And like I think some of Andrew's experiments probably are. And you then try to see whether they navigate towards or away from the odor after the odor has been associated. So that's how you could see whether they form an attractive or aversive memory.
Okay, interesting. I can only wish you the best of luck in this endeavor. Thank you very much, all the speakers. It was amazing sessions, amazing talks. I wish you all the best with your research, and have a nice day of the time that you have around wherever you are. Thank you very much again, and see you.
Bye.
I apologize for not having the nice background of NeuroMatch, but my computer is too old and it doesn't it doesn't handle Zoom virtual backgrounds. I feel like I'm the last one left. But I did manage to turn the computer, so there's not my kitchen. But
yeah, my room is too messy to be shown.
But your virtual background, it works perfectly. It works perfectly.
Yeah, I quickly chose one earlier.
Also, I move too much when I talk. It's my hands and everything, so it just doesn't work for me.
So where do you guys go to school or research at? Sorry.
I'm at Johns Hopkins right now doing a postdoc, but I'm presenting, I just started here, so I'm presenting PhD work.
Nice.
I'm at York U, which is in Toronto.
Oh, okay. Yeah, I'm at MIT. I'm doing second year grad school.
Okay, I think we are hitting the 5 p.m. mark, so we're gonna get going. Hello, everyone. I'm Valentina, and with the amazing and fundamental help of Sid behind the scene, I'm here to bring you the next 3 speakers for this great session here at NeuroMatch. We will have Robin Mildren from Baltimore, Maria Ayala from York, and Kami Fioza from Cambridge. Each of them will have 5 minutes to present their research, followed by 10 minutes for your question. So please start writing your questions as soon as they come up in the Q&A section at the bottom of your Zoom screen. It will be my pleasure to read them to the speakers right after. And check from time to time the chat, because that's where we will be posting some link and further information on our great speakers. So with this, let me introduce the first one, Robin. Currently a postdoc at Johns Hopkins University in Dr. Kathleen Cullen's lab, but she will be presenting us her final project of her PhD thesis that was conducted at the School of Kinesiology at the University of British Columbia. Please, Robin, the floor is yours.
Thank you for the introduction. So I'm Robin Mildren, and I'm going to be presenting some of my work from my PhD thesis at UBC. So my work focused on the stretch reflex pathway. So a stretch of a muscle activates muscle spindles, which send signals to the spinal cord to generate a rapid contraction of the muscle. And so I'm interested in studying this pathway in populations that have poor balance control, because we know that proprioceptive information from the ankle joint is really critical for the control of balance. So with aging, there can be progressive changes in different elements of the stretch reflex pathway. And following a stroke, there's a disruption of descending information sent from the brain to the spinal cord, and that primarily affects one side of the body. So how this pathway is typically assessed is by applying a transient stimulus to the tendon to stretch the muscle, and then recording the evoked response around 40 milliseconds later. And so I'm interested in studying this pathway during standing, so when this information is relevant to the control of balance. And one of the drawbacks of this approach is that these abrupt stimuli generate a perturbation to balance. So we've previously developed this method that involves delivering a continuous low-level noisy vibration to the Achilles tendon, and then looking at its linear correlation with ongoing EMG. So this stimulus contains frequencies between 10 and 115 hertz, which are frequencies that muscle spindles can encode and are generated naturally during normal activities. And this allows us to probe responses across a relevant frequency bandwidth without noticeably perturbing standing balance, and also with relatively short trial durations. And so using this technique, the aim of this study was to examine how healthy aging and chronic stroke influence lower limb muscle responses to proprioceptive stimuli during standing. And we also wanted to explore potential mechanisms that might underlie these changes and whether they're related to postural control. So here we tested 54 healthy adults across a broad age range, and that allowed us to examine changes across the adult lifespan, and 8 individuals with chronic hemiparetic stroke. And we examined soleus responses, postural control, and mechanical properties of the muscle tendon unit. So this is showing the cross correlation between the vibration and EMG. So as you lag the EMG around 40 milliseconds relative to the stimulus, those signals are correlated. And this is plotting the peak-to-peak cross correlation as a function of age. So you can see the soleus response amplitude declines with age. So here I've grouped individuals into young, middle-aged, and older groups. And this is showing coherence, which is the strength of the linear correlation between the vibration and EMG across frequencies. So coherence became weaker with age and also spanned a narrower frequency bandwidth. So gain is the muscle output relative to the input. And similarly, we found that gain also became lower with age. So to examine whether changes in mechanical properties of the muscle tendon unit could mediate some of the changes we saw in soleus responses, I examined mechanical admittance, which is the relationship between the force and the velocity of the probe against the tendon. And we found minimal age-related differences in mechanical properties. So overall, these progressive changes in the characteristics of the soleus response to proprioceptive stimuli is likely not driven by changes in mechanical properties, and also seems to reflect the progressive changes that have been described in the morphology of muscle spindles across the lifespan in animals. And we also found that a combination of age and soleus response amplitude could predict postural sway frequency, but the soleus responses alone had minimal relationships with postural control measures. So overall, I think that insight into how proprioceptive information changes across the lifespan could have some implications for prehabilitation with aging in order to reduce falls. So next, I examined individuals with chronic hemiparetic stroke, and this is showing center of pressure traces under the more affected limb and the less affected limb during quiet standing. So both the frequency and amplitude of adjustments was lower on the affected limb, meaning that that limb is contributing less to the control of balance. And so unlike with aging, we did see differences in mechanical admittance, where the affected side tended to show lower admittance, particularly at these higher frequencies, meaning that the tissue is impeding the motion of the probe more. And these individuals with altered admittance also tended to show larger soleus responses on the affected side. So here there's potentially some relationships between postural control, muscle tendon mechanical properties, and soleus responses to proprioceptive stimuli. So I think that potentially assessing and trying to mitigate some of these frequency-dependent changes in mechanical properties could have some implications for improving postural control post-stroke. Thank you for listening.
Thank you very much. Thanks. So please type away your questions in the Q&A section, but I might get started because I don't work with stroke patients, but I do work with dementia patients. So I was wondering, my mind immediately went and wonder whether you have tried or are thinking of trying it with Parkinson's or other kind of non-healthy aging populations and compare the results.
We've definitely thought about it. The one population that would be really interesting to test is peripheral neuropathy. Parkinson's would also be interesting. I've actually tested one individual with Parkinson's. The problem with the reflex responses is they're quite variable. So with one individual, it's really, really hard to tell. Peripheral neuropathy is like something that would be very interesting because you have this sort of distal to proximal loss of sensory information, and how that really affects spindles is a little bit less clear than how it affects skin receptors in the periphery. So that's something that would be really interesting to test. And did you say, what other population did you mention?
Oh yeah, I work with neurodegenerative patients, so I just thought in general, not healthy aging, so still aging, but with some Yeah, no, it's incredibly interesting. You mentioned the variability. So, oh, sorry, we have questions from, and I'm not going to monopolize. Ruyan Wei is asking why the less effect leg have more movement trajectory?
Yeah. For the center of pressure traces.
Yeah.
Yeah. So the affected side will have less movement. Overall, if you merge the force plates, the individuals that have chronic stroke will actually have more sway altogether if you sum it all up. But when they're standing and you're just looking at each limb independently, they're basically using that affected side more as like more as a passive brace. I mean, it's maybe contributing a little bit to balance control, but I think most of the adjustments are being made.
Okay.
Oh, sorry. I have those. Yeah. So most of the adjustments are being made on the less affected side. Does that make sense?
It does. I think Kami as well has a question.
Yeah, thank you for your presentation. On this slide, I was actually wondering what it means when the affected leg has variance in the admittance compared to the less affected leg in the high frequency. Like, what do the frequencies mean physiologically?
So it's a little bit, I don't have a great answer to that because to me, this is all fairly high frequency. What you could pull out is where you have peaks is where you might have some resonance, which is something that I was trying to look for. And then that resonant frequency should be related to stiffness. So stiffer structure, higher resonant frequency. And so you do see those differences. But overall, I think I'd have to look into this in more detail in terms of both like what the tendon is doing and what the muscle is doing to come up with this pattern when there's sort of like an inflection, sometimes around 50 hertz. Because this is looking at like the combination of the mechanics of the tendon along with the muscle. And there's some evidence that the tendon's actually becoming slightly more compliant while the muscle is actually becoming more rigid. And so how they're really interacting is something that we really need to look into further and potentially with some imaging like ultrasound imaging.
Right. We have a question from Carly Sombrick asking whether you would expect that the stroke deficit would be related to the extent of the gait recovery.
Gait recovery, you said?
Yeah.
Yeah, sorry, the extent of the
extent of, would you expect that the stroke deficit would be related to their extent of gait recovery?
So which deficit, the change in admittance or?
Yeah, this is how I'm interpreting the question.
Yeah, that's a good question. We did do a lot of clinical tests, Fugl-Meyer, Timed Up and Go, and Shadok-McMaster, some other tests. So I like the Timed Up and Go test. I should look into that in a little more detail, but I think definitely there are relationships between like motor control, gait, and the mechanical properties. That seemed to come out fairly strongly. So these individuals with this like sort of blunted admittance at higher frequencies really had like poor measures on tug test, which involves gait as well as transitions, and also like more impaired motor recovery on the Fugl-Meyer as well.
Yeah, I believe that the next question from David Granite is always in this line. So he's wondering whether the changes in admittance in the stroke population are results of differences in tonic activation of the muscle. Like if there's...
I don't think so. I did also record EMG, and EMG tends to be lower on the affected side during standing, even if I try to get them to stand symmetrically with equal forces under both limb. So I think that I think almost like higher muscle activity would stiffen that muscle tendon unit and cause that sort of blunted admittance, but it's actually the opposite. There's lower activity on that affected side.
Right. We have another question on the stroke patients because it's so interesting, the result. Nick Kitchen is saying, well, first of all, thanks for the talk. And wondering how old were the patients and could their age explain some of the differences, some of the deficits you observed?
Yeah, so the beauty of this project was I had 54 healthy adults to age match too. So I have age match controls that are all within 2 years. So there's clearly differences both between affected and less affected side, and also between stroke and control, like the affected side of stroke and healthy controls. And within healthy controls, there's no differences between the right and left leg in any measures.
Nice. Amna Lawrence is kind of asking a following up question on possible alternative populations and wondering whether you think your study would be applicable in patients with multiple sclerosis.
Yeah, I'm actually really interested in that disorder, but potentially some different studies. Like I'm really interested in how that affects transmission through, how MS affects transmission through spindle afferents, because I think potentially there could be some changes in where spindles branch and branch point failure of action potentials through that branching, because muscle spindles branch to contact almost every single motor neuron in the motor pool of the muscle that they've come from. And so I think there could be potentially quite large changes with MS, and that's something I'd love to look at.
Very interesting paradigm that it sounds like you're going to have to keep on working on this and applying to numerous different populations. So thank you very much, Robin. Keep an eye on the Q&A because people might still write you something and you will be able to type down your answer.
Okay.
Meanwhile, we are going to switch to our second speaker. Maria Ayala, a PhD student at the Center of Vision of York University, led by Dennis Enriquez. And she's going to show us how efferent and afferent estimates of end location do not optimally integrate. Very curious to see your result, Maria. Please take it away.
Awesome. Thanks for the intro. So today, like what Valentina said, I'll be presenting on efferent and afferent estimates and how they don't, we saw that they don't optimally integrate. So our central nervous system can rely on at least two sources of information in order to localize the limb position in the absence of vision. So first we have our proprioceptive afferents from receptors in our limbs. And we also have an efference copy of the motor command used by the cerebellum to generate a prediction of sensory consequences. To produce a reliable single estimate about the location of the limb, these two sources of information could be optimally integrated using Bayesian integration or maximum likelihood estimation. And while some work has been done to investigate whether the CNS optimally combines vision and proprioceptive cues, less is known for the integration of non-sensory signals for limb estimates, such as these efferent signals, given the difficulty of isolating them from the ever-present proprioceptive afferent signals. So here we use our new paradigm to isolate efferent-based estimates of hand position from proprioception, and we had over 220 healthy participants, both younger and older adults over the age of 55, and we had them localize their hand after actively reaching in a self-chosen direction, and we call this our active localization condition, or after being passively moved by robotic manipulandum, and we call this our passive localization condition. So here's our experimental setup. We had participants localize their unseen right hand using their seen left hand, and they viewed along this horizontal plane. We had participants do this, localize their limb along this arc right here, so you can see on the bottom right panel. We had them do these active localization conditions after reaching within a line cursor, And we also had them do that after training with a perturbed cursor. So our first question was whether estimates of hand location based on both efferent and afferent signals, so that's our active localization condition, are more precise than those based only on proprioceptive information. So that's our passive localization condition. According to the Emily rule or other straightforward Bayesian mechanisms, variance should be lower in the condition, in the localization condition where the two signals are combined. So that's our x-axis here, the active localization condition. And it should be higher when you only have a single source of information, which is our passive localization that only has proprioception. But we found no evidence in support of this. So here you'll see variance in passive localization over variance in active localization. And although there are some younger cohort members who could be showing optimal integration, you'll see in the light orange colors, the majority of them sort of just fall along this identity line. When we look at our older participants, there are some that do show that, but they, again, more or less fall along the identity line, which suggests to us that one or two signals are about equally reliable. Our second question is whether older adults display greater proprioceptive variability than younger adults. Here you'll see the probability density function for the active localization condition and the passive localization conditions. And in the warmer hues, you'll see the younger participants and the older participants in the cooler hues. The dashed lines represent the variances for each localization condition. And as you can see, there seems to not be an effect of age on reliability of localization. In our final question, we looked at whether weaker proprioceptive priors led to greater learning-induced shifts in hand localization, as would be predicted by Bayesian inference. So here on the X, you'll see, on the Y, you'll see post-training localization shifts over pre-training localization variances. So here you'll see active localization variability, and on the side, the passive localization variability. And as you can see, we don't see a relationship. It's also something that we see with our older participants. And as you can see, we don't find that noisier, highly variable estimates were more susceptible to larger proprioceptive recalibration following adaptation to a perturbation. And this suggests to us that Bayesian explanations about integration of afferent and efferent signals are not as likely here. Together, these findings make it less likely that the CNS combines efferent-based predicted sensory consequences and proprioceptive afferents optimally, at least when estimating the final limb position. Right, so that's the end of it.
Thank you very much. I'm gonna fill the time that people take to type down their questions. Asking you for a clarification on your groups, the younger and older, what's the age range we're talking about, and whether you have tried a more continuous effect of age instead of binarizing?
So we haven't done that. Our younger participants are between the age of 18 to 35. Older are around 55 to around mid-70s.
Okay. So yeah, coming from dementia research, I still call them like adults, older adults. They're not that old for me, but yeah, it makes sense. And so yeah, you said you haven't tried a more continuous age effect, possible correlation with. But yeah, I agree that it doesn't seem to be there. Looking at the graphs. If any of the other speakers want to unmute and ask questions, just motion me. Yeah, don't be shy. I was also wondering how you, because I know these presentations are so fast, if you could elaborate a bit more on the motor area variability and whether that could explain the variability that you see in localization.
Okay, so I've got a slide for this, just in case. Right, so here you'll see on the X is motor area variability, so just variability of baseline reaches, and on the Y you'll see localization variability, and we don't see a relationship for either passive localization or the active localization condition. So motor noise doesn't seem to explain localization variability.
What are you planning on doing next? So if we're not using Bayesian, what are we doing to establish how to move our hands?
Yeah, it's unclear, but please follow us on Twitter, and you'll see like our next publications on this.
You're planning on using the same setting, but with other populations, as we were mentioning in the previous talk, or to tweak your paradigm to dive more into possible explanations with the effect?
Yeah, I don't think like, oh, sorry, I'm just gonna stop sharing.
No problem, yeah.
Yeah, yeah, I don't think we have plans to look into like more like specific populations. Yeah.
But maybe you can make like the task harder or change the paradigm.
Possibly, yeah.
If none of the other panelists, I don't want to monopolize the stage, but if there are no further questions, we can move to the next speaker. Our third and final speaker for the session. I see that she's already on screen, ready to jump in. Kami Shioza, from the grad student supervised by Prof. Neville Ogden at the Eric and Evelyn Newman Laboratory of Biomechanics and Rehabilitation at MIT. And her talk is titled Frequency-Dependent Force Direction Elucidates Neural Control of Balance. Thank you very much, Kami.
Okay, thank you for the introduction. I will share my screen. Is the screen visible? Okay, great. Okay, so human balance is an unstable task that requires the control of translational and rotational acceleration. In general, we use foot-ground reaction forces to manage these accelerations. Previous work measured balancing humans and identified a point of intersection of foot-ground interaction forces that exhibited consistent frequency-dependent behavior across multiple subjects. Because the height of this intersection point relative to the center of mass dictates the relative directions of translational and angular accelerations, the intersection point provides a compact geometric representation useful in understanding the dynamics and control of human balance. To test whether the intersection point's frequency dependence observed in humans was a distinctive signature of neural control or was a necessary consequence of biomechanics, this study simulated quiet standing and compared the results with human subject data. Separating the neural contribution from biomechanical factors by only observing human motion is challenging. So to simulate balance, we estimated the human body as a double inverted pendulum that is controlled by an LQR controller. A key feature of LQR control is that a cost function is minimized and the result always yields a stable controller. The parameters of interest were alpha, which penalizes the relative cost on the state deviation versus control input, and beta, which penalizes the relative cost on the control input in the ankle and the hip joints. We added noise to the hip and ankle joints to replicate the variability observed in human quiet standing. So alpha, beta, and sigma r were the parameters we varied to match the simulation data to human data. Before we get into the results, I will define the intersection point that I've been mentioning. If estimating the ankle as a pivot, there is a ground reaction force acting there along with the ankle torque. This is equivalent to a ground reaction force acting at the center of pressure, which is some distance away from the pivot. Assuming that there are two net force vectors in time that are defined by their direction and point of application, we define the intersection point as the height of the position or the extension of the two force vectors crossed. With a little bit of math, we can the intersection point relates to the center of mass, center of pressure, and the orientation of the ground reaction force in this way. The center of pressure and theta F were bandpass filtered into 38 frequency bands to find the intersection point at each band. 40 trials for each varied parameter were run to allow for statistical analysis of the results. This is a simulation data that best replicated the human subject data overlaid on top of the human data. Before getting into the best fit parameters that I found, we saw that for all stabilizing controllers, the intersection point followed a trend where it was above the center of mass at low frequencies and below the center of mass at high frequencies. Because changing the controller parameters didn't alter this general trend, it most likely signifies a necessary contribution of biomechanics. However, the frequency at which the intersection point crosses the center of mass, denoted by this value 1 in the y-axis here, or the high frequency asymptote of the intersection point varied across different tested parameters. For example, varying alpha affected the frequency at which the intersection point crossed the center of mass. A large value of alpha, such as 10 to the 6th or 10 to the 4th, minimized the control input required to stabilize the inverted pendulum and resulted in a close replication of human subject data. This makes physiological sense because humans likely minimize control effort during quiet standing. Varying beta also affected the frequency at which the intersection point crossed the center of mass. Beta is less than 1, where there was more penalty in the hip torque, resulted in close replication of human data. This also makes physiological sense because during quiet standing, humans primarily use ankle torque to balance. Lastly, varying the noise ratio affected the high frequency asymptote. The noise ratio that was slightly less than 1, 0.9, where there is more actuation noise in the hip, resulted in close replication of human subject data. So in conclusion, we successfully distinguished the biomechanical constraint where the intersection point declined with increasing frequency from the neural strategy where the humans tend to minimize control and penalize the use of the hip torque more than the ankle torque. This method that I just presented can be extended to different experimental conditions such as sagittal and frontal plane balance with and without assistive devices and in aged and impaired populations to identify differences in controller characteristics employed by humans. Thank you.
Thank you. Thank you very much. Really clear, even though I know nothing about it. So I'm probably going to have very naive, silly questions. So if anyone else wants to jump in with something more. Also, it was very ironic to try and listen while I'm trying to balance myself and clearly doing a terrible job. But my first very naive question is, why did you choose as a model for the human, the double inverted pendulum? Like it's something that is known in the literature that is the ideal model?
Yeah, great question. So actually in balance, some people use a single inverted pendulum as well. And having, of course, the single inverted pendulum is more simple. It simplifies the whole model more, but we've actually found that the single inverted pendulum could not reproduce this. Oh, can you see my cursor?
Yes.
Okay, yeah, so it could not reproduce this intersection point frequency dependence where it goes below this dashed line, the center of mass. At high frequencies, it actually converged at above the center of mass at high frequencies. And we just solved that mathematically. And so the single inverted pendulum was kind of out of the question. And so the next simplest model in that case was the double inverted pendulum.
I see. This makes sense. And so the fit with the human data is impressive. Could you remind us how was the human data collected and what's the sample we are talking about? And that's potentially how you might want to compare to other samples, but in this case.
Yeah, so I actually didn't collect the human data. It was done by researchers at University of Wisconsin-Madison. But the human subjects were healthy, young subjects, I think at under 30, and there were 10 subjects, and they were standing without any external perturbations on a force plate to measure the force vectors and find the intersection point as shown here.
Right. And so you were mentioning that a possible development of this project would be to try and apply this model to other, like other population. Seems to be the main topic of this session.
Yeah, I think I'm quite interested in developing assistive technologies for impaired populations, and I think understanding the neural control and the dynamics of impaired or just people in general during balance is an important first step to that. So I think that if we could apply this method and find neural control strategies for people who use assistive devices or aged and impaired populations, that could inform the design of future assistive and rehabilitative technologies.
Could you perhaps elaborate a bit on this minimal control solution? Like, what do you exactly mean, and how do you ensure that it's actually been met, like that this solution has been met?
Yes, so here, what Valerie's talking about is this minimal control solution that I found to be one of the best fit parameters. And this happens when alpha is greater than 10 to the fourth. I actually have a maybe useful slide here. So this kind of gets into control theory a little bit, but the essence is that basically when you're stabilizing a double inverted pendulum, it's inherently unstable because it will fall with gravity. So you're trying to move these open loop unstable poles to the left side of this dashed line here. And then when you apply a minimal control solution where alpha is a fairly large number, such as 10 to the 6 here, then we see that the poles get directly mirrored across this dashed line. So the red crosses are directly above the black crosses here. However, as alpha starts to decrease and it's no longer in the minimal control region, then you see that the poles start to deviate away from the mirrored position of the poles, and that's how we ensure that we are that the minimal control solution yields results similar to human subject data, and how we inferred that humans probably employ a control strategy that minimizes their control effort.
I see. I see. I don't want to force you to stop sharing, but if any one of the other speakers and the other panelists wants to come back on screen and ask questions, they're welcome to do so, as well as if any of the attendees want to type or come on stage to ask their questions. I know I keep bugging you about your model, and you explained very well the pendulum. I'm convinced, but why a linear controller? Is that justified? Or could you? Yeah.
So I used the linear controller because in quiet standing, which the human subjects were not subjected to any external perturbations. So in quiet standing, humans only deviate about a very small angle about the center position, about the upright position. And so I think the linearization is justified. Although I used a nonlinear plant here, and then just applied a linear controller here. And we also saw that when simulating, the center of mass did not change its height very much. So that also says to how much the pendulum deviates about the upright position. It doesn't really deviate about the upright position, basically.
Okay, so just for me, the 20 friends we have here and the 100 people that are following on YouTube, we are going to keep it quiet. But are you happy with this model, or if there's anything you would like to tweak? That you haven't tweaked yet, and it's going to be your next.
Yeah, I think right now we've only added actuation noise to the joints. There's much more complex sensory noise, measurement noise, things like that in actual human data. So those could be some next steps, some ideas for next steps.
Sounds very exciting. I also know I would be a terrible subject because, as you can tell, I'm unable to stay still. But yeah, so I have definitely learned a lot from these 3 talks and amazing session. If anyone has any last questions or any last comment. Oh, we do have one last question. I did well in asking. Reza is asking, what would happen if you have signal-dependent noise?
Signal-dependent noise.
And by the way, Reza, if you want to talk, if you want to explain your questions live, we can even bring you. Sure. Okay. Let me get.
Okay, great. Yeah.
Hi, Kami.
Hi.
Thanks for the good talk. I was just wondering if any of these intersection point heights would change if you have a noise value that scales with the size of the torque. That's what I meant by signal-dependent noise. Because in the motor control literature, usually people argue that the neuromuscular noise increases by increasing the force.
Yeah, exactly. So actually, what troubled me the most, the result that troubled me the most is that here, I found that sigma R is less than 1, where there's more noise in the hip, resulted in the best fit parameter for the noise ratio. And that was a little counterintuitive to me and counterintuitive to the previous work that you mentioned, Reza, because I found from data that humans likely use the ankle more than the hip. And so it didn't really make sense that there was more noise in the hip, even though we were using the ankle more. So I think that, like I said before, I think that could be a result of just simulating using actuation noise here. So perhaps I think that's why an important next step could be to add sensory noise to account for all sorts of different noise inputs.
Right. We live in a much more complex world than than any model can understand yet. I'm sure we're going to get there.
Yeah, but I think that simplifying to glean some insight is important, even though it can't capture all of the things.
Absolutely, and I loved how you explained that you try to model like the simplest model, and then if that doesn't work, you can add a piece and make it. You make it complicated as you need it, not more than.
Yeah, exactly.
Well, unless anyone else has a very last question, I'm gonna thank again our 3 amazing speakers and all of you for attending. I know there's a bit of a break between one session and the others, but I keep enjoying your match, and see you all next time. Thank you.
Excellent. People are coming into the room. That is good. Come on inside. There's plenty of space. It's a little bit like the TARDIS. It might look small on the outside, but there's almost infinite capacity inside. I don't actually know what the capacity is for a Zoom webinar, but I'm pretty sure we're not going to hit that capacity today. So plenty of space, not a problem. All right, while we're waiting for any more that might be popping inside. My name is Ulrich Bayerholm. I am an assistant professor in Durham in the north of England, and I will have the pleasure of being the MC for the session today. I'm going to do my best to actually talk as little as possible, so there's more time for excellent speakers for today. We have 3 exciting talks, all in the realm of computational neuroscience, so that should be a lot of fun. So we might as well go ahead straight away with our first speaker, who is Matt Getz.
Matt is a grad student at University of Chicago and at University of Pittsburgh. Somehow at both places at the same time, must be confusing. And he is going to talk about a circuit-based theory of the impact of cortical state on information flow. So if you can get your slides ready, Matt.
All right, great. Can everybody hear me okay?
That's great. Sounds great.
Perfect. All right, so I'll jump right in. And so the motivation for the study is that changes in cortical state are related to changes in behavior. So for example, the hit rate of a rodent performing a sound detection task, shown by this green line, varies as a function of its arousal state. Similarly, with attention, the performance of an animal performing a discrimination task varies as a function of its attentional state, as shown by the variance in these three lines. But importantly to neuroscientists, right, these state changes are coincident with changes in circuit activity. And so, for example, in arousal, one might see a change in the response gain. And with attention, one might see a change in the correlations. And so taken together, this anticipates how we expect neural processing metrics to change. And in particular, the metric that we're interested in here is the linear Fisher information, which is a measure of the smallest change in a stimulus that can be reliably discriminated by an optimal linear decoder. And you might be familiar with it with this form here, where f prime is the response gain and sigma is the response covariance. And while some fancy nonlinear decoder might do a better job, we like this because it also has a clear biological instantiation through synaptic weights. And so the question we're after is how cortical state affects information flow. And so we need to set up some assumptions and basis for building our model. And the first thing is that state changes can be local or global. So we're considering things like feedback drive and neuromodulation. But axiomatically, we're going to assume that state changes only affect circuit activity independent of a stimulus or some external inputs. So if we consider a nonlinear EI rate model, where we have EI pairs oriented around a ring, where position on that ring corresponds to a unit's preferred value of a tuning variable theta, then we can introduce a modulation as either a uniform current drive or a change in the conductivity strength. And so if we simulate this model, we see that in the unmodulated regime, we might have a response profile for the excitatory units that looks like this. And with modulation, in this case, we're seeing a reduction in the rates. And we can go ahead and calculate the gain. And we see a reduction in this case, and the covariance. And again, we see a reduction with the modulation. And now if these changes were unique to some state change influencing a behavior, we would expect information to reflect that. And so recall the definition of Fisher information we're using. And of course, these are the two metrics that factor into this equation. Of course, both are changing quite dramatically, and yet Fisher information does not. And so it turns out that this is true in general. And so the first thing we want to do is understand why. And to derive some intuition, we're going to go to a reduced model. So we're just going to simplify the network down to a single EI pair. And it's important to note that we're linearizing around the steady state solution of a nonlinear rate model. And so in particular, given a stimulus, a neuron, or if you want to think about it as a mean field, isotuned population is responding variably about some mean rate. And the only way to perturb away from that is to introduce a modulus. So noise is not so big that we're perturbing off of our operating point. And so now the EI population is defining a joint probability distribution over the rates. And one can visualize that here with the 95% confidence intervals over the rates responding to a stimulus S and a perturbation of that stimulus delta S. And now modulation can change the shape or the position of these distributions. So to example moduli, say, might rotate these distributions or rescale them. But yet, by the linearity of our system and decoder, the overlap or the error, right, of a discriminator is not changing through each of these states. And this sort of gets at this invariance. But yet, this picture might be a little disconcerting because what does our decoder mean if we're reading out from both E and I? We know that E cells are the primary projection units along the cortical hierarchy. And so if our decoder is representing a downstream region, then really the picture should look more like this. And in fact, this corresponds to a projection of these distributions down onto the excitatory axis. And now we see that the distributions over the excitatory rates can change as a function of our modulation. In this case, the error is going down. And so now we can see an improvement in information. And since our decoder is restricted to a subset of the population, we're going to call this a subpopulation code. And so we see that FI can change if we consider subpopulation codes, but now we're interested in addressing how can FI change. And so to that, we're going to parameterize our network. And we're basically taking the same model, but now we're writing it down with external parameters that feed into the network and network parameters, which define how the network responds. And so again, we'll look at Fisher information. And if we write it down for the whole system and work through the algebra, we see that FI only depends on the external parameters, which are not allowed to change with modulation. And so this is why, in general, FI is going to be invariant. But if we formally define our subpopulation code restricted to E as FIE, then now when we write the expression as a function of these network parameters, we see that the dependence on network parameters is maintained, but the dependence is through some function x. And so x is given by this expression here. And what you notice is that x is only composed of parameters that are highlighted in blue here, and those correspond to the inhibitory population, despite the fact that E is what's being decoded from. And as a result of this, we call x the normalized, or x really is the normalized projection weight from I to E. And so we call x the effective coupling. Now, if we split the input parameters into those which act on E and those which act on I, then the subpopulation codes can become effectively feed forward in terms of x, and we can take this complicated recurrent network and rewrite it like this. And so now to develop a little bit of an appreciation for what x is doing, first, we're just going to note that x has reduced the effective parameter space. So we went from having to consider 6 different network parameters down to 1 hyperparameter. And just to develop our intuition for what the effective x, we're going to consider the effective variant inputs to I. So we're going to change the correlations with E and a stimulus. So start with this picture here where I is only receiving noise. Then we can write down FIE as a function of x and the input noise correlations. And we see right away that FIE is maximized for some high value of noise correlations and non-zero value of x. And the reason for this is that if the input noise is highly correlated between E and I is just feeding that forward to E with a minus sign. And so if these noise sources are highly correlated, then tuning x can effectively cancel that correlation with E, therefore optimizing information. For that reason, we call this the correlation cancellation regime. But by contrast, if I now receives a signal, then this picture changes quite a bit, and we see that x is maximized at low values. And this makes sense because now I is effectively canceling signal. And so the optimal thing to do is to shut down I. Right? And so for that reason, we call this the gain reduction regime. But the second case might make you a little uncomfortable because we know that I also plays a stabilizing role. And so if we fix the parameters in our network with some exemplar, then we can draw the instability boundaries on our network. And these are going to depend on the full network. So X is not getting away from this. We still need to fully consider the stability of our network in order to appreciate the bounds on X as well. So the next question, or really just the last question, because I spoke quite rapidly, is that how will these subpopulation codes generalize? So we've gone through this in just a singular EI network, but we know that the brain is a lot higher dimensional than that. And the short answer is well. So if we look back at the original ring model, then the one-dimensional X that we described earlier has a natural extension to n dimensions, and it has the matrix analogous form. But it's important to note that subpopulation codes are not unique to an EI partitioning. So we motivated this by thinking about the excitatory propagation of information across cortex in a picture that resembles this. But the truth is that cortex is a lot more complicated than that. And so you might have sets of recurrently connected excitatory units, only a subset of which project to some downstream region. And so in this case, the picture looks a little bit more like this. And our theory extends to this case as well, where X, rather than only depending on I parameters, is going to depend on the unobserved network parameters. And so to conclude, what we've seen is that in a linearized framework, the total information flow is invariant to state changes, and subpopulation codes better reflect anatomy and allow for information increases with state change. the caveat is that one must know the downstream projection profiles of certain cells to appreciate how changes in activity affect information flow. And so with that, I just want to thank my co-authors on this project, Brent Duan, Chen Huang, as well as everybody who's helped out. And we do have a working draft of this. It's going through final edits. And so if you want to find out when we post it or get a copy, just shoot me a quick email. And thank you for your attention.
Thank you, Matt. You can all hear me clapping, but I'm sure there are a lot of other people elsewhere who are doing it at the same time too. So you just have to imagine those two. Brilliant. Thank you very much for that. Maybe I'll start us off with just a single question. So you motivated this as being, as an example, for example, with attention being something that causes a state change. Is that kind of the kind of paradigm that you think of specifically with this? And if so, are there any specific brain areas that would be more relevant for? Or?
Yeah, so I think broadly, I'm thinking about anything that causes, a perturbation in the set point of a cortical circuit. So it could be pretty much anywhere. I do think about this in sensory systems, mainly because that's where a lot of this this data comes from, say, in arousal studies or attentional studies. But I think it's pretty flexible. And so that last slide, for example, came from a motor area.
Right. Sure.
With the pathway splitting.
Yeah. Right. So there's a question from the Q&A. Raina is asking, interesting talk. I was wondering how this changed in a nonlinear regime, or would you consider full mutual information instead of Fisher information? That's interesting.
I'm not actually sure that I got the, what they mean by full nonlinear. Oh, you mean like not using linear Fisher?
Yeah, so if you're not using Fisher.
Yeah, that's a good question, and I actually haven't worked out the math for that.
I think we're expecting you to have worked out all, so don't feel bad about that. All right. Any other thoughts on that though, or yeah? Should we leave it as for future research?
Yeah, I mean, I think it's hard to say, right? So it's not that I'm being lazy by using linear regimes. It's just that if you think about cortex as asynchronous, or if you think about things like tuning curves, those come from operating point sort of solutions that are linearizable generally, because we don't think that noise in the brain is so big that we can't compute a tuning curve. And so in that context, I think that a lot of these assumptions are quite reasonable, actually. But I do, so thinking about like mutual information, I know that, there is quite a bit of literature. I think Nicolas Brunel, for example, back in the 90s was talking about the relationship between Fisher information and mutual information. And so I might refer to a project like that to gain some insight into that question. But generally, Fisher information is sort of the small delta limit, I guess, of discriminability, for example, whereas...
If I can just cut you off, I think Rainer might want a follow-up question on that. Okay. I don't know whether we can put him through a microphone just really quickly.
Yeah, sure.
Hey, thanks so much. Really cool talk. Yeah, just to clarify, it was basically two sub-questions. One is, what happens if your input signal is so strongly modulated that this linearity assumption doesn't work anymore? And secondly is, what happens if you don't consider the just Fisher information, but like full mutual information? And like, would you then get a lower bound or an upper bound, or can you still say something about that regime, like both numerically, or maybe is there like an analytical approximation?
I see. Okay, so the first question is, yeah, this framework would break down, and I don't know the answer to that. As far as full mutual information, yeah, I don't see why it would be a problem. I think that this would still have relevance. But again, this is something that,
no, it totally has relevance. I was just wondering, like, whether you then overestimate information or underestimate, like, what direction you basically. I don't have a good intuition for that, like when it's nonlinear.
Right. Likewise.
Cool. Yeah, maybe we can chat more about it.
We'll file that one under future research. Okay?
Thanks so much again. Really cool work. Anyway.
Brilliant.
Thanks. Yeah.
Thanks, Matt. Okay. With that, let's move over to our second speaker. Thanks. Second speaker is Dilip George, who is the CTO and co-founder of Vicarious AI, working on research and development of general intelligence layers for robotics. And he's going to talk about a detailed computational model of thalamic and cortical microcircuits based on inference in a generative vision model. So please go ahead.
Thank you, Ulrich, and it's great to be here. Thanks to the organizers for the opportunity to present. This is going to be a very different kind of talk. It's not really a circuit level talk, and it's mostly going to be an infomercial for this preprint that we have on bioRxiv. I'll just give you the overall motivation and some key results from that paper. So the question that we are after is, how can we understand visual cortical circuits? By understand, we mean we should be able to answer all these questions. What is the function of a cortical column? What computations are performed in different laminae? What is the computational role of clonal neurons? Which connections in a cortical column are innate? Why are feedback connections there? What's the computational role of thalamus? All these things that we are interested in, and we should be able to assign precise roles for each of them. That's our goal. Now, of course, you can bottom-up study the circuits. They provide very important clues in neurophysiology and modeling, etc. But the computational model will be too local. It will be very hard to connect it to the overall behavior of vision, recognizing objects, parsing, etc. You can do connectomes, but connectomes themselves are not very informative. You cannot just hope to simulate a connectome and get computational insights out of it. The connectome is not going to start working like the visual cortex. So how do you attack this problem? So we have a different methodology. We call it the triangulation strategy. The trick is to look at the brain, the world, and computation at the same time. And the brain is cognitive science and neuroscience is giving us a lot of insights about how the brain is organized, what the brain does, what are some of the organizational principles. And if you can connect it to the organization of the world and connect it to real world test sets, then you're finding something informative there. And of course, those have to be formulated in terms of computational and algorithmic models. And you should be able to come to reasons on why these structures exist from an algorithmic angle. And when you can connect all three vertices of the triangle for an organizational feature in the brain, that's when you have found something useful algorithm-wise. So if you, for example, if you ignore the world vertex, then you don't address the overall task of vision in solving the problem. If you ignore the brain vertex, that is just pure machine learning and statistics. You don't take any insights from the brain, and of course you cannot tell anything about the brain. And if you ignore the computational vertex, then you can make local things, but it's very hard to build large models. So the first step in our approach was to take insights from the brain and build a computational model, an algorithmic model, so that model can stand on its own. Although it uses insights from the brain, it is purely specified as a computational and algorithmic model. And this is a generative vision model that we published in Science in 2017. It got a good amount of attention because this was also used to break CAPTCHAs, fundamentally break the difference of text-based CAPTCHAs using this generative model. And it is much more data efficient than deep neural networks. And one key differentiator is that it uses feedback and lateral connections to explain a scene of characters rather than just doing pattern recognition feed forward. In fact, we have lots of details on what were the computational feature, how it is connected to representation in RCN, and what is the computational algorithmic reason for that. For example, I'm just taking one row from here, the top row. Blobs and interblobs is an organizational feature that we see in the visual cortex, and it is connected to the algorithmic principle of separately representing contours and surfaces, and that is suitable for representing natural signals. Just to give you a flavor of how we think about these ideas. Now this paper, this preprint that I talked about, is working backward from there. We are basically take this computational model, can we find out how it can be implemented in biology? And of course, then it will be filling in a lot of details that biology did not predict. You will be using a lot of biological data in coming up with that instantiation, but
but it will also be predicting a lot of the details because the computational model is kind of complete, and it will fill in the details that biology did not predict. So that is this preprint that we put on, so I can just give you an illustration of how this works. The computational model is a graphical model. It's a probabilistic graphical model, which can be also represented as a factor graph. And then from that factor graph, we are deriving a neural implementation. And even for a simple graphical model, so here in A, you have a very simple graphical model, which is features ABC connecting to pixels. It's basically saying these features represent, these lines generate these pixels. And it is represented in the factor graph form in panel B. Well, keep in mind that this is a graphical model. That means every link is a bidirectional interaction. So that means if you want to implement this in biology, you need a lot more neurons than a node is expanded into multiple neurons because they have to communicate messages in one direction and in the other direction. And you have to also compute the belief of that node. What does that node believe in? Should I be on or off? That has to be represented in a separate neuron, which is different from the neuron that is receiving messages. So, and then even this simple circuit can produce a lot of, so this is on panel E, you see the implementation of this circuit, and it can produce a lot of inhibition, excitation, effects based on context, very nonlinear interactions. But we are not hard coding in that nonlinear interactions. It is coming from the factor graph. But it can produce many of these gating mechanisms we see in the brain. This is, for example, maps to some of the gating mechanisms in thalamus. Of course, the factor graph for RCN is much more complex. It has some of the properties of a convolutional neural networks, but it is generative and it has more things. For example, it also runs in the backward direction. So it has features, ladders, and pools. But keep in mind that all these connections are bidirectional. So the interactions are both ways. So when you map that to cortical circuit, it will, and of course, each of these factors have their own equations associated with them. And when you map that to cortical circuits, this is what it looks like based on our mapping. Of course, this is using information from biology to figure out how this maps onto cortical laminae and columns. And one key thing to keep in mind is that the circuit looks like this. You kind of need two copies of that circuit because you need to have feed-forward messages and feedback messages, and their beliefs to be combined. And similarly, lateral messages also need to keep two copies. And some portion of this mapping, when two levels interact, when V1 interacts with V2, there has to be some explaining away computation between them. By explaining away, I mean when a pixel is being claimed by two higher level features, you need to have competition between them. This happens naturally in a probabilistic graphical model, and this competition can be thought of as gating. For example, if attention turns on one feature, you need to turn off the other feature. So that gating function, which naturally happens in the probabilistic graphical model, when we looked at the anatomy and physiology, the only way it can fit naturally is in the thalamus, and it predicts the circuits of the thalamus to a striking degree, which was pretty interesting to see for us. And one conceptual thing that you might want to take away from this whole thing is that you can think of a microcolumn as a random variable, as a feature, for example, and the column itself is representing this feature, but the different laminae in the column are representing different aspects of this feature. For example, how does this feature participate in a higher level feature? How does that feature participate in different lateral contexts? How does it feature participate or unfold into lower level components? And finally, how do you combine information from feed forward, feedback, and lateral to produce the belief of the microcolumn? And here's what I mentioned about the thalamus circuits. If you study Sherman and Guillory papers, you know that there is a feed-forward pathway from different regions that goes through the thalamus with gating involved, and this is very much predicted by our mapping from RCN to cortical circuits, including to the details of the gating circuit and the dynamics which I showed earlier, and also predicts interactions between blobs and interblobs. And another thing that you can do is you can actually do virtual neurophysiology, because now what you have is a functional in a circuit which does recognition, segmentation, etc. And so you can actually do virtual neurophysiology. So you can simulate things like subjective contour effects, where you're given these illusions but you see contours where it doesn't exist. For example, all the magenta portions of these contours are hallucinated by your brain and also by our system. And you can also study the dynamics of that on, which layer of the cortex will turn on and turn off at what time during the inference of this subjective contour effect. And you can also do that for things that have a combination of subjective contour effect and surface models. For example, neon color spreading, where the boundaries are hallucinated and also the color is hallucinated. You can, in our system, for example, replicate many of the things that are observed in biology, where the feedback computation first settles for the contours, and then the contours kind of act as boundary for filling in the surface information. And that is why the color settles later, and the outer color kind of bleeds into the inner segments. So that's basically what I wanted to convey. That's just a very brief summary of a pretty lengthy and detailed paper. You can find out more about our work at vicarious.com, and you can always follow me at Dilip Learning on Twitter to get more updates and more information. Thank you for listening.
Thank you. That's some very exciting stuff, definitely. I think there's a lot of information there that's very hard to press into a 12-minute talk, but I appreciate that you tried, definitely. Let's see, there's a question in the Q&A from Gonzalo saying, very nice talk. Does the implementation of learning in the model in neural circuits require plasticity at all synapses, or is plasticity restricted to few layers and locations?
Yes, and in fact, very good question. That is another prediction from the model. So all the synapses are not equally plastic. In fact, some synapses are not plastic at all. And one thing to take away from it is that most of the vertical connections within a column can be pre-established, except maybe connections to layer 4, which comes from the other region, that cannot be pre-established. But many other connections within a cortical column, the vertical interlaminar connections, can be pre-established and just programmed genetically in. And a lot of the synaptic plasticity is on the lateral connections, the inter-column connections, and that's predicted by the model.
So how does the plasticity, is that somewhat close to realistic plasticity, or?
We don't make strong statements about that. We haven't studied that in detail. So yeah, we don't worry too much about the biological plausibility of the learning algorithms. But this one is looking at mostly from the inference side, but it can make predictions about, okay, these are the connections that need to be plastic.
Okay. So a follow-up question from Gonzalo. He asks, so that should come out in connectomes, right?
Well, depends on whether you are modeling the connectome of an infant, or whether it's in the fetus or later, because some of these connections get established as part of development, right? So in a fully developed cortex, you might not see a difference in a static connectome. You might have to see the variation in the connectome with time.
Okay. We also have a question from Joseph. He says, very good talk. I would like to know if this predicts saccade suppression.
No, not this one, because currently the model, the computational model, is about recognizing a single snapshot. It can deal with a scene, parsing a scene, but the computational model does not do anything which includes saccades. Although we have internally worked on a computational model that includes saccades, etc., but that we haven't published that computational model or try to map that to biology. But we are working in the forward direction, where we are taking insights from biology and building the computational model. That's a state we are in. And once we are done with that, we'll of course come back. But it'll be another 2 years.
We're looking forward to you coming back in 2 years and telling us about that.
That's
brilliant. Thank you very much, Dave. I think we're going to move on to then the next speaker. So Arish, if you can get ready to share your slides, please. Arish, are you with us? Oh, something's happening. Oh, good.
All right. Can you guys hear me?
We can hear you, but we can only see a background, Windows background.
Yeah, just give me one moment.
Okay.
Excellent.
Great. So our final speaker is Arish Alveya, who's going to talk about constrained brain volume explains fraction of excitatory and inhibitory neurons in sensory cortex. So please go ahead.
Thank you. So first off, sorry that my video is not visible. It appears there's an issue with my camera that I wasn't aware of. But hi, everyone. My name is Arish. I'm a graduate student at CMU. And today I'm pretty excited to share with you the results of a theoretical study done with, sorry, I'm having some odd issues, done with Ilya Nemenman and Chris Rosell at Georgia Tech that provides an insight into the relationship between neural coding and a structural property of sensory cortex, which is the fraction of excitatory and inhibitory neurons. So as motivation, let us consider how the anatomy of mammalian sensory cortices varies across species and sensory regions, like the primary auditory, visual, or somatosensory cortices. The number of neurons in these areas vary by orders of magnitude. But in comparison, the ratio of excitatory to inhibitory neurons only varies by about a factor of 3, with mice being on the higher end with fewer inhibitory neurons as compared to cats and monkeys who are comparable to each other and have more inhibitory neurons. This begs questions like, why should E/I ratios in such a narrow range be in such a narrow range? And why are the ranges we see the way they are? And how do they relate to sensory coding? And perhaps these are all part of a broader and more general question, which is, can structural properties like E/I ratios that are relatively conserved across species and brain areas be understood in the context of circuit computation? And in the work I'll present today, we explore this global question in a local context of sensory coding, specifically vision, using computational models. So in this study, we use a sparse coding model of vision, which encodes stimuli with excitatory neural activity. This model is grounded in the efficient coding hypothesis put forth by Horace Barlow in 1961. And Barlow argued that the brain must seek a parsimonious representation of sensory inputs because it is tightly constrained by the metabolic energy available, metabolic energy budget, if you will. And sparse coding models of the visual cortex sort of fall into a category of models which sort of abide by the efficient coding hypothesis. They've been around since at least 1996, and they're considered by many to be a faithful representation of V1 because they exhibit many experimentally observed effects. For our purposes in today's talk, their most useful trait is that we can implement them in a recurrent network that can be approximated with a given number of inhibitory interneurons. For a model with a given number of excitatory neurons, this allows us to control the ratio between excitatory and inhibitory neurons. It's also interesting to note that increases in the number of either type of a neuron or both have also been shown to improve stimulus representation in these models. So I'm going to take a brief look with you at the computational loss function for sparse coding. On the right-hand side, it reveals a competing fidelity and sparsity term. These terms are balanced against each other by the sparsity parameter lambda, which, when increased, forces the model to produce a sparser neural code. We'll look at the effects of changing sparsity levels as well as E/I ratios in our results. Although we said that adding neurons may improve stimulus representation, it is important to recognize that cortical volume is a very real and important biological constraint. The number of neurons of either type cannot increase endlessly. We incorporate this volume constraint by fixing the total number of neurons for all models that are used in the study to be a particular number, which is in this case, n is 1200 neurons. And using the recurrent network approximation that I mentioned earlier, we can then vary our structural property of interest, which is the E to I ratio, in a systematic manner, while we still adhere to this volume constraint. And we evaluate how changes to the E to I ratio affect a network's performance to better understand what the relationship is between this structural parameter, the E to I ratio, and actual neural coding. So how do we measure performance? The first measure that we use in the study is related to how well the stimulus is represented in the neural code. So it's the reconstruction error. The second measure we use is population density. It's basically the additive inverse of a well-known sparsity measure called the modified TR metric, which is a non-parametric statistic that quantifies the sparsity of the neural code, and it's often reported in experimental studies. We compute the population density only for the excitatory subpopulation of models. And the third measure that we use applies a metabolic energy consumption model to all neurons, excitatory and inhibitory, to estimate ATP consumption. So next, let's just take a look at the actual results and see how these performance measures change as E to I ratios vary. So the plot on the right is what we'll focus on first. The x-axis of this plot features the E to I ratio increasing from 1:1 to 9:1. And the y-axis of each of the three different subplots represent the three performance measures that we just discussed, but in a normalized form. And lower values are better, with 0 being optimal. So first, let's focus our attention on the solid line with the gray shaded band. We observe that as the E to I ratio increases, the reconstruction error decreases in the top subplot. The population density, which is in the middle subplot, follows a similar trend with sparser encoding as the number of E neurons increase. And all too conveniently, the metabolic energy consumption also decreases as the E to I ratio increases. However, the performance only improves up to a certain point, which I'll call the optimal E to I ratio. When a model goes past the optimal E to I ratio, its performance deteriorates rather rapidly. It's interesting to also observe that these performance measures are all optimal at similar E to I ratios. In fact, in this plot, the E to I ratios are identical, and they're indicated. You can see the connection with the dotted vertical lines. Additionally, we also see that the optimal E to I ratio happens to fall into ranges observed in biology. So that's kind of neat because that wasn't really anything we could control or configure. Now let's talk about the other dashed trend lines. We repeated our analysis for models trained at different sparsity levels, and those dashed trend lines reflect the performance of models at different sparsity levels. We find that the sparsity level of a model is actually positively correlated with the optimal E to I ratio, i.e., a sparser model actually requires fewer inhibitory interneurons to achieve optimal performance. This is kind of odd, isn't it? Shouldn't a sparser neural population need more inhibitory interneurons? And crucially, one wonders if this trend corresponds to biology at all. At this point, I should also point out that although we have a fairly specific theoretical prediction from a computational model, it's not otherwise informed or fitted to any kind of biological data. So such a specific prediction coming out of a theoretical model is kind of rare. So first I'll tackle why sparser models need fewer inhibitory interneurons. We began by examining the static structure of inhibition, and we found that models at higher sparsity have stronger inhibitory to excitatory synaptic connections than those at lower sparsity. So if you look at the figure on the left from the perspective of a single inhibitory interneuron, you can see that as sparsity increases, the weights are getting stronger. Figure 2, the figures on the right, the 2 figures on the right, capture the distribution of synaptic weights, which is the one on the left, which shows that models with higher sparsity levels have fewer synaptic weights which have a small magnitude. So most of the weights start getting stronger as models become more sparse. And this is compared to the models at lower sparsity levels. The rightmost plot just plots a summary statistic called kurtosis, which decreases as the synaptic distributions become less concentrated around 0. Next, we examined how inhibition actually flows over the static structure during stimulus encoding. And here what we found is that as sparsity increases, the diversity of activity in the inhibitory subpopulation decreases. This can be seen in the figure in the bottom row on the left-hand side. The variability as you increase sparsity decreases. So inhibitory interneurons in the inhibitory subpopulation are not firing at very many different rates. The bottom right figure reveals that through all the structural and functional changes owing to these changing sparsity levels, the balance between recurrent inhibition and recurrent excitation remains pretty stable, and the models, even though they are at very different sparsity levels, remain stable as a result. So even though so much is changing, the models continue to operate in a stable regime across all these changes. Lastly, I want to tell you about how our theoretical predictions match up against biology. So the plot on the right basically shows the relationship that you saw earlier, except it's a little bit more compactly represented. Earlier, we were looking at trend lines with different sparsity constraints. And here it is a single line which shows what the optimal E to I ratio is for a model against its sparsity. And you see there's generally a linear sort of relationship. But validating this computational trend against biology is pretty tricky. It actually requires comparing V1 population activity recorded across experiments in mice and monkeys or mice and cats. This is pretty hard because experimental studies are not really designed to facilitate interspecies comparisons typically. So things like how long the stimulus is kept on, how big the bin sizes were for spike sorting and things like that are different and hard to compare. And this makes it difficult to compare numbers that might even be published across studies. We had a hard time finding literature which allowed us to make reasonable comparisons. However, we've performed some preliminary comparative analysis on recently available large-scale data sets of V1 data from mice, monkeys, and cats. To our knowledge, this is the first analysis of its kind, and the preliminary results, and I caveat these as being preliminary results, the preliminary results support the model prediction showing that population sparsity activity in mouse visual cortex is higher statistically significantly compared to cats and monkeys. And we're able to show this not just with natural images, but also natural movies. So this seems like an early trend, which seems pretty consistent with our model predictions. So that's kind of neat. So I'll just I'm almost out of time, so I'll do a quick recap. If you can only remember one slide, this is all the pictures I'd like you to walk away with. I'm not going to walk through all of this, but essentially we asked how do structural properties that are conserved across species and sensory areas relate to neural computation. And it looks like they're related. And structural properties are not just related to neural computation, they're related to population activity levels. And it looks like we have some agreement between our theoretical predictions from our computational models and some preliminary analysis of publicly available experimental data. And we've got some interesting insights about how the structure of inhibition changes and how inhibition flows over this changing structure in different configurations that you might observe in biology. Unfortunately, we are not able to compare that to experimental data just yet. Hopefully, with better experimental technology and data sets, we can one day. I'd just like to acknowledge Chris and Ilya, who guided me through this project, and all the folks below who gave feedback or put their data sets online for public analysis. Without the data sets, we wouldn't really have been able to validate whether the model matches biology or not. And thank you all for listening. If you would like to read more about this work in detail, there is a lot of detail. You can check out our preprint. And otherwise, thank you for your attention.
Thank you, Arish. Very interesting. Yeah, I liked especially the fact that for your three measures, you found very similar looking ratios indeed. So we have maybe time for like one question or so if there is anyone. Otherwise, I have a little bit of a clarifying question. So when you talked about the metabolic measure, could you just remind me again how you calculated that? Because
you can think of it as, let me just pull up the formula. So you can think of it essentially as the sum of excitatory and inhibitory activity. And I didn't want to make this math dense. I almost ran past my 15 minutes. So I didn't put in how we estimate inhibitory activity. There's actually a fair amount of math that I'm hiding and a fair amount of detail. But essentially, we estimate the activity of inhibitory neurons, and we estimate the activity of excitatory neurons, which we can already tell from the stimulus encoding. And then there is some baseline cost, just like maintenance cost for the total number of neurons. And Atwell and Lawlin did some calculations based on which they said that you can approximate the inhibitory activity costs about as much ATP as excitatory neurons. It's not exactly correct, but they suggest that approximation is reasonable. So essentially, it's adding up the activity and scaling it in math, but the model does have some biological legs underneath it.
Okay, good. I think we're out of time though, so we probably need to stop at this point here. Thank you to Arish and thank you to all the speakers. And certainly not least, I do want to make sure to also thank you to Mitra, who's sitting in the background and making sure everything is working fine. And yeah, unfortunately, this is the end of it. I hope you all have a wonderful day and a continued